Am I ready to go? Yep. My name is John Axford, and I bid you a very warm welcome to the Jenner 14 Glycobiology and Medicine Symposium in the impressive and inspiring environment of the Rager Institute in Leuven. To orientate yourselves, we're in Belgium, we're to the south of Holland, we're to the west of France, and the east of, and the west of Germany, and the south east of the United Kingdom. The Rager Institute was the mindchild of Professor Piet de Sommer, and the buildings were initially built in 1954 with new buildings in which we're holding this meeting in 2017. The finances for this started from Joseph Rager, and that's interesting because this is in the same era as Edward Jenner, after whom these lectures, these meetings are named. And it's rather interesting that Piet de Sommer helped eradicate polio from Belgium and be one of the first countries in the world to have polio eradicated. And this is from his work in the Jewel of the Crown of Research and Innovation at the University of Leuven. This is a painting my daughter painted in earlier years, and I've used it ever since to demonstrate that innovation is blooming, and it is certainly blooming in the Rager Institute. So, who is Edward Jenner? Here he is sitting in around about 1780 in his drawing room in Berkeley in Dorsetshire with a picture of Bess the cow, whose fleece hangs in St. George's Hospital Medical School, and with whom he extracted cowpox serum to use as his vaccine. He was an English physician and scientist, and he pioneered smallpox vaccination, and he's known as the father of immunology, and he's known to have saved more lives than any other human since then. But things were not simple. He was the source of much contempt, and he was accused of witchcraft. So things do not change. Here he is with James Fitz inoculating. This is rather like an NHS vaccination clinic now, but you can see the concerned looks of the ladies around him. As I say, things have not changed. We have vaccine hesitancy with regards to COVID now. 27% of non-college educated multiracial adults in the USA are not vaccinated. There's rational reason for this, i.e. it may make your disease worse, such as lupus, and irrational concern from anti-vaxxers who have conspiracy theories and create a world of self-reinforcing truth. Perhaps we can discuss more about this with Greg Winter later on today. So, building upon the momentum of Jenner and following in his example, the Jenner Glycobiology and Medicine Symposia are at the cutting edge. They explore new scientific frontiers, and they focus upon young scientists as they work. The Jenner meeting is now in its 31st year, which must be some form of record, and we have travelled through 8 European countries before. We visited the USA for the first time last year, going to Harvard, and now we're in the beautiful city of Leuven in Belgium. So let me take you through a photographic travel through time. Our first meeting in 1990 was in Berkeley in Edward Jenner's home, and the quiz we can have here is, can you name any of the young scientists standing here? And I can see Ivan Reut. And is it intriguing that we were very well dressed in those days, and every man in that photograph is wearing a tie. We then moved to St. George's, through to the beautiful Il Ciocco in Italy, and then getting a flavour for meetings in the sun, we went to Loutraki in Greece. You can see hot countries mean large numbers of people attending. We then moved back to the Royal Society of Medicine, who have been our sponsors since 2000. Through to the beautiful Domaine de Sauillac in France, through to Exeter College and the spires of Oxford, and you can see Raymond Dweck and Jim Van Dyke in the front row there, Terry Merry, I can see, Tony Merry, and a few other notables. And then the impressive surroundings of University College Dublin and this superb lecture theatre, which we inhabited for a number of days in 2017. And then to the Royal Society of Medicine in the heart of Brussels, this imposing building. And then lectures on the beach in The Hague in the Netherlands, in Scheveningen. Through to back to Paris, the superb, sophisticated surroundings of the 16th arrondissement, the new Fondation on the left-hand side here, and then through to this beautiful lecture area in Dubrovnik, Croatia. And here we are in Harvard. We have 4 sessions today. They're in your program, and we have 12 speakers for you. We're breaking new frontiers. We are going to be talking about pivotal biological mechanisms, critical for our understanding of disease mechanisms, and the impact of these meetings and this meeting is going to be enormous. A big thank you to the local organizers at the Rega, Dominique and Inga, AV support, Philippe and the 2 Daves, and Emeritus Professor in Service, Ghislaine Obdenacker. A big thank you to the Scientific Committee, but really the driving force and the orchestration of Rogine O'Flaherty. And our inspiration over the years, Raymond Dweck and Ivan Reut. Thanks to my patient who runs Newcastle Fighters and has paid for all this. They are as diverse as sugars. We will be in Porto in 2023 under the care of Kelso Rees, and enjoy the meeting, enjoy discussing, planning, pushing forward the boundaries and collaborating. Many thanks. I will now hand you over to Kelso and Claudine. Have a great meeting and don't forget to ask questions.
Okay, I don't know if the audience can see us. Maybe we should move there. So, maybe we can start. Ladies and gentlemen, welcome to all participants of the 14th General Meetings, either the participants who are present or participants who are online. It's really a great pleasure for me to be together with you, and that's once again. Thanks to this hybrid formula, a possibility which was offered to us by Professor Ghislaine Obdenacker, who organizes this meeting in the Catholic University of Leuven. A wonderful place, a wonderful and impressive place for all glycobiology lovers, or glycomaniacs, as one of us used to call us. My name is Claudine Chieda, and I am pleased to co-chair this session, this first session on innate and adaptive immunity with Professor Celso Reis, who was kind enough to replace Professor Henrik Clausen, who just lost his voice, but he's with us by video, of course. This is the occasion for me to tell to the audience that the questions will have to be either direct for the happy few present, or the Q&A option for the online participants. Now, after this really very, very, very deep and very deep welcome, I wanted to open this first session by welcoming our first speaker, Professor Emma Reeves, who is the principal investigator at the Department of Medicine at the Irish Centre for Genetic Lung Diseases at the Beaumont Hospital at the Royal College of Surgeons in Ireland. Professor Reeves' research interest is centered on the elucidation of cell and molecular mechanisms in lung inflammation, especially about the role of the phagocytic neutrophils in respiratory diseases as cystic fibrosis and chronic obstructive pulmonary disease, with a central interest, which is illustrated by the title of her talk, on alpha-1 antitrypsin deficiency. So, Professor Reeves, I hope that we are all together, yes, and the floor is yours. We are eager to listen to your talk. Well, we cannot hear you. Okay, here we go.
Sharing now?
Yes.
So can everyone see the screen?
Yep.
Mm-hmm.
That's
yes.
Great. So good morning, everyone.
Thank you.
I'm delighted to be here. I'm delighted to have this opportunity to share some of our results, and also delighted with the opportunity to present, and I thank the organizers. I particularly like to thank Roisin O'Flaherty for her excellent support over the last number of weeks. So as mentioned, the title of my talk is Glycosylated Repurposes Alpha-1 Antitrypsin for Resolution of Community-Acquired Pneumonia. So first of all, I included this slide as an introduction to alpha-1 antitrypsin. Alpha-1 antitrypsin is a 52 kDa glycosylated protein. It is produced by hepatocytes. And your liver actually produces 2 grams a day of alpha-1 antitrypsin, and it releases it into the blood circulation. Serum levels of alpha-1 are approximately 27.5 micromolar in healthy individuals. Monocytes, macrophages, neutrophils, and eosinophils, and also airway epithelial cells can also produce alpha-1 antitrypsin, but at extremely low levels. Alpha-1 is an irreversible antiprotease inhibitor of neutrophil-derived serine proteases, including neutrophil elastase, cathepsin G, and proteinase 3. It plays a key role in protecting the lower respiratory tract from activated and disintegrating neutrophils. The structure of alpha-1 is key to its function. In the image on the right-hand side, you can see the ribbon structure of alpha-1. The alpha-1 peptide is in light blue. The reactive center loop is in red at the top of the molecule, and the reactive center loop is key for the antiprotease activity of alpha-1. Alpha-1 is glycosylated. N-linked glycans are illustrated in dark blue at residues 7107 and asparagine 271. As I've mentioned to you, alpha-1 is a key antiprotease, but over the last number of years, there is increasing evidence that alpha-1 is a multifunctional protein and modulates cells on a number of different levels, including immunity, inflammation, cellular apoptosis, and also senescence. Our own work within this field has shown that alpha-1 can regulate neutrophil chemotaxis, and it does this by binding IL-8 or CXCL8. We have also shown that alpha-1 modulates neutrophil degranulation, and by doing this, it regulates autoimmunity. Alpha-1 does this by modulating TNF-alpha signaling and directly binding to TNF-R1 and TNF-R2. If we go back to the ribbon structure on the right-hand side, there is a core hydrophobic pocket located between S2A and his D and E on the alpha-1 molecule. This hydrophobic pocket on the alpha-1 molecule binds to LTB4, leukotriene B4. When bound to this hydrophobic pocket, LTB4 cannot signal, and therefore alpha-1 can modulate neutrophil adherence in response to LTB4. So a lot of research has been developed over these years that is confirming that alpha-1 is a potent anti-inflammatory molecule. Now, if we turn our attentions back to the N-linked glycans associated with alpha-1, as I've mentioned to you, it is N-glycosylated at 3 asparagine residues at position 70, 107, and 271. There is considerable diversity at site 107, which gives rise to different glycoforms. The N-glycans of alpha-1 are composed of sialic acid, mannose, galactose, fructose, and N-acetylglucosamine, which exist as bi-, tri-, and tetraantennary structures on the native alpha-1 molecule. Now, I thought I'd use this opportunity and this slide to introduce you to alpha-1 antitrypsin deficiency. Alpha-1 antitrypsin deficiency is a genetically inherited codominant condition. It arises due the Z mutation. A Z mutation is the most severe manifestations of the alpha-1 antitrypsin deficiency, and it results in the switch of glycine to glycine at position 342. The Z alpha-1 protein is misfolded and it accumulates in the endoplasmic reticulum of hepatocytes. As a direct result of this, individuals who are homozygous for the Z mutation have less than 10% of circulating levels of alpha-1, and the 10% that is circulating is Z mutant alpha-1. The carrier frequency of alpha-1 deficiency is 1 in 25 in Ireland, and it is estimated that there are over 20,000 individuals who are homozygous for the PiZ phenotype. So as I've mentioned, alpha-1 antitrypsin deficiency is characterized by early onset emphysema. However, it can also lead to liver disease. The liver disease is due to accumulation of the misfolded Z protein in the hepatocytes. It can also lead to paniculitis, where the Z protein is accumulating in the skin. So alpha-1 deficiency is diagnosed through phenotyping. And in this gel on the right-hand side, it's an IEF gel, an isoelectric focusing gel. And it is run over a pH gradient of 4.2 to 4.9. There are 3 samples on this gel. The first sample in the left is an MM, so that's a healthy controlled individual who has the MM phenotype of alpha-1. We can see that we have labeled these M glycans as alpha-1 as M2, M4, 6, 7, and 8. The concentration of alpha-1 in this healthy controlled individual is approximately 1.5 grams per liter. On the right-hand side of this IEF gel, we have the glycans that are in the plasma of an individual who is homozygous for the Z mutation. The glycans are called Z2, Z4, and Z6. You can see there's an awful lot less of it. In the middle lane, we have a person who's heterozygous, who's a carrier for the Z mutation. And you can see that this individual has 0.9 grams per liter in their plasma, and they present with both the M glycans and the Z glycans. So one treatment for alpha-1 antitrypsin is alpha-1 augmentation therapy. So augmentation therapy for people who are deficient is with purified human plasma alpha-1. So the alpha-1 is purified from human plasma. And it has been shown that this is a treatment for lung disease associated with alpha-1 antitrypsin deficiency. Alpha-1 antitrypsin augmentation therapy is delivered intravenously at 60 milligrams per kilo weekly, and it has shown to be safe and well tolerated. This treatment has been shown to increase plasma levels of alpha-1 and also to increase epithelial lining fluid of alpha-1, and has been shown to reduce loss of lung tissue damage. In a rapid study, which was published by The Lancet in 2015, it was a double-blind, placebo-controlled trial. And the results of this are illustrated in the right-hand side. In this graph, those who were receiving plasma-purified alpha-1 are in the black bar, and those that were receiving placebo are in the hatched bar. You can see that the annual rate of decline in patients who were receiving alpha-1 augmentation therapy was minus 1.5 grams per liter per year, but those who were receiving the placebo was minus 2.26 grams per liter per year, illustrating the benefits of alpha-1 augmentation therapy. But as you can imagine, plasma purified alpha-1 is extremely expensive, and therefore people have been trying to develop new treatment options. One of these was the production of recombinant alpha-1. However, recombinant alpha-1 was not glycosylated and therefore was secreted from the body rapidly. These results indicated to us the importance of glycosylation on the alpha molecule for retention within the body. Now, in the lab here in Beaumont, we run the National Alpha-1 Screening Programme. This means we get samples from all over Ireland to screen for alpha-1 antitrypsin deficiency. And what we noted over the years was there's a great variety in the glycans of alpha-1. And in this IEF gel, you can see that I have run 6 samples from 6 donors, so 6 plasma samples from 6 donors. They are all PIMM, so they are not alpha-1 antitrypsin deficient. However, what we can see on the left-hand side is that there's a great variation in the glycoforms. We have M0, M1, all the way down to M8. However, this M0, M1 glycoforms were only present in individuals in samples 1, 3, and 4, but are not apparent in samples 2, 5, and 6. So we didn't understand this. We were wondering, why do some people have these M0M glycans and others don't? So we went over to Professor Pauline Rudge and Dr. Radka Fahy, and we knocked on the door in the NRBRT Glycoscience group, and we had a great meeting. We discussed these results. And at the end of the meeting, we had 2 further questions. So does the expression of sialylated alpha-1 antitrypsin change over time? And are increased expression levels of sialylated alpha-1 required for resolution of acute inflammation? So to answer this question, we employed community-acquired pneumonia as a model of acute inflammation. And the aim of the study was to investigate alterations of alpha-1 glycan signatures during the time course of acute infection and after resolution. So patients were admitted to Beaumont Hospital here in Dublin, and we had 11 patients recruited onto the study. Our inclusion criteria indicated radiological evidence of pneumonia, clinical diagnosis of pneumonia with a curb score 65, score less than or equal to 2, and an age greater than 18 years of 18 years. So over the course of the pneumonia, over a course of 0, which is the day of admittance to the hospital, and after 2, 4, 6, and 8 days post-antibiotic therapy, we obtained a blood sample. We ran this blood sample for the white blood cell count, which is in panel A. We also measured CRP levels, C-reactive protein in panel B, and as alpha-1 is an acute phase protein, we also measured its levels in panel C. You can see in panel A that the white blood cell count on day of diagnosis, that is day 0, was approximately 19 by 10 to the 9 cells per liter. And following antibiotic therapy on day 8, this significantly reduced into the healthy control range. In panel B, we have significantly elevated CRP on day 0 at approximately 200 milligram per liter, and this fell with treatment to approximately 60 milligram per liter on day 8. Finally, in panel C, the alpha-1 levels fell over the time course from 2.86 grams per liter down to just above 2 grams per liter. So we concluded that community-acquired pneumonia, referred to as CAP, is an excellent model to explore the alterations of alpha-1 glycans during the time course of acute infection. So what we did first of all was to run the samples out by isoelectrofocusing. So once more, the blood samples from the patients on day 0 up to day 8 were run out on an IEF gel, a pH gradient of 4.2 to 4.9. And what we saw for all 6 samples that we ran like this, that on day 4 and day 6, there was an increase in the M0, M1 sialylated alpha-1 glycoforms. I don't know if you can see my cursor, but I'm indicating it there. So there was an increase in the M0, M1 sialylated alpha-1 glycoforms. And this intensified on days 4 and 6 and resolved with clinical recovery of CAP on day 8. Now, ST6-GAL1 is a sialtransferase that is upregulated in hepatocytes during inflammation and is under the control of IL-6. In this graph, you can see that IL-6 on day 0 before treatment was approximately 62 picogram per mil, and on day 8, it fell to approximately 10 picogram per mil. When we exposed primary hepatocytes to IL-6, and we used IL-6 50 picogram per mil, as this is the recorded level 48 hours prior to the occurrence of the M0, M1 glycan forms. And you can see at 48 hours, there's a significant rise in the ST6-Gal1 on a gene level. Correspondingly, at 48 hours of exposing the hepatocytes to IL-6, we saw a significant increase of ST6-Gal1 protein. Moving on then, we wanted to confirm that there was an increase in silation over the time course on alpha-1. To do this, we worked well and extremely well with Dr. Radka Fahy, and what we did was we purified the alpha-1 using alpha-1 select resin. We carried out wax HPLC to establish the sialic acid content of the alpha-1 glycans. And immediately you could see at day 4 and day 6, there's a significant increase in the glycans containing 4 sialic acids. Indeed, there was a 3-fold increase in the mean percentage of glycans containing 4 sialic acids. On day 0, this was 0.61, compared to day 4, which is 1.63, and it went up higher again on day 6 to 1.66. So we confirmed that there is a glycan shift occurs in the alpha-1 between days 4 and day 6, which resolves on day 8. And these time periods are in keeping with the resolution of information as measured by C-reactive protein and also levels of the white blood cell count. However, although we've confirmed that there is a glycan shift, we were unsure what was the purpose of this glycan shift and was it involved in resolution of inflammation, which is what we next explored. First of all, it was important to understand whether the silation of alpha-1 affected its antiprotease activity. This was key because remember, the antiprotease activity of alpha-1 is its main function. So to do this, we purified alpha-1 protein during the acute phase on day 1 of community-acquired pneumonia. And this is illustrated in the IEF gel. So on day 1, it's the acute phase alpha-1, and we refer to this in the talk as AP alpha-1, acute phase alpha-1. We also purified resolving phase alpha-1, or alpha-1 during the resolving phase of pneumonia on day 4. So that's AP alpha-1 and RP alpha-1. And these are both run in the IEF gel in panel A. You can see that the resolving phase alpha-1 had sialated M0 and M1, but these were not apparent in the AP alpha-1 fractions. We carried out a neutrophil elastase FRET assay to compare the anti-NE capacity of AP alpha-1 to RP alpha-1. And you can see on the graph on the right-hand side that the IC50 values are identical. So the increased sialation that we observed during resolution of CAP, did not affect the antiprotease activity of the alpha-1 molecule. So we wanted to understand what was the cause or what was the function of having this increased sialation of alpha-1, and did it bind to inflammatory mediators within pneumonia? So to do this, we immunoprecipitated the AP alpha-1 and the RP alpha-1, and we profiled the binding partners ex vivo. We pay particular attention to CXCL8 or IL-8, as IL-8 is a potent neutrophil chemokine. So we immunoprecipitated the alpha-1, and we then carried out an ELISA for CXCL8. And in the graph on the right-hand side, you can see there were significantly higher levels of RP alpha-1 bound to CXCL8 compared to the amount of CXCL8 that was bound to AP alpha-1. Indeed, CXCL8 bound to AP alpha-1 was 0.9 picogram per mil, and this was compared to 0.34 picogram per microgram for the RP alpha-1. So significantly higher levels of IL-8 were bound to RP alpha-1. This set of experiments then were extended to look at the binding profile of CXCL7, also referred to as NAP2. NAP2 is a second potent neutrophil chemokine. And in the graph on the right-hand side, you can see that we found significantly higher levels of CXCL7 bound to RP alpha-1 compared to AP alpha-1. Levels change from 2.9 picogram per microgram of alpha-1 to 2.2 to 2.9 picogram per microgram of alpha-1. So we wanted to confirm these results in vitro. And to do this, we carried out slot blot analysis using HSA as a control. In this experiment, you can see that RP alpha-1 in the black bar found significantly high levels of CXCL8 compared to AP alpha-1. Indeed, there was a twofold increase in the binding capacity of CXCL8 to the RP alpha-1. Moreover, we found a threefold increase in the amount of CXCL7 binding to RP alpha-1 compared to AP alpha-1. The results so far indicate that both CXCL8 and CXCL7 bind sialylated alpha-1 during resolution of a community-acquired pneumonia. However, at this point, it was unclear if this binding event was required for resolution of inflammation. And that is what we next explored. So we turned our attention to neutrophil migration. And in this assay, we carried out a chemotaxis assay, and we looked to see whether RP alpha-1 and AP alpha-1 could inhibit neutrophil chemotaxis in response to CXCL8. The neutrophil chemotaxis was carried out using purified neutrophils from healthy controls in a Boyden chamber. The x-axis on this scale is the chemotactic index. And you can see that in response to CXCL8, there's a significant increase in the level of cells that are migrating with a chemotactic index approximately 1.8. However, RP alpha-1 was found to significantly reduce the chemotactic index compared to IL-8 on its own, and with a p-value of 0.001. In comparison, AP alpha-1 was less significant at reducing the neutrophil response, with only a 37% reduction in the chemotactic activity. Moreover, in response to CXCL7, the RP alpha-1 significantly reduced neutrophil chemotactic activity. So this slide did indeed confirm that there was a reduction in chemotaxis. The mechanism of inhibition remained unsolved. So to find out more about this, we carried out flow cytometry, and we investigated whether the binding of CXCL8 and CXCL7 to AP alpha-1 prevented engagement of these chemokines to CXCR1 and CXCR2 on the neutrophil membrane. By flow cytometry, you can see that the percentage of positive cells in response to the addition of CXCL8 is extremely high, but RP alpha-1 reduce the engagement of CXCL8 to CXCR2 on the neutrophil membrane. Moreover,
1 minute left.
Thank you. I'm just there. And in addition to that, we found that AP alpha-1 also significantly reduced the binding ability of CXCL7 to the neutrophil receptors on the membrane. So in summary, results indicate that alpha-1 glycosylated is altered acutely. We found that sialylated alpha-1 functions to modulate inflammation. Sialylated alpha-1 during the resolving phase of pneumonia binds CXCL8 and CX7 and modulates neutrophil migration. This shift in the function of alpha-1 has implications for resolution of and re-establishment of homeostasis and resolution of acute phase of inflammation. These results also indicate the therapeutic potential of sialylated alpha-1 for a range of neutrophil-driven inflammatory disorders, including neutrophil-driven rheumatoid arthritis and chronic obstructive pulmonary disease. So in my acknowledgements, I'd like to thank Professor Jerry McIlvanney and the team in the Irish Centre for Genetic Lung Disease, and also in the NIBRT Glyco Science Group. Of course, I'd like to thank Professor Pauline Rudd and Dr. Radka Fahy. My colleagues in the National Institute of Cellular Biotechnology in DCU were also key in the identification of the alpha-1 binding partners. I'd also like to thank my funding bodies. So thank you, and I'd like to ask any questions.
Thank you very much. It's a very, very nice talk, and we have a question for the Q&A, which is coming from Daniel Koterich, and who says that he thanks for the really interesting talk. And did you investigate? Wow, what is that? There is a problem with the sound. Did you investigate the level of sialic Lewis X on AAT during the resolving phase of pneumonia?
So if I understood the question correctly, so we analyzed the percentage of sialylated glycans, and it was total sialylated glycans, and we did see an increase on day 4 and day 6. So we understand there is much more to this project, and we are looking to advance into other acute phase diseases, and those are questions that we will need to answer.
Thank you. Unfortunately, we have to move on. There are other questions, but we have to move on. So maybe you can be able to answer to the questions writing.
Thank you. I will do indeed. Thank you.
Thank you very much. So it's time to welcome the next speaker, who is Professor David Alsteins from the Catholic University of Louvain-la-Neuve, and who is going to explain to us how we can use the nanobiophysics. He's at the nanobiophysics labs, and how we can use the nanobiophysics methods to develop a new visualization by the atomic force microscopy, the famous AFM, at the level of the molecular interactions, as well as at the level of the cellular phenomena and interaction. More precisely, he will speak about the role of glycan binding during viral attachment to cell surface using AFM. We have very, very interesting ways of derivatizing the model of AFM interactions, measurements. Thank you very much. You are welcome. Waiting for your talk.
Can you hear me?
Yes.
But I think you cannot see my.
We cannot.
You cannot see me. You see my screen, right?
We see your screen, but we cannot see you, which is a shame, really.
Yeah, I know. I just try. I try to relaunch my camera, but it seems to does not work. So unfortunately, I think I will have to do it without my camera. Which is a little bit.
Yesterday it worked.
Yeah, yesterday it worked. I know. That's really weird. So anyway, I will try to explain it clearly. Okay. So, my lab focused on the atomic force microscope, and we try to study interaction at the single virus level. So, to introduce a little bit this study, I like to show this cartoon. So, as you see here, in the context of virus-host interaction, the cells impose multiple barriers to the virus entry. However, the virus exploit the fundamental cellular process to gain entry to cells and in turn deliver the genetic cargo. So virus entry, and we believe this in our lab, is quite largely defined by the first interaction that can take place between a single virus here and the cell surface receptors. So those interactions, they will determine the mechanism of virus attachment to the cell surface, the uptake, the intracellular trafficking, and ultimately the penetration to the cytosol. And there is mainly two kinds of interaction that may occur with the cell surface. So it's the interaction with the attachment factors that are seen to help the virus to concentrate on the cell surface. And such interaction are believed to be quite non-specific. And then there is the viral interaction with the cell surface receptors that actively promote the virus entry. They can do so by initiating some conformational change into the virus particle, by activating some signaling pathway and by promoting the endocytic internalization. So how we try to study a virus-host interaction is to use single virus force spectroscopy, which is a method based on the atomic force microscope. And this technique can be comparable to walking your dog. So you just try to hang on a virus on a leash, and then you let the virus or the dog walk around and explore the area. And suddenly the dog or the virus can bite its prey. And then what you will do, you will just pull back on the leash, and you will be able to monitor or to feel the force that acts between the virus and the cell surface. Of course, we are not working with a leash. We work with an atomic force cantilever and tip AFM. So, and there we use a leash, which is just a polyethylene glycol linkers that enables us to graft a single virus particle to the very end of the tip apex. So here you see, this is the AFM cantilever, the tip apex. You have the linker, which is based with a peg linker that enables to the virus to freely diffuse into the solution. And then you can check, for example, by confocal microscopy, that you have single particle at the very end. And you can see here that the cell surface receptors, and then you can then probe the interaction with the cell surface receptors. To measure those interactions in physiologically relevant condition, we use this setup. So this is the AFM cantilever with the tip apex here. You have to imagine that you have a single virus particle at the very end. And we have all this setup directly under optical microscope on the top of living cells. And to ensure that the cells are alive, we work in an environmental AFM chamber by controlling the CO2, the relative humidity, and the temperature. And as you can see here on the movie, I will try to relaunch the movie. You can see that the cells divide under the microscope, and you see here the histone that's at some point disrupt to have the two of the cells. So basically, you here have the DIC image of your setup. You approach your tip on the top of the cells, and to have a kind of internal control, we always try to mix two different kind of population of cells: the cells that express the receptors that we try to target, and the cells that does not express. And here you have fluorescently labeled red cells that express the TVA receptors. On the AFM tip, we have an envelope rabies virus, and we probe the interaction. Here you see the overlay with the fluorescence image. This is the EFM8 topography image. So you just scan the sample here with your tip, and for each pixels, each point, you record the age of the cells, and you see here the area where there is the nucleus. And this is really the topography of your mammalian cells. These cells are MDCK cells. And together with the 8 image, you also acquire the adhesion channel. When you have a black pixels, it means that there is no adhesion. And if you have a gray or white pixels, it means that you measure some binding between your virus and the cell surface. And the color codes for the force. And you see here that we record between 0 and 250 piconewton of forces. So this is in the range of expected value for bonds, ligand receptors bonds, such as antibody and antigen and so on. And as the control, you have seen here that those cells does not express the receptors. So these two cells, we expect to observe no interaction. And here on the adhesion channel, we observe no interaction on those cells. You can go a little bit further into the analysis. If you zoom on single cells, you can clearly see here some other shape of the cells. And you see that all the adhesion even, they are located on the cells that express the receptors. And if you extract a single pixels, and if you look behind this pixel, in fact, what you measure is a force distance curve or force versus time curve. And this is exactly the movement of your EFM tip. So you approach your tip to the cells. At some point, you touch the cell surface, you press a little bit on the surface, and then you retract your tip, you try to disrupt the tip from the surface. And if there is an unbinding event, you can observe...
Excuse us, we have a problem. The slides are not passing.
Oh, that's a big issue. So I will try to...
That's a big issue.
Yeah.
Because we are stuck at the Same slide.
I
Oh,
will try to relaunch.
now we have got you.
So you can see me, of course.
Mhm.
Okay. I will try to just relaunch. So I stopped the party. Yeah, there is an issue. I don't know why.
Pixels. Yeah.
I cannot share the screen again. I'm very sorry about that.
Okay, so we were at the.
And we've got you as well.
You have both.
What can you see now?
Yes.
Yes.
You and now you see my full screen.
Yes.
Yes.
And you see the mapping now.
Yes.
Which slide is it at the moment?
Well, the binding to cell surface glycans is tightly regulated.
Okay. It's very weird, you know. So I was at this slide. So I tried to show you that, you know, that's we try to observe the interaction between the tip and the surface. And you can see that there is a fluorescence image where the cells express the receptors, and on the adhesion channel, there is the single bonds or the adhesion event that are in gray pixels over black pixels. So black pixels mean no interaction, pixels means an interaction event, and you can really locate it, the unbinding event, over the cell surface. And then we have the force system curve. This is the actual movement of your tip on the cell surface. So you approach your tip and then you retract your tip. And then at the unbinding event, you clearly see the rupture events. You see that there is multiple peaks here. And these peaks are the real unbinding event between your tip and your sample. So the unbinding between your virus and the cell surface receptors. Here, for example, you observe like 3 individual rupture bonds, meaning that during a tip contact with the cell surface of around 1 milliseconds, the virus is already able to establish 3 individual bonds. After that, you can extract all this, the physical parameter behind this interaction. I will not enter too much into details, but the individual points here are the unbinding events, the force versus the loading rates. The loading rate is related to the speeds at which you unbind the event. And what you see is that we can fit those data and extract kinetics properties of the bond. So you can extract the off rates, something that show the stability of the bond. You can also extract parameters that describe the energy landscape of this interaction. And also you can use different models to look at the cooperativity of this measurement. So if you have multiple bonds, you can try to predict if those bonds are all similar, so they are just parallel bonds, or if there is some kind of cooperativity between those bonds, and if the affinity for a second bonds, it's much higher than simply 2 parallel bonds. I will try to explain in a few minutes now the different mechanisms that we have found and where glycans are involved. A first example, it's the binding of herpes virus. So this is a murine herpes virus that binds for the first step to the attachment factor that are GAGs on the cell surface. And for these steps, there is 3 different glycoproteins that are involved into this binding. If you look into the literature, there is the GP150, the GHEL, and the GP70. There is not exactly the roles of each protein for this step. So we try to use our atomic force microscope to determine the roles of the GP150, which is the major viral glycoprotein on the murine herpes viruses, and that has been shown to have a low affinity for GAGs in vitro. So we look and compare 2 different viruses, the wild-type viruses and the GP150 minus viruses. So this is the virus where the GP150 has been deleted. And we observe exactly the same pattern. So there is no difference of the ability of the virus to bind to the cell surface receptors. This is done first in vitro on purified heparin sulfate, but we also monitor the interaction on living cells. And from a point of view of binding, we do not observe any difference. We check very carefully into details. We use also different mutants, GP minus 70 minus, or the incubation with antibody. And what you get here is just that you can explain all the binding just by the influence of GP70 and GL. And of course, you measure several parallel bonds, meaning that all these glycoproteins that are expressed at the cells, at the virus surface, just are involved and able to do multivalence bonds on the cell surface or with the cell surface receptors. So, but what we observe is, if you look very carefully to the results, is that the pattern or the number of bonds is not similar. If you compare the wild type and the GP150, what you observe is that the wild type, most of the interaction that you measure are in the very low forces range. So that means that this virus mainly maintains a low number of parallel bonds with the cell surface. So it seems that even though the GP150 is not directly involved into the binding to the GAG, we observe a role more for as a regulator of the number of bonds that the virus can establish in the first binding step to the cell surface. And this can, of course, have a huge influence for the entry, but also for the release of the virus. And if you look here on this TM picture, we observe that for the GP150, there is a deficient release of the virion that have been replicated within the cells, mainly due to the fact probably that this virus established much more interaction with the cell surface gags. And this can also have influence for the virus diffusion on the cell surface, or also the entry when the virus looks for the specific receptors. I think just in a few slides, I will also try to show that for other viruses, this is the real viruses, we try to understand how a virus can combine the interaction with the GAGs or with the glycosylation, and then promote some specific interaction with specific receptors. So we focus on the reovirus. Reovirus is not recognized as a common pathogen, but it has been shown to have a role in triggering of the development of the celiac disease. And this virus possesses many proteins at its surface, but we will focus mainly on the sigma-1 protein. That is a trimeric glycoprotein, and which possesses two binding sites. A binding site for the specific entry receptors, which is the GMA at the very head domain, and then a binding site for the alpha-linsilic acids. So we probe the interaction for these two binding sites. First, the interaction with the salic acid, and we try to monitor this first on model surfaces. So you can extract all the parameters that describe this interaction. We did, of course, several controls. So if you look at the interaction with the salic acid, you observe that you have quite a huge binding frequency. If you do a single point mutation into the binding sequence, you observe that you decrease significantly the binding frequency. And then if you do co-incubation assays with salic acid, you also observe a decrease of the binding frequency. If you inject much more complex salic acids molecules that possess the alpha-linsilic acid, you also decrease the binding frequency, which is not the case if the alpha-linsilic acid is not present in these molecules. So we did the control on cells. As I mentioned, we always try to co-culture different cells to have an internal control. And here the cells that does not express the receptor, the salic acid, does not bind to the virus. So only the cells, the fluorescent cells here that express the salic acid can bind to the virus. And then on cells, you can monitor and check the number of bonds that are established between the virus and the cell surface. You can do that also for the specific entry receptors, the GMA. So we extract exactly the same kind of parameter, first in vitro, and then on living cells, you extract the number of bonds. And then we came to the point that this structure is quite particular, having the first attachment factor more closer to the virus body and not at the head terminal part. And so we try to understand why is this case for this virus. And then we try to combine both experiments. So monitoring the binding to the entry receptors while injecting the salic acids. And this, what we observe is that when you incubate the virus with the salic acid, you have a huge increase of the force. So this means that you have much more multivalence bonds between the virus and the specific receptors when the salic acid, so the attachment factors, came into the game. And this has been controlled with injection of LSTA and LNT that does not interfere with this binding. So this seems to be a very innovative mechanism that the first binding step to the salic acids change the conformation of the sigma-1 protein, probably to a more extended conformation that increase then the avidity for the specific entry receptors. And this is also confirmed by a test with the ASVP, which is an intermediate subvirus particle that has been shown by cryo-EM that the sigma-1 proteins directly more extended onto this virus. We also validate this kind of experiment by other techniques like SPR or biolayer interferometry. We also did single particle tracking, and we observed that the number of virus bound to the cell surface or the diffusion of the receptors is directly affected by the presence of the attachment factor, and then you can modulate the number of virus bonds or the diffusion of this virus by co-injecting the sialic acid. So this came with a new kind of model where each monomer of the sigma-1 protein can independently and that the number of established bonds, of course, but it also exists a cooperativity between the attachment factor and the specific entry receptors, where the alpha-linked sialic acids will modify the binding to the GEM-A receptors. And so this brings me to the end of my presentation. I hope that all the issue with the connection did not interfere with the message of this presentation that showed that atomic force microscope can be a very useful tools to probe and monitor the first binding steps between a virus and cell surface receptors, and that we show that there is a huge importance of the attachment factor during this first step. And so I would like to thank all the people in my lab that have been involved into this study, the collaborator, in particular Professor Laurent Gillet from the University of Liège for the work with the herpes virus, and Terry Dermody from Pittsburgh for the rare virus. And I will be very happy to answer any question.
Thank you very much. It's very, very, very nice talk. So it's a very impressive method and a very impressive new ways of analyzing the interactions. Are there questions in the audience? Because I don't see any written question. Question of the audience first? Yeah. Yes, I can. Okay, I can ask you the first one, and then okay, but for practical reasons. Yes, I would like to ask you whether in this type of interaction, can you, as you can really see the difference when you have the sialic acid at the same time, etc., that you change the conformation. Can you make the difference between the change in conformation or the change, or let's say, make the difference between the regroupment, the grouping of the sites of the receptors, and the change in conformation in the avidity that you described?
So in the particular case of the virus, it's a non-enveloped virus. So the sigma-1 protein cannot diffuse on the virus. So the fact that we could, for example, observe some regroupment of the sigma-1 protein cannot occur. And now what we did, just to be sure, this is something that is under progress, we tried to push the regroupment of the trimeric spike by doing some cysteine residues that will covalently attach the head domain together. And we observed that by putting together the head domain, we observed the same behavior. So showing that it's really a change of this conformational of the individual sigma-1 into this trimeric form.
Well, in terms of not cells, but molecules, how low can you go into the derivatization of the cantilever? How many molecules? What is the lowest number of molecules you can?
So we dilute. This is a chemistry point of view. We can dilute a lot, and so finally we can reach a single molecule. So we did some experiment, for example, with GPCR on model surfaces, and we can really, for example, put a small peptide, for example, 27 amino acids at the very end of the apex, and then monitor these single bonds events.
Okay. Thank you. Oh, there is a question here. Yes. So from Goran Larsson, asking, does this elegant technique be used for measuring attachment in addition to the detachment force?
Yeah, in fact, what you look is a little bit a mixture in between, because you just put your tip, approach your tip, then you measure the binding. There is the binding of the virus, and suddenly you can disrupt this binding. So we believe that if you put the virus in contact for less than 1 millisecond, this behavior is very, this time is very short. So what you probe, in fact, is the binding, and then you disrupt. And then what you can do is you can extend the binding. So you can leave the virus for 2 milliseconds, 1 second, 10 seconds, 1 minute, and then you will start to probe the increased process of the binding, even start to monitor, for example, the diffusion or the recruitment of other receptors. And then you can have a huge window of the mechanism of binding of virus here in the context of virus-host interaction. Yeah.
That's great. That's absolutely fascinating. New approaches. Thank you very much. I pass the phone to Pauline.
Thank you very much. That's a really elegant talk. I just wondered, is that technique capable of looking at the strength of binding and whether it's multivalent? Because I'm thinking, is it possible that this could allow a virus to kind of crawl across a cell looking for its main receptor, rather like the flu virus does with, the sialic acid binding site? I mean, could it be that it doesn't just attach it? Maybe it attaches, detaches, and then moves on to a different receptor, or you get many binding sites on one virus, and that makes a sort of much stronger attachment. I mean, can you look at any of those things with AFM?
This is a very nice question. So this is something that we, at the very beginning with the atomic force microscope, as I presented to you now, it's very difficult. So we try to increase the length of the linker to allow the movement of the virus a little bit more, but that's quite very difficult. So we changed a little bit the mode. I didn't have the time to show it here, but what you can do, you can functionalize not AFM cantilever, but you can functionalize a beads. So you use kind of very small beads around 200 nanometers, and then you put a single virus on it. And this will allow you to look at the movement of the beads on the cells. And then you use a kind of microfluidic device, and then you can then pull onto these beads, then you pull onto the virus. And what we observe is that the virus was able to diffuse on the cell surface and then also induce signaling into the cells. And you can even show the recruitment of clathrin within the cells. So we believe by this combination of technique that you can probe, as I showed it to you today, the first binding events, but you can look also at longer term, show the diffusion of the virus, single particle tracking, as I show a few slides here, and then look even at the first signaling to the cells and the recruitment of intracellular protein toward the endocytosis of the virus.
Yeah,
okay.
No, that's really interesting, because then you can think about the geometry of the binding and how many receptors you need to have on the virus in order to be effective. I mean, there's so many other questions you can ask if you can get that kind of information.
Yeah,
it's excellent.
Yeah, it's a little bit our goal, yeah, but it's we have to always push a little bit more the techniques. So the techniques show that we were able to measure the first bonds at the first binding step, the first milliseconds, and now the idea is to go for long term and check at all the individual step, the number of bonds that are established and how those bonds are regulated or increase over the time or decrease even at some point. I don't know. That's something that we are trying to look, and also searching for the best model for because each virus or different type of virus have different way of entry.
Okay, so we have to. Sorry, Pauline, and sorry, David. I'm very sorry because it's fascinating, really. Thank you so much. There is another question, but I will ask you to answer by typing. Thank you again, and now I pass the phone to my co-chair. Thank you. I pass the phone to my co-chair, Professor Celso-Reyes.
Thank you very much, and it's a pleasure to co-chair with Claudine this morning. Well, we go for the next speaker is Dr. Elisa Fada. She's a senior lecturer at the Department of Chemistry, Maynooth University, and she's going to talk about the fine-tuning the spike, the role of the glycan shields, nature and topology in the SARS-CoV-2. Elisa.
Hi, everybody. Just gonna share my screen 1 second. There you go. Can you see my slides?
Yes.
Can you?
Yes. Perfect.
Great. Thank you very much. Thank you very much for the invitation. And it's actually lovely to see an audience, a live audience. So first steps, little steps. So I'm going to start by with the slides, which shows like our reconstruction of the spike proteins from the coronavirus 2 at the atomistic level of detail, who features on the cover of the New York Times about last year. So we've been really busy trying to understand how this virus infects our cells at the atomistic level by using a combination of computational approaches. So defining the structure and the dynamics and the role of the glycans in the infectivity of the SARS-CoV-2 virus. So as a recent or more recent, let's say, insight on this effects is that the spike is involved actually in binding host cell glycans. In particular, the work from Jeff Esco, has highlighted that the heparan sulfate interaction is essential for infection, also supported by work by Mark Skidmore, and more recently, actually, by other work from John Klassen and Matthew McCauley and colleagues from the University of Alberta, who highlighted that the spike doesn't only bind GAGs, but it also binds cyalylated, in particular monocylated gangliosides. So it's a very, let's say, rich interactions that the spike has with the host cell, and not only involved the main receptor ACE2. So we've been looking at that amongst other things, and this is where I'm going to end up today with in my talk. But the actual essence of the talk, it's the role of explaining the role of the glycan shield in the spike activity, and how the evolution of this glycan shield allowed for this spike in particular to interact with host cell glycans as co-receptors. So within this framework, I'm going to give you a quick recap of the SARS-CoV-2 architecture, the spike architecture, and how the glycosylation at specific sites affects S activity, which was one of the main discoveries of our work published last year. So more recently, we looked at the changes in the topology of the evolution of the glycan shield, and in particular, I'm going to focus on the most newly acquired change, which of this unique to this virus, like to the SARS-CoV-2 S, which is the loss of glycosylation on N370, and how this loss of glycosylation actually allows this spike to bind cell surface glycans with high specificity. So, and within this framework, I'm going to give you some conclusion and perspectives. So as a quick recap, pretty much everybody has seen different representation of the spike. This is the most accurate representation at the atomistic level of detail that we have worked on last year and published last year, where in blue, are represented the glycans and the glycan shield that covers most of the surface. To the point that only a few spots on the spike are actually visible and then detectable by the immune system, and they are the basis of many vaccines already in use and approved. So what you see usually from cryo-EM, which is the essential information that we'll need to reconstruct these structures, it's a shaved version of it, which doesn't really exist. So we like to stress that the essential working of the spike hinges on the presence of the shield. So in these previous work, we actually calculated how much of the surface of the spike is accessible, the surface protein surface, and can be therefore recognized by the immune system, and that's very little, more than in viruses such as HIV-1, where the GP120 is virtually invisible through a very, very dense glycan shield. However, it's well covered, and especially when the spike is closed. When it's open, the spike actually has what is called a receptor binding domain, which is here represented in orange, which is accessible and recognizable. And one of the main, let's say, discoveries of our work was that the glycan shield is not only essential for the virus hiding from the immune system and therefore infecting and ravaging through your system, but it's also essential for activating the spike. So it's essential to its mechanistic process of infection by propping up the receptor binding domain, which ultimately binds the primary receptor, which is ACE2. These glycans are position 165 and through 234 and N343 are all essential. So as all of you know, I don't have to explain it for sure, like to this great audience, like the glycans, this type of glycans can be very diverse. And what we asked ourselves is that if all type of glycans, so if all S glycoforms would be equally functional. So by changing the glycans at those positions, you would have an attenuation of the infectivity of this virus. So we looked at that in the first paper by, let's say, mutating the position, in particular 234. So in single mutants and double mutants, and noticed through multi-microsecond molecular simulations that the absence of just one glycan causes instability or triggers an instability of this open conformation, which becomes very dynamic, like and would actually suggest a lower or weaker binding. So we teamed up with Jason McClellan's lab at UO Texas, who performed biolayer interferometry experiments on mutants, single mutants in particular, and discovered it and found out, like confirming like the MD predictions that mutating the N234, so removing that glycan actually decreases the binding to ACE2 by 60%, confirming that the glycan shield is indeed not only protecting the virus from the immune system, but is also mechanistically essential for the working of the fusion protein. So this, going back to our questions, are all glycoforms equally functional? It is not something that is like what is, can be perceived as an academic question. But it's actually a very important question in terms of all different forms of the fusion proteins that are being studied, or the fusion proteins that are in the vaccines and in the actual virus, not only in the recombinant spike that it's studied through cryo-EM. So, and this is confirmed in particular by two papers that came out in around April 2021, back to back, that from Max Crispin's lab, one, and from Weston Struve and Nicole Sitzman's lab, the second one, that look at the glycosylation of the Oxford AstraZeneca vaccine, and at the virus and ultimately the recombinant proteins, seeing significant changes in particular in combination to weakening of the spike in terms of structural integrity, which is understandable in the process through the process of glycosylation and glycan maturation. So, are all the glycoforms equally functional? Well, this question is incredibly difficult to answer experimentally, but difficult also to answer computationally. It took over a year of extremely long simulations, but we did it. So we mutated like the oligomanos at N234, like going from the very large man9, which is in the recombinant spikes, most stable spike to other forms like MAN5 that is present, most present in the virus and somewhat in the AstraZeneca epitope. So the smaller MAN5 is not as effective as the MAN9 in supporting the RBD. However, it is like the RBD is still up with a MAN5 in that position and is still competent, although is not as competent as a MAN9. So it's a kind of a weaker spike in that case, binding to ACE2. So like the weakening is even further confirmed if you mutate all of those three positions and the rest along with the rest of the shield to a shorter MAN5. And this is which causes the spike to be extremely unstable. And you can see here like extremely large opening degrees within conventional IMD, which is actually quite a feat to see. And this supports or it's in agreement with results presented in earlier work by Rob de Vries' lab, like where he found that fully glycosylated S proteins are superior in receptor binding compared to maturely glycosylated variants from GMT1 mutant cells. So as a process, so like a theoretical limit before the mutation or the removal of the complete removal of the glycosylation, we studied like we put in POSI mouse, like extremely small, which doesn't support at all like the spike, the RBD in the up conformation, and within the conventional molecular dynamics, also extremely long and in repeats, you can see almost a closing, spontaneous closing effect, supporting the role of the glycan, the essential role of the shield in stabilizing the spike in its active or dangerous conformation. So because the glycan shield is functional, it's only logic that the virus will evolve it to in combination to the evolution of the protein to maximum optimize activity and infectivity. And this is what is happening actually. Like if you see like the spots in magenta or all the spots that have been like where the glycan shield has been mutated along the phylogeny of the SARS-CoV-2, so the family of SARS-CoV-2. This is work in collaboration with Ben Schultz's lab at University of Queensland. So you can see here, like this is the SARS-CoV-2S compared to the SARS-CoV-S, SARS-CoV from 2003. We have like acquired a mutation that negates the seq1 at 370 that is just acquired in this virus. And this is the position of N370 relative to the one that I showed you before, 234, which is very important for propping up that RBD. So this mutation is also unique amongst all the known SARS coronavirus 2 or similar in circulation now. So we studied what is the effect of this glycan at N370, and that has been very highly conserved along the phylogeny, only now like lost. And we've seen that it's very effective in actually propping up the RBD. However, if you look at the RBD in the closed position, like the white one here, you can see the N370 in yellow, which actually straps down the RBD in a down conformation, binding along a cleft extremely stably. So what we see is that this cleft as what we call as a quintessential glycan binding site lined by aromatic groups that can bind or stack with the CH pi interactions along the complex and glycan at N370 here. One thing is just that is important to understand is that that N370 comes from another chain. So like come from the adjacent chain, strapping down the RBD and making its opening more difficult. So while the N370 is efficient at keeping it up, like when it's open, it actually makes the opening more difficult and energetically disadvantageous. Therefore, like this we think that the loss of the N370 actually has been an efficient modification for the spike to increase its infectivity, which is in agreement with recent work from Kang and co-workers in Cell, where the loss of the N370 glycosylation leads to an increased replication in human cells. Also, if we look at this N370 binding cleft, or the quintessential binding cleft that I highlighted before, it's vacant. In SARS-CoV-2S, now it's vacant because it's not like that glycan is not there anymore. Therefore, it can be filled with other glycans, such as GAGs. Like this is actually the binding site that is obtained, has been proposed by Jeff Esco through docking. You see, like it's very non-specific and is approximately around where our cleft is, although like docking has its own limitations in the technical limitations. So close enough. So the within the framework, like the work from John Klassen and Matthew McCauley in Alberta has shown that heparan sulfate is not the only glycan, so surface glycan bound by the spike, but also monosialylated gangliosides, as you can see here, GM1, GM2, and which bind with actually the same affinity. And you can see here that also with a lower affinity, the spike also binds type 1A, type 1H blood groups. So why is this? Like the spike binds all many different glycans, charged and uncharged, all in the RBD with very specific sequence. Some of them are not bound at all. What we see, what we find is like these glycans tend to bind the cleft that it was occupied by the N370 in different spots, depending on their sequence and structural and electrostatic complementarity. This is the binding site that we found for type 1 blood group A antigen complex. So you can see here it's very cozily hosted at the top of the cleft where the 1,3 arm was bound. And then this is GM1 and GM2. Actually, they are bound with the same affinity because the terminal galactose actually doesn't bind, which was a nice find. And where the sialic acid is actually bound in the same position, where a sialic acid of that N370 glycan would have ended up. Like, so that's the specificity for monosialic acid or ganglioside comes here. Disialylated species do not bind. So it's a complementarity of that cleft for different structures that allows them to bind. So, and these actually can function as co-receptors either directly by, let's say, sandwiching the binding to ACE2 like the type 1 blood groups can do, or by actually modulating what is called the receptor binding domain, the receptor binding motif conformation, which is intrinsically disordered. And for example, GM1 constrains that part when it binds like to a very stable conformation. So to conclude, a few insights from high performance computing through the past 20 months and some. So I hope I convinced you that the SARS-CoV-2 glycan shield is intrinsically involved in regulating the glycoprotein stability in its active form. So the type of glycosylation matters. It's just not any glycan shield. It has to be the glycan shield, optimal glycan shield. And its topology actually, like the distribution of this shield affects the stability of the open conformation, the opening energetic, and then ultimately ACE2 binding, so infectivity. So the evolution of the glycan shield has allowed SARS-CoV-2 to bind also host cell glycans, so surface glycans, such as GAG, ganglioside, blood group antigens, and provides actually a good strategy to design inhibitors which prevent this interaction and would limit the localization of the virus on the cell surface. So with that, I'd like to thank you for your attention. I'm sorry if I ran a bit longer than expected. All my students, Aoife is actually graduating today and I'm missing her conferring ceremony. So I'm I'm eager to see her, in her gown when she comes out. Carol Akash, and then my collaborators at UCSD, like Rami, Lorenzo Zayed Abi, and Jason McLellan, U Texas, Ben Schultz, Tuan Pung, and John Classing, Matthew McCauley, Lynn Nguyen, Laurie West, and Rob DeVries in Utrecht, and all the other collaborators on this and other projects. Thank you.
Thank you very much, Elisa. It was for this excellent talk. There are a few questions. The first one from Niklas Carlson. Would you think that tissue type differences of the glycosylation would relate to the susceptibility of different tissues for propagating of the infection?
That's an excellent question. Tissue, I guess, if I understand correctly, so the different expression of the virus by different cells might regulate its activity. That is, yeah, I would think so, because Nicholas is asking me if I think so. Yes, I would, but experimentally, this is proving to be very difficult. So what has been proven, and we're in agreement with what we all know, is that the stability of the spike regulates the type of glycans, with the least stable spike with more complex and glycans, so very multi-antennary acylated glycans for spikes that shed profusely. So, but yeah, there is a difference with the expression system, definitely.
Okay, we have to go for the second question, which is from Jake Young. After N370ST is mutated to N370SA, can this S371 be O-glycosylated?
Well,
I don't know if you got it.
Well, yeah, the O-glycosylation, this kind of relates to that would be a big close to the N-glycan, but recent work from Copenhagen, so Jeva Bagdonite, has shown that O-glycosylation is very frequent within the spike 3 position after the N-glycosylation. However, the occupancy is so low. It's the highest occupied is less than 2%. So O-glycosylation might be present, but is not functional.
Okay, so there are other questions, but maybe you can answer later on. Thank you very much again.
Thank you so much.
We have to move on, and we have the pleasure to have the next speaker. It's Dr. Kai Dahlmeier. He's a group leader from the molecular vaccinology and vaccine discovery from the Catholic University Leuven, and he'll be presenting animal models and vaccine candidates for COVID-19, right? Thank you.
Thank you very much, Chairman, and it's a great honor to be invited by the Society and to this symposium to represent actually here the Rega Institute. I would like to share and discuss with you what a large team of us has been doing from the beginning on during the lockdown. We have been kind of confined night and day to our labs, and we have been working on many topics related to Corona. And I will present today 2 main topics of mine, which was the development of animal models, which we then later use for the testing of drugs, but also for vaccine candidates. It's obviously, there are many vaccines. I will discuss this later. And we can still learn from vaccine candidates, which are likely the candidates to be second-generation vaccines, what is really needed for the ongoing pandemic. And I realized that I'm of the few people who makes this hybrid meeting a real hybrid meeting. So it's also funny and I have a real pointer, so you will see me struggling with that. So the SARS-CoV-2 virus is a part of a larger family of viruses which form a kind of continuum regarding the pathology. So there are these common cold viruses which cause upper respiratory tract infections. And then they have the very severe and acute infections with the SARS-CoV-1, so the original, the MERS viruses. In fact, the SARS-CoV-2 is a bit like the ugly sister in this family, who can first cause the upper respiratory tract infection, which actually lets us then to walk around, or in this case, we now wear mouth masks to protect that, to transmit to others. And only later then we come down with a severe disease. So it's actually perfectly adapted to human behavior. So we go to the club and then the next day we fall sick. So this is part of the misery, and this has several implications. Obviously, the virus emerged as a zoonotic disease from an animal source, likely bats, and it can infect a plethora of different animals, carnivores, next to humans. It can also, and this was discovered by us and others very early on, in Syrian golden hamsters. What is particular in this hamster model is if we take a clinical isolate of SARS-CoV-2, so in this case we use the index case from Belgium, so the first isolated here at the virus isolate here at the institute, and put it on the nostrils of a hamster. In this case, there's a rapid replication of the virus launched. And in fact, the disease we see and the infection resembles more the second phase of COVID. So basically the lower respiratory tract infection, we see a massive growth of the virus. It's a true explosion. So within 3-4 days, we have a 4, 5, 6, 10 log increase in viral loads in the lung. And this results in a pathology, and I show here just one picture out of that, which very much resembles what is seen in patients with bronchopneumonia. So there's a consolidation, infiltration, many hallmarks of what is seen in patients. So we believe with this model we have a small animal model which is tractable in the lab, but which also resembles a pathology in contrast to many other models like transgenic mice, which in the most simple version rather cause neurological disease. We had access to some unique animal models which are related to this hamster theme, namely knockouts in receptors for key signaling molecules in the innate immune pathway. These are STAT2 knockouts and interferon lambda receptor knockouts. And not as fully unexpected, in the case we have a STAT2 knockout, so you may consider that as one of the most severe compromises in innate immunity. You see that the virus in the lung really explodes. So we have 100 to 1,000 times higher viral loads in the lung. And under these conditions, the virus is even not anymore confined to the lung, but it gets systemic. So we can detect it in the bloodstream, which is not seen in the wild type animals, but also in the intestine, in the spleen, in the liver. So it gets really systemic. However, and much to our surprise, these animals appear healthy. So, and this can be seen by different, if you score for the histology, or if you look with tomography at the lungs like the radiologist does in the clinic, the wild type animals have consolidations in the liver, which sometimes even resemble these ground glass opacities or other typical lesions seen in the infected lung. However, the STAT2 hamsters look much more healthy, even cytokine or other markers expressed during inflammation, like MX2 or interferon lambda, they are almost normal. So as if these hamsters are not infected by this virus. So this, in a very simple interpretation, means that obviously the SARS-CoV-2 replication is definitely limited or restricted by this strong innate immune response in the lung. But on the other hand, exactly the same innate immunity in this later phase of disease drives the severe pathology. And this has likely some very important implications for clinical use of intervention, for medical interventions, clinical use of antiviral drugs. So the question is, can we treat corona at this stage of disease? Or was there a point of no return where the virus pushed the trigger and then it's too late? So then the immune system, immune pathology took over. Likewise, we have to intervene earlier, so basically by prophylaxis, and there we are, of course, with the theme of vaccines. And we are really lucky, and therefore actually we can do at all a hybrid meeting even today. We have several vaccines in the run, which have been developed really at enormous speed. And some of them are extremely efficient even. So we have vaccines which reach up to 95% vaccine efficacy in clinical trials, but also in the real world. So they have a massive impact on public health systems, including admission to ICUs. They are based on several platforms. So we have classical inactivated viruses. We have vectored viruses, actually first in class in human. We have first in class in human RNA vaccines and subunit vaccines, which follow a bit more the old paradigm of classical vaccines like the flu vaccine. However, there are some boxes we cannot tick off yet. Actually, there are some remaining problems, and this is obviously, and this has become now overt, is fairly short duration of immunity conferred by these vaccines. So we are now considering boostering or other means to continue the fight, even in the vaccinated. There is an emergence of new variants, and in fact, all vaccines in current use have been developed against a virus, which is something for the museum. So this virus does not exist anywhere on this planet anymore. So we are treating with a reagent which is not fit or not matched with the current pathogen. And there is true evidence for escape from vaccination. I will show also some data on that. These vaccines do not protect against transmission, so it's not a population-wide effort, and some of them have prohibitively strong, difficult logistics, and therefore large parts of the world, besides just of the cost of the vaccines, have no access. So it's more than almost 2/3 of the world population did not get a single vaccine yet, despite 6.5 or almost 7 billion doses have been deployed. And this is a very impressive number, because if you consider that the annual vaccine production on this planet prior to COVID was about 5 billion doses for any indication. So we have just doubled this in this time. We use another approach for our vaccine here at the Institute. We use a platform which is based on the legacy vaccine for yellow fever. And this is the 17D, live attenuated yellow fever vaccine, which has a fantastic track record. It's after the Jenner vaccine. It's the first human vaccine, which in this case, it was made by semi-rational design. And it has been dosed in billions of people in the meantime, and has some very specific characteristics. Namely, it induces a very broad and long-lasting immunity, and maybe even after single shot. What we have learned in the last 10 years is that if we use this as a vector, then many of the characteristics of yellow fever also are translate to the antigen we use in this vector platform. For the corona, we obviously chose as a target the spike protein, and this has been introduced in much detail. And of course, today we have an impressive knowledge on this antigen into greatest detail, even the glycosylation, as you heard in the talk before. But back then, there was almost no clue. There was a nucleotide sequence published by the WHO. So what we did, we took best guess antigens or antigenic sequence out of that, and made a panel of vaccine candidates in our yellow fever vector platform. I show here mainly 3, and I will focus in my talk on one which we call the S0 or YFS0, which is expressing the pre-fusion stabilized non-cleavable spike protein. So with this respect, it's very similar to what is used in the Johnson vaccine or in the RNA vaccines. So it's not the cleavable, but it's a kind of a pre-fusion form of the spike. And I will show you some differences, because then we tested in our hamster model, and I will show these data, how we selected then for vaccine efficacy and down selected to a clinical or preclinical candidate, which is this S1. And this is my contribution to glycobiology. You see that when the cell is transduced by the virus, expresses a spike protein, here just in very dim green in an overlay with the red yellow fever proteins. But these proteins expressed, so the S proteins, the spike proteins, are highly glycosylated, actually many forms. If we use this material to vaccinate hamsters, we see a rapid seroconversion to antibodies, in particular with this construct S0 to high levels of binding neutralizing antibodies. And this can be within 7 to 14 days. Yeah, we did then all kinds of, hamsters are cute, but they are little reagents. So therefore we needed to use, also in collaboration here in the institute, use mice to profile more the spectrum of CMI and the polarization of immune responses. To cut it very short, our lead candidate has a particular favorable Th1 polarization of its immune response with expression of a lot of cytotoxic virus-specific T cells. Okay, then in the challenge experiment, it shows that the S0 vaccinated animals have, if we challenge them with the virus, as I showed before, have a massive reduction in the viral RNA loads at peak, so about 6 log reduction. And most importantly, there may be no virus detectable anymore in these lungs. And if you then look in the histology and also in the radiology in these lungs, these animals, the lungs, by contrast, of course, as I showed, the sham vaccinated animals have lesions and consolidations, infiltration, pneumonia. These appear, these S0 vaccinated animals appear healthy. And even the cytokine profile is normalized as if these animals had not been exposed to the virus. We pushed it a bit further and asked ourselves, can we achieve the same also with a single dose vaccination regimen by just adding, leveling up the vaccine dose by tenfold? We can get within 21 days after single dose vaccination, all animals protected in a larger cohort of animals. And even if we shorten the time after vaccination before we challenge them, we can even achieve still a decent amount of animals which are fully protected from virus challenge, which shows that this is a very fast mechanism of action of this vaccine. And of course, this may lend, especially in outbreak situations, as we still have worldwide, and this may lend its use in such a situation, or also in medical systems where we don't have the possibility to have very complex vaccination schedules like come back in 10 weeks, which is almost impossible in almost all parts of the world. Okay. You may say mission accomplished, but as I mentioned, and this is a common problem for all vaccines, we have these new variants popping up everywhere, and it is that dramatic that even in countries like, I showed just a few selections, countries in the UK, we have had this first variant, so-called alpha variant. These are each time epidemics in an epidemic. And if you look now, just the UK case, the alpha variant, which was that dominant that it swapped the world, within 2-3 weeks, it was completely substituted by the Delta variant. We here in Belgium have a particular situation. We have an excellent surveillance, included by colleagues here from the Rega Institute, but sometimes you may discuss if we have a good management of the disease. So we had very deep insight in the evolution of the variants here on site. So we have seen all of them. And but also here, Delta is dominant. There is one scary guy in the club, which is the mu, which is a combination even from the antigenicity between the aggressive Delta and the immune escape Beta and Gamma variants. So, and you see, for instance, in Colombia, it's a variant which emerges in the background of the otherwise dominating Delta. So what we could do in our animals, also with the clinical isolates we had from all the variants, we could check, okay, nice, if you vaccinate an animal, we can still protect against the UK, so the alpha variant. However, or against the beta variant, there is loss of protection. So the vaccine efficacy drops down to 40%. We can protect any more part of the animals with our otherwise fairly potent vaccine. So, of course, this may mean, and as I told you, we use the same antigen as all current vaccines use. So this may be a more common issue. And in fact, we cannot define anymore a correlate of protection, so a protective antibody level anymore for the beta variant. And you see there's an increase from 20 to 100 from wild type, or from prototype to alpha variant, and this further increase in the antibody levels we need to achieve 50% protection. Okay. of course, being optimistic, we tried to come up with a universal antigen combining the most relevant mutations seen in the 4 prevalent variants of concern, and this is a universal spike antigen put in our yellow fever vector, and now again achieve full protection against the full spectrum. So, just to summarize, I think we have found a model which relevantly mimics the disease and the infection of corona, and can be used for all kinds of preclinical research. In this model, we to some extent may study even the pathogenesis of disease by investigating innate involvement in this disease. We developed yellow fever-based vaccine COVID candidates, and I did not have the time to show all that, so we tested it in several step-up models, including in monkeys, and it holds its promise there as well. There's a particularly high immunogenicity, but also rapid induction of protective immunity in the hamster and non-human primate model, even likely from a single dose. However, this I showed you is compromised by the loss of protection, partial loss of protection against variants, but this can be solved with adapting the old antigen for the new one, and this should be taken serious by many people, including by those who are now developing or deploying vaccines for the next season, actually for the current season, to adapt it. What I did not show you is that our vaccine has another asset. It also fully protects against yellow fever. So this is of course, it's not relevant here in Europe, but in other regions of the world, this is a vaccine which is available, but not readily available for everybody who actually would need it. And as an outlook, based on our platform, we have been working on other indications, and this actually helped us to understand how to use the platform and how to come up so fast with a candidate for corona. So we have been working on rabies, Ebola, Lassa, and others. And each time it induces then a strong yellow fever protective immunity, but also a CMI and humoral immunity against the targeted antigen. Okay, as I mentioned in the beginning, this was not the work of only few people, but of a gigantic group of very dedicated colleagues from the Rega Institute Virology, from the clinical and epidemiological virology, from immunity and inflammation research. We had collaborators at the Primate Center in Rijswijk and expert pathologists. Yeah, I cannot mention all of them, but this is a gratitude to all of them. I leave it here.
Thank you very much for this excellent presentation. So, while we wait for the questions from the audience, I just wanted to a couple of points. One is if your strategy with your antigen includes some protection against the mu variant, if you know that.
We don't know yet, but variant contains a certain mutation in the receptor binding domain of the spike, which is known to lower the affinity for neutralizing antibodies. Likewise, the Delta variant contains a mutation, or key mutation in this variant is the one which enables maturation cleavage. So, and the mu variant contains both of them. And so it's likely as fast spreading as Delta and as much escaping immunity as Gamma and Delta, Beta and Gamma variant. So best guess is with our antigen, where we cover both Beta and Gamma, and still a bit with the nomenclature, this should also cover the mu.
Yes. Yeah, and a curiosity, do you know how updated are being the people that are deploying the vaccine, updating the vaccine?
I know that they are still at the level of press releases. They are all heavily working on that, which is good. And some of the technologies actually allow it easily. So, for instance, mRNA vaccines, definitely some vector vaccine are maybe more difficult for the process, downstream process, but I'm not sure what is the choice of antigen, because there's always a lead time, and yeah, maybe that this may be interesting. Such a universal antigen is really in sight also for the future, of course, like the universal flu is the kind of the holy grail. Yeah.
So, so I would like to thank you again for this excellent presentation, and thank you. We close the session, and we thank all the speakers for this very exciting session this morning, and we come back for the session 2 in 15 minutes. 15 minutes. Thank you.
Well, I might have to let you ask the questions if I can't, if you can't.
Yeah, I think he's going to widen.
We can turn on the voices if we like. Is there any chance you can get Greg big time somewhere and me little time? Like over there or there?
I think you.
Fantastic.
Oh, John, I think you must have something unmuted.
I haven't got anything on at all.
Oh.
Yes, there was an echo.
So if we can have Greg in big.
Just to show when I look shifty, is it?
You repainted the room.
No.
We're trying to get your face up here. Dave, can we get Greg's face up?
Yeah, it's the left.
Dave, I think the echo is actually coming from that mic up there, is it? Above John?
Ah, no, it's because someone joined from here. Who
Okay.
has also the mobile mic.
So how are we doing this? That's going to go.
Yeah, otherwise you have to just look on the 1 minute to go.
So can we have a big Greg?
That's my duty.
Oh, yeah.
Very important.
This is now what people are seeing. Yeah, it's just two same size.
So I'd like to have a big Greg there, or big Greg there, and a little me here.
Yeah, but then it will change for the people at home also.
So that's how it is?
Yeah.
Okay. Greg, are you hearing us well?
Yes, I'm hearing you very well. Are you hearing me well?
Fantastic. No, it's perfect. Perfect. Good. I'm glad you managed to get your meeting done, and we will start. So, welcome everybody to session 2. My name's John Axford, and it's a great pleasure to introduce you all to Sir Greg Winter, who I know has been waiting 31 years to come to a Jenner Glycobiology and Immunology meeting, and we've captured him between 2 other meetings he had today. So it's great to have you here, Greg, and welcome. Now, Greg always professes to know very little about immunology if you listen to some of his talks, but he knows an awful lot about antibody molecules, and this is why he was awarded the 2018 Nobel Chemistry Prize for phage display of peptides and antibodies. And it's my view that Greg's invention, as I mentioned about Jenner in the first intro talk here, is one of the greatest of this century, because this has been a major benefit to humanity. I don't want to embarrass you, Greg, but I have direct experience of this in a number of ways when all my patients were queuing up in 1990 for the various drugs that you invented. And Humira adalimumab, for one, has been a major power in our armory, certainly in rheumatology, and has changed my clinical practice. But we'll get to that later on. But it's always interesting to go back and think about where people started. And Greg, you were born in Leicester, but then I was interested in your CV. You grew up in Ghana, where your father was a French professor. So you went to Ghana. When did you emigrate?
When I was about 2 months old. So
Right.
I was a very premature baby. So we emigrated roughly about the time I was due to be born, I gather, perhaps a month or 2 after that. The thought was that I would die if left in rationing England, so that there was no extra coal permitted, despite the fact I was premature. Everything was very bureaucratic in those days, and my mother persuaded my father, who at that stage was not able to get a good job in French in England, to emigrate, well, not totally to emigrate, but to go to West Africa, because my father had been told that the colonies was a first-rate place for third-rate people. So he said, oh, I better go in that case. So off he went.
And as it happened, you went to what became a major centre for the Medical Research Council. So there you are in Ghana. So you emigrated to Ghana for health reasons.
Well, I think yes. And actually, so I was pretty sickly before I went, and I look at pictures of myself and I think, I don't think he's going to survive. But actually, very shortly after being in Ghana, it really rather saved my life, because I then turned out to be rushing about and doing all kinds of stuff. So I think the heat was what I needed at that point, and I got it. Of course, I also got some other diseases later on, which nearly knocked me out again, but nevertheless, I came through it.
And at what stage in your life did you return to England? Were you educated in Ghana to a certain extent?
Yes, I think my primary school was in Ghana, in a sort of almost a mission school. We used to sing hymns in the morning. I can remember, so I used to know all things bright and beautiful. It was all very sweet. And when we came back to England, my biggest shock was being expected to know the English country classics, of which, I kid you not, one of them was "The Fly is on the Termite." Now, in fact, I doubt you heard of it, but we're all thought, right now, school, a rendition of "Flies in the Termite," and the master went around looking at all our lips. I hadn't the faintest idea what he was talking about. Anyway, I had to write it out 1,000 times, even though I explained I'd only just arrived and I didn't know this. He said, "No, it was done last year, you should know about it. Anyway, this will teach you to." So that's what I experienced when I came back to England.
And I guess you had a super accent, as some of my friends whose families were taken off to Kenya developed over time, which was quite an amusement for us. So you returned to England when you were around about 10 or so?
Well, I suppose it was when I was about 8.5 to boarding school, but that didn't go very well. And after about 2 or 3 years, I left and came back to Ghana for a year. So in fact, in Ghana, after that, my father came back. His job was Africanized, and he returned to the UK.
And he continued as a French teacher?
No, he didn't. In the meantime, in Ghana, he'd moved to an organization called WACO, which was West African Examinations Council, running exams to get Africans qualified to come to England. And he decided, when they set up the first CSE boards in the UK, it turned out that a lot of the challenges with testing, most of the population who weren't necessarily academically gifted, had been faced by WACO and its corresponding organizations throughout the empire. And so so in fact, it was the empire came back. The empire came back, and a lot of the positions in the London board and the other boards, Midland boards, were occupied by these colonials. And they all used to get together and have gin and tonics and remember the old times.
Those were the days. So I guess you went to the Royal Grammar School in Newcastle when you were 11.
Yes, actually about 13. I went when I was about 13, returned from Ghana. Yeah.
Were you always a chemist, or did you have other ideas?
No, we didn't really do much chemistry very early on, but I would say I was more a biologist, or more interested in biology, but we weren't allowed to do that until the year before GCSE, or GCE as it used to be called then. Yeah. So in fact, I did chemistry, I did physics, and I enjoyed them. Even though now I look back and I think they were pretty frightening. Boys with test tubes with things leaping out the end of them.
And no glasses.
No glasses, burnt fingers, but we learned it all. Things going bang in the fume cupboard.
And was there a specific mentor that you had in Newcastle? Because how many Cambridge graduates did Newcastle produce?
I don't know. I think it produced a fair number. I think it still does. The Royal Grammar at Newcastle. It was a pretty good school then. Right. And certainly there was a cohort of us went. I don't know whether anyone came back again. I know the headmaster was from Oxford. So there was a scattering of people who, by virtue of being Oxford or Cambridge, seemed to have a slightly higher profile in the staff room. And the headmaster just told me, you know, the end of my time at Newcastle, right, it's Cambridge for you, and I've decided you should go to Trinity College. So I said, yes, sir, and, you know, went.
If only life was so easy. Although things have revolved, haven't they? In that you're probably better off at the Royal Grammar School than you are at Eton these days.
Possibly.
And at Cambridge, you did biology. What was the how did the degree evolve?
It was a natural sciences degree. So actually, I was interested in biology. Actually, at that stage, I was thinking of being a marine biologist. So I did the various biological options, but it was biology of cells, chemistry, physiology, and a bit of mathematics. And I think in the physiology, I came to dislike it thoroughly, largely because of all the decerebrate rabbits, and my view, unnecessary brutality inflicted on the animals, which, you know, I sort of understood a bit, but I couldn't see why I had to do these things to these animals. And I didn't want to do any more of it. So in fact, I then focused in my second year on chemistry and biochemistry, and settled in the end on biochemistry. Although in fact, I enjoyed chemistry more, but I felt that biology had the more important questions. Chemistry, I felt, had gone up a bit of a dead end at that point. It all seemed to be quantum mechanics, which I didn't really want to get engaged with. And biology, you know, this was the stage just after, oh, excuse me, I'm going to put them off. It was a stage when, bloody thing, I don't know how to stop it. Sorry. So it was a stage, I suppose, when people, were a lot of invention, a lot of discoveries were being made. It wasn't so long after. Well, I suppose, of course, you'd had Crick and Watson, but there was, about the time I was in the sixth form, there was James Watson's book, The Double Helix, published. And so there was quite a buzz about the developing field of molecular biology. And I was really very excited by it, very excited. So,
and I guess
sorry?
You were rubbing shoulders with some pretty eminent people then, weren't you?
Not straight away in Cambridge, no. I mean, as a student, you didn't rub shoulders with the greats of the field, but later, when I came to do my PhD, so my supervisor was a protein chemist called Brian Hartley, a very blunt northerner, who'd actually been involved in working on the active site of chymotrypsin, so identified the sort of charge relay system there. He was my director of studies at Trinity College in Cambridge. And I got on very well with him, and I was interested in working with him because I came to like, I was interested in protein structure. I thought they were intrinsically very beautiful. And I thought, what a life to spend your time immersed among these structures and trying to work out how they work. I mean, enzymes are really very exciting.
So roughly you saw where you were going there. So did you get a first or did you get a third grade?
No, I got a 2-2, and
right.
but I got a first in my first 2 years, but I got a 2-2 in my last year, which I think was a very big mistake of the examiners. And I've been and actually, when I started my PhD, my primary, I think it was very good for me because my primary determination was to prove the bastards wrong. So I worked incredibly hard, and with increasing satisfaction as I ascended the rungs, thinking, right, that's another one up them kind of thing. So it actually took quite a few years to stop feeling that.
And did you go straight into the LMB, the Laboratory of Molecular Biology, or did you work elsewhere?
No, I went straight into the LMB working with Brian Hartley, but shortly after that, he became made a professor at Imperial College in London. So I sort of, I managed to stay a year there by myself, tolerated by Fred Sanger. But then in my third year, I had to go down to Imperial College and set up a protein chemistry lab. So in fact, I was, which of course was what Brian wanted. He wanted me to go and do the work of setting up the protein chemistry lab, which now I was familiar with how protein chemistry worked. And on the basis of that work, I got offered a fellowship at Trinity College, a sort of junior research fellowship, which I then used to return to Cambridge, which meant it was great actually, because the fellowship was independent of of where you worked. So you could just go touting it around and say to different departments in Cambridge and say, look, I've got some money. I'd love to work in your area. Will you take me? And so it gives you a negotiating position. And in fact, I asked to go and work with Fred Sanger, but he said he was full, which was true. He kept a very small group, but he passed me on to one of his lieutenants, a chap called George Brownlee.
Right.
He was sort of later went to Oxford, and George was working on RNA sequencing. So my PhD, I learned protein sequencing, protein structure. Then on my postdoc, I was learning RNA and DNA sequencing.
So this is all early days, wasn't it? Because this is all 1980.
It was actually, it was 70s. This was 70s. So in fact, cloning had been invented, roughly 1973. The dideoxy method was 75, well, actually 76. And I was working with it in 77. And all I can say is in those early days, the method was extremely fickle. And, you know, we had to clone bits of DNA first to be able to use the method. But it was extremely fickle. And, you know, Fred's lab could go down a week at a time with no one able to get the method to work. And it was just to do, I think, the quality of enzymes or the quality of buffers or the radioactivity. Something had gone off because you were incorporating radioactive nucleotides. We'd spend all our time running from one potential hypothesis to why. So someone would get something to work, and they would then be subjected to interrogation in detail about exactly what water they'd used and what, you know, what the enzyme, what batch enzyme number it was, and where the triphosphates came from. We'd all rush and do that, and sometimes it would make a difference, sometimes it wouldn't. It was very, but it was really frustrating, but at the same time, quite educational in making you know, if biology, if everything worked first time, you know, probably I could have got my entire career over in 5 years, but actually things don't work first time. It's really hard. And some
At the time of your experiments, these experiments must have taken a long time. Yeah,
well, we were sequencing influenza. We were trying to sequence it, and we did the first complete sequence of influenza. That's Stan Fields, George Brownlee, and myself, and that was completed in about 82. But we largely finished in 81. But that involved, you know, cloning segments, and it involved stitching together about 14,000 nucleotides, and all of this was done manually, you know, with P32 labeled material and radio autographs. And, you know, actually it was pretty accurate. I don't think we found any errors in it because you used to overdetermine it. In fact, the methods, I would say, you know, we're very close to. We used to try and get the reverse strand as well. And with the two, we didn't have to get the level of degeneracy you seem to have now with these machine methods. We could do it with a couple of well-chosen gels. You'd know where you are.
Now, I may have got this slightly wrong because you are also a tRNAs synthesized expert, but this is the target for an autoantibody in a lot of the diseases I look at, rheumatoid, Sjogren's syndrome, etc. And I guess you weren't aware that this was going to be a target antigen for a disease.
No, that was indeed that was the subject I did my PhD in. Was the sequence, was the protein chemistry involved determining the sequence in protein sequence space. They're not using nucleic acids because we didn't have the gene. Of determining the sequence of that enzyme and trying to understand how it evolved. So no, I had no idea. I would have been very surprised if you told me that.
And just looking through various articles about you, as a Nobel Prize winner, you have no CV. So what was your first paper?
So what do you mean as a Nobel Prize winner? I don't have a CV.
Well, you haven't got a CV like mine.
You never asked for it. You never asked for it. So
what was your first paper?
My first paper was a paper on the sequence of tryptophan tRNA synthetase.
Oh, there we are.
That was it. So that was the first one of its class to be sequenced. As far as I know, it was right. I mean, subsequently people sequenced the gene, and you know, I think it was pretty much spot on, which is a source of some satisfaction. But it was on the basis of that I got my fellowship at Trinity and used that then to, you know, move up into learn DNA sequencing. And the reason I was keen on DNA sequencing is I knew what a slog protein sequencing was. And I'd seen a talk by Fred Sanger, and I thought, my God, this is the future. I'm going to be in a redundant field if I don't move. And so that's why I wanted to learn DNA sequencing.
So the NMB has many successes and many Nobel Prize winners. What's its secret? Its secret is succession planning?
No, it's very difficult. People talk about the culture of the LMB, and that probably is it. It's a form of culture, but culture depends on tradition, I would say, and the mentors you had. And so all I can talk about is the secret of the LMB as it was when I was there. I don't know whether it's exactly the same now or not, but certainly it was a very difficult, tough place for a student. There were only about 3 students in total. We had 1 supervisor. We had no psychological props. You just had to deal with that. You were treated a bit like a postdoc. So in fact, a bit like a rather ignorant postdoc, because of course you didn't know how to do most things and you had to learn from people. There were no protocols. You had to be able to persuade people to give them to you. It wasn't as if there was a book of protocols you could follow. So you learned to be charming, helpful, which is probably quite good. I can be that from time to time. And you also learned to work very hard. And in particular, that's what I mainly remember about it. It was 12 hours a day, 7 days a week, most of the year.
I think you answered my next question, and that what did you do in your spare time? So
there was very little spare time at that stage. I can remember doing an experiment at the same time as Fred Sanger. It involved having to get access to a containment laboratory, and we were going in and out alternately. So I knew where he was in the process of preparing M13 clones. And I remember sort of noticing on several days, he was working till about 1 in the morning. And then when I got in at about 10 in the morning myself, I noticed he was, he was clearly in the lab at that time. And I thought, I wonder when he gets in. So one day I having seen him leave the building at 1 o'clock, I cycled all the way back home, 1 hour by bike, and then returned the following morning at 6 o'clock in the morning. I thought I would see when he turns up, you see, to finish off a particular part of the experiment. And my God, as I walked into the lab at 6, he came out, he winked at me, said, "Morning, Greg, nice to see you in Brighton early this time." it was just incredible. I thought, that man, Fred was obviously very bright and very creative, but God, he worked hard. And I remember another occasion, during a weekend, for example, he would work on Saturdays, sometimes Sundays. But I can remember one Saturday seeing his wife wandering the corridors of the lab. Of course, those days were days when people could get in, everything wasn't totally locked up and secure. And a colleague of mine said, "I've just been to the loo and Fred seems to be hiding in there." Anyway, he said, he'd asked him why he was hiding in the loo, because it's a bit suspicious. It's just a loo with 2 urinals and your boss is hanging around, not doing anything. He said to Fred, what are you doing? He said, "Well, I think Joan's after me. So this is the one place she won't come in." He said, so I'm going to, so I thought I'd stay here. And could you let me know when she goes? So basically, when we saw Joan off the premises, we let Fred know, and he went back to his experiments. But I suspect he was supposed to be somewhere else or doing something for his wife. But anyway, he worked.
So there was humour in the lab, even if you weren't allowed out for a drink.
That's correct.
So your wife then...
Hang on. We were allowed to drink at Christmas. On Christmas Eve, he'd come round with some of his elderflower wine or elderberry wine. And if any of us were still there on Christmas Eve, we could get a drink from him, elderberry wine, 4 o'clock in the afternoon, Christmas Eve.
So that was the lab party, dear, right? That was the lab party. So then things took a bit of a change, didn't they? Your career did grind to a halt, and you pirouetted. Not many people in the audience will know this story, Greg. What actually happened to you then?
So you're thinking about the injury, are you?
Yeah.
Yes. So I was working. I'd started work from DNA sequencing. I moved to. I thought actually this is actually getting rather boring. and I thought, I'd originally thought it could be used to explain how proteins work. But then I began to realize actually it didn't really, because we'd got the entire structure of flu and there wasn't much we could deduce about it without having lots more information, like from mutants or from structure. So I thought, do I want to work on structure? No, I thought I'll work on mutants and it would be great to have defined mutants. And instead of working on a virus, decided to go back to some of those tryptophanyl tRNA synthetases and things I've been working on to try to elucidate the catalytic mechanism. At about the same, so as I made a decision to do that, it was also the lab, Fred was stepping down, and he told me I'd got to go and talk to Cesar, who was Cesar Milstein, who was going to become the next head of the laboratory. And Cesar made it clear he wasn't that interested in enzymes. He loved what I was doing, proposing to do, was to make site-specific mutations and to use lots of heavy recombinant DNA methods on proteins. But why couldn't I work with a really interesting molecule like antibodies, rather than these enzymes, which he agreed were fundamentally interesting, but actually he felt antibodies was so much more exciting, and I'd also fit into the divisional strategy. So it got he needed to maintain a coherence in the division. So I took the hint and decided I'd better work on antibodies. And shortly after that, I was attacked in the street on my way to work, and by I think a madman, and he ripped out my right arm, and I had a brachial plexus lesion, and which was not only extremely painful, but
did he not throw a hook at you? It wasn't sort of.
No, he actually tried to run me down. I managed to avoid that, and then he came out of his van, and I thought he'd accidentally nearly missed me. And as he came up to me, he whacked me on the head with an iron rod. And then as I fell, obviously semi-conscious, he ripped my arm out. And then when I'd fallen in the gutter, he started moving the lorry towards me. I managed to roll out of the way. But it was very difficult because the arm was stopping me rolling. It was really hard.
And did he think you were someone else? Did he think you were someone else?
No, he said to the police, I saw this sort of vaulting little academic on his way to work to cut up bodies, so I thought I'd run the little bastard over. That's what, I mean, what can you do? I mean,
well, so, hopefully he's still inside.
Well, no, he's not, unfortunately. No, he came out after about a year. That's such as English justice. So
there you were with no arm.
So I had a paralyzed arm and no knowledge really about what would happen. I asked one of the medics what would happen, and this chap, who I later turned out had joined the army after he'd seen me, which I think is a fit place for him, said, "Well, I think you just want us to cut it off for you," he said. "No point in having that thing hanging around." So
man up.
So absolute medical sympathy. So I just had to wait for the nerves to re-innovate, which fortunately they did, but the pain lasted a long, long time. And the only way I could relieve it was to go into the lab and look at structures. In fact, I looked at antibody structures in great detail. And actually, it was quite interesting, just the power of your mind over pain, that actually I would go there at 6 o'clock in the morning and try to be on that computer to about 12 o'clock at midnight, and then go home. Couldn't sleep very well because of the pain, but I was so tired. But during that time,
cognitive behavioural therapy with antibodies.
Yeah, it is. You just get totally involved in looking at antibody structure and how they come, learning how to use the program, showing how to display them. And during the course of that, I started to speculate about the possibility of, well, I won't say exactly why I did that, but this possibility of transplanting loops in those structures from one antibody to another, and then came up with the idea of humanizing antibodies by transplanting loops. So that was my first foray into therapeutic antibodies. And Cesar was very encouraging of that, as was Michael Neuberger, who was also working with antibodies at the time.
So I guess you then moved on with a lot of help, with not a right-hand man, a left-hand man.
Well, I was given some help, actually. I was very grateful at that stage. So I couldn't do the experiments because the experiments involved gene synthesis, and we'd not done gene synthesis. So we had to make all our oligonucleotides by hand to make these synthetic antibody genes. And yeah, this was at the front of technology then. Sidney Brenner gave me a summer student, and Cesar Milstein agreed I could have one technician. So I had one technician and one summer student. Then later I had a postdoc to help.
So yeah, so not many people.
No, it was very. I mean, the lab had the view that you were expected to deliver yourself, and so for many years, I did most things myself with very little help. Actually, after I'd started, all the flu work was done really with myself and Stanfield's operating independently, but we had to do all our own experiments. There wasn't a sort of technician you could offload it onto and pontificate. It was up to you to get things to work. But as a result of that, I would say I've got I learned about every artifact in recombinant DNA that could occur, and therefore was able to, when I got a group of people who were then working with me, as I expanded the group, I was allowed to expand the group in the light of success. I was, I could diagnose where things were going wrong. I could tell them what to do very quickly. So actually, I don't know whether I would have been better to have continued with my own experiments, rather like Fred Sanger had done for the whole of his life, or whether in fact I am a better manager of people. I don't know, because I never really got that chance to show them. I wish I'd been able to continue doing experiments, because I think I'd have ended up being a lot fitter and probably have more varicose veins. But it was more enjoyable. I used to love working with my hands, and I can't really do it now. It's the hand is it's okay, but it's not. I can't expel a Gilson, the tips from yellow tips from a Gilson pipette. It's too with my thumb. It's just not possible to do that.
So opening champagne bottles is a bit arduous.
Oh, my arm's all right, actually. My arm's all right for drinking and for opening champagne bottles, but it's a very blunt grip.
So, Greg, you picked out that I didn't quite get the sequence of things that sort of happened then. But take us through your ideas of how you could humanize antibodies. The first monoclonal you invented,
yeah,
and then where things progressed to the phage display and fully humanized antibodies.
Yeah. Well, at that point, we were interested. Monoclonal antibodies was at a stage when in fact it wasn't flavor of the month or the year, because it was clear that the mouse monoclonals could be made, but they were immunogenic, even when you've purified them very extensively. But various groups started to say, well, maybe we could make these more human, and started to make chimeric antibodies. And Michael Neuberger was one of those, putting forward mouse entire variable domains onto human constant regions. But my insights in that computer graphics have made me believe the structure of those domains was sufficiently conserved. You could just transplant the tips, the antigen binding tips, and they should be more or less supported in the same way, which means you could make a humanized antibody that would have 6 loops transplanted, tips of the loops transplanted, and that would probably bind the antigen. So that was the first experiment we did in model systems of no interest to anybody. And then finally working with Herman Waldman, who was then in Cambridge, to make the antibody Campath-1. And I was involved in, we were involved in making that antibody, and Herman put it into a patient.
And that's an anti-B cell antibody, or straight?
It's an anti-CD52W antibody. So it's against a small antigen, the surface of B cells and T cells. So it's pan B and pan T. And he originally wanted, I think, to use this as just an immunosuppression, but it turned out it was also useful for debulking lymphomas. And so the patient we saw was the first patient it went into was a patient who'd got non-Hodgkin's lymphoma and a vastly expanded spleen of several kilograms, and was neutropenic and on death's door. I mean, she was really in a very bad way. And they gave her the antibody, and it just basically dissolved away, very gradually dissolved away this huge mass of tumor. And it had a very big impact on me, actually, because at that stage, I suppose I'd been thinking of moving on. I hadn't been quite sure what I was going to do after that first demonstration, but meeting the patient made me realize that that she was a lovely lady. They told me I should go and see her.
So they got you on the board, did they? They made you.
Yeah, they got on the board. They said, look, I tell you what, put a clean lab coat on that doesn't say Medical Research Council, so it doesn't look like you're experimenting on people. Take this stethoscope, you know, and make sure you've had a decent haircut, but try to look like a junior doctor. Go and talk to her. Just go on the ward, because I didn't think they'd let me in. And I got talking to her, and she was sort of asking, you know, she asked me, you know, what I thought of the treatment. I said, well, I said, you know, we haven't the faintest idea. She said, well, what are your hopes? I said, I hope it doesn't kill you. Because actually we've given us this antibody the first time with human effector functions. And so actually it was a huge relief to us that she hadn't actually just croaked it straight away. And I said, which is, well, how long will it last? I said, you know, we don't know. We've done our best to humanize this thing and make it as human as possible. You know, maybe it'll last a couple of weeks, or maybe it'll last a few months, or maybe it'll last a year, or maybe it'll be forever. We actually just don't know. Because the lady was about in her 70s anyway. And she said, oh, well, actually 2 months is enough for me. And I thought, that's a very strange comment. So, well, why is 2 months enough for you? She said, well, my husband's dying and he'll be dead in 2 months' time, and I want to be with him when he dies. Actually, it had a huge effect on me. And she said, if I get a bit more than that, that'll be great, because I've got 2 grandchildren. I can bring to terms with the fact their granddads died and their grannies died. And actually that encounter, probably more than anything else, persuaded me that I'd got to try to do something applied. You know, I can continue looking at enzymes, or I can continue looking at the recesses of immunology, but I wasn't a medic, and I was in a very unusual position of being able to do something that most medics couldn't do by building something. And I thought, this is just, you know, when I saw her, I thought she can do nothing. She can do nothing, but maybe we can do something. So actually,
and you gave her 2 months?
Well, we gave her a year, actually. So in fact, it turned out that the tumor came back after a year, but by that stage we'd run out of antibody, or they'd given it to a number of other people. And in fact, they gave the last dose to a child instead of to her, because they felt it was ethically better. In fact, the child died and she died, which was a bit of a sorry story. But nevertheless, it made me realize that I'd got to think about, you know, it's all very well having a technology, but how the hell do you can't just keep making this stuff in the lab. Who's going to pay for it? The research councils won't pay. So you've got to engage with industry. So at what stage do you engage with industry? So I started thinking about how that should be applied. And in fact, I'd filed a patent on it to protect the MRC's position at Cesar Milstein's advice. And we decided that actually we should try to license the patent widely so lots of people could use it. And in the end, after lots of arm wrestling with the MRC, they agreed to do that. And in fact, the class of humanized antibodies is probably about the biggest class of antibodies that have, certainly in terms of sales, that have been approved at the moment. I mean, there's other technologies coming along, but it's been a very successful approach and very heavily used.
But you moved on from Campath 1.
We did.
Yeah. Because Jeremy Isaacs is a rheumatologist that worked with Herman, and he always knew when the drug was being given because all the patients got anaphylaxis. So you heard the beds shaking as the infusion was going through, which was
right. I see. Okay.
Rather intriguing. So how then do you go from that was a sort of a generalized antibody against wiping out cells.
Yes.
How do you then go for the story of TNF is intriguing because its name implies no one really knew what it did.
Well, in fact, it wasn't based on, so we started off not necessarily trying to make an antibody against TNF. What I was worried about was I saw these at that stage, we didn't know how successful the humanized antibodies would be. It took many years of going into patients to realize they do basically look pretty human, and you don't get so much of a response to them. So we didn't know how successful they would be, but I thought probably we would find at some stage ourselves limited by the response, and we better make as human an antibody as we could. So I started thinking about it. At the same time, because other people are also interested in making humanized antibodies, I came up with a very fast method of cloning them out of hybridomas involving PCR primers and cloning directly into expression vectors. So this was all my recombinant DNA experience coming to the fore. So then I started to think to myself, well, we had a company, I think Behringwerk wanted a monoclonal against a particular mouse monoclonal against a particular antigen. And I think they'd failed to make it in Germany, and they wondered if we could have a go. And I thought, well, I'll give it a go. And I remember, I thought, I've never tried monoclonal antibody technology, so I'll try it. And I thought, after about a month of being contaminated and frustrated, slow tissue cultures not growing and mycoplasma getting into it and everything, because I had no proper training. I mean, the LMB never believed in proper training. You just basically pick up a cyclostide protocol from someone and just work with it. So, but it was absolutely awful. I'd done biological stuff, but this business with tissue culture was a real problem. So I thought this is the most useless method that one could possibly have. I need to invent a better method. So decided maybe we could try to clone the genes rather than take as we were a spleen, fuse it with myeloma cells, and then isolate monoclonal antibodies, and then take the genes from monoclonal antibodies and do something with them. Why don't we just take a spleen and clone the gene straight out of it using this PCR method? So I started doing that. And that was the basis for what later became Humira. We thought, well, let's just try to clone out what we can get in spleen. But at the same time, of course, if you do that, you don't know which heavy chain goes with which light chain. And therefore you end up with either random combination or you use an artificial pairing that you've put together. But we decided we need a very rapid method. We decided that we need to sort out the screening method for this. But by going into bacteria, we realized we could greatly simplify the screens. And furthermore, by going into phage, we might be able to use that as a selection vehicle for the very rare clones that we were going to get. So we started doing that. And in the end, we were able to make a repertoire, take human lymphocytes, make a repertoire of human genes, and then select out the ones which would bind to any particular antigen, whether TNF or anything else. And that was sort of the underlying technology that was deployed in part for making Campath. Sorry, for making Humira.
So quite simply, you would have purified tumor necrosis factor in your system, and you pick out the bits of the antibody that bounds to it.
You had to stick it to a column and pour our artificial immune system on top of it. It would run through the column, and then we'd select out the ones we want. And it actually, in the end, that approach worked, and we spent some time developing that in the LMB and in the Center for Protein Engineering, which I also became associated with.
And like all these things, its application was rather serendipitous in that it was made as an antibody for patients that had sepsis. But it made the sepsis work.
I think actually that that particular, there was an interest in anti-TNF antibodies then, but I think that Cambridge Antibody, I set up a company to try to take this forward because actually there was no other company interested in doing it. So for example, when I went to talk to Celtech, their attitude was, look, this is a whole humanizing technology we've just invested in, and we've got a huge plant dedicated to it, we've got teams working on it, and now you're telling us we should use some completely other method that involves making vast repertoire of genes and then sticking these into bacterial viruses and then selecting those. Have you actually got it to work for anything? We said, well, no, but it will when they said, well, when you've got it working, come back. So in fact, by the time they might have wanted to come back, we'd already started working with BASF Pharma, who wanted us to make an anti-TNF antibody. Largely, they'd wanted to humanize it, actually, but Celtech had the exclusive license to do that. So therefore they couldn't, and therefore had to use my other patent. I said, well, okay, it's all right. That's one of my patents down. You can't use that, but I can help you find a way around it, and we can make you genuine human antibodies. So Cambridge Antibody went ahead with that technology, and in the end, it ended up being Humira.
Right. And the expansion required certain private investment. Your big people weren't queuing up to invest in you, and I think you relied upon a few individuals to help out.
Yes, there was an Australian chap in particular who provided the money. I got to know him because Fred Sanger had asked me to, after Fred Sanger stepped down, he said, well, Jeff Grigg is a good friend of mine, and he'd like to come to Cambridge, he said, to have he's just retiring at CSIRO in Sydney. And I think he needs a base, because he needs to get his lab to support him when he's over here. I said, well, we'll be getting any work out of him. And he said, no, you won't get any work. He mainly likes going to college and drinking and wining and dining and things, but he's a very agreeable person. And in fact, I put aside part of a fume cupboard for him, thinking I wouldn't get much work out of him. And in fact, I didn't, but he was a very interesting man. And apart from the habit of wanting to discuss everything over a meal, which I just didn't, I didn't have time to do it. And in the end, he heard through the Australian mafia about my new methods to make. He was there for about 6 months, and about, I guess, 4 or 5 years later, he heard about the methods I was trying to develop and the possibility I was thinking about a startup company. And he came across to Cambridge and just settled in the Garden House Hotel, which was then one of the swankiest hotels there, invited me to dinner, and then said he wanted to buy it for his company, PepTech, in Australia, which we refused. But he then said, well, if you can't let us buy the technology, we'd like to, if you want to set up a UK company, we'll invest in it. We'll take a share in it, and we'll commit to not taking it over and such like. And I managed then to persuade the MRC and Aaron Klug and other people that this would be the best way forward with this technology, because otherwise we weren't going to find anyone to take it over.
So when one reads about this stage of the career, you're very tight in keeping your patents and your hand over your inventions, but you gave away an awful lot of knowledge to other people.
Yeah, we gave knowledge in the sense that the humanized antibodies were licensed. We did have a patent. They were licensed non-exclusively for fairly modest fees, actually. But because of the sheer amount of business they created, in other words, the number of antibodies that were approved, they've earned the MRC at least £500 million over the course of their life. Now, the repertoire patents, the prize for which I got the work for which I got the Nobel Prize was, we did file patents both on the idea of making repertoires and gene repertoires, and also on the phage, the use of phage for this purpose, and those were held between the MRC and Cambridge Antibody Technology. And the MRC had an equity stake in Cambridge Antibody, so it did actually make some money out of that and from royalties on that. I don't know exactly how much, but probably not as much as it made from humanizing antibodies and making it widely licensed. But basically, there are two technologies there which I felt that we'd facilitate in some way or another, one by providing the information and the other one by virtue of trying to do it ourselves and putting the investment behind it.
Now, I've got one question to ask, and then I'm going to, Dave, if you can get the Q&As up on the screen, I'll work through those. But I was quite amused that I've seen this before, that when you look through your citation, your Nobel Prize was with Francis Arnold and George Smith, but Francis Arnold got half the prize, and you only got 25%.
Yeah.
they just work that out on importance, do they?
Well, yes and no. I mean, let's say I feel reasonably comfortable with that. I'm just very grateful to have had a share of a Nobel Prize. That's, to my mind, the key thing. But the other thing is, Fred Sanger only got a quarter of a Nobel Prize for DNA sequencing. So he had to share that with Wally Gilbert. And then Paul Berg got the other half for cloning. So the Nobel Committee have often seen fit because they've got a number of worthy people who deserve a prize. They've only got one prize they could award in each year. They do try to use 3 if possible, if they can, reward 3 people. And in this particular case, they've tended to divide it into the different bits of work they've done. So for example, in this case, it was perfectly reasonable. They had Francis was developing enzymes, we were developing binding molecules. And that's a perfectly reasonable distinction because she wasn't using phage, she was using a different kind of approach. We were actually creating completely novel binding sites. She was starting from existing catalytic sites and sort of tweaking their activity upwards. So there were quite a few differences. And I would say that in a way, her approach was perhaps more surprising than ours, that namely you can involve enzymes by quite a few sequential changes. In other words, you just have to get some weak activity and you can probably turn one enzyme into a range of other functions. So, actually it's really good work. But, I'm just happy with the fact I'm there. I'm not going to quibble about it.
Well done. Yeah. So an example of how you can be accommodating.
I'm very accommodating.
Now, I've got tons of questions here, but I think Ghislaine, we've got a big audience at the back here.
Sure.
Ghislaine, you have a question, so rather than me asking it, perhaps you can just summarise the last paragraph. Where is he?
Let's read it, John. Please go ahead.
Ghislaine said to you that what would be your vision to obtain better insights about post-translational protein modifications, in particular glycosylation?
Yes, I mean, that's a very difficult question because it's not my field. The only experience I've had of it has proved quite problematic. We tried making antibodies against glycoproteins and we had a lot of trouble doing it. That was using the phage approach. But I think it depends why you want to do it. So in other words, so whereas, for example, proteins are very precise structures, well, the basic backbone is very precise. You've got a bit of DNA, it specifies that protein will fold up. What you've got with glycosylation and other post-translational modifications is a kind of fine-tuning. And quite often it's not just one residue that's modified, it's a spectrum of them, a different class of them, And quite often it's very variable as well. It's heterogeneous, so you've got a different number of modifying enzymes that are involved in putting this together. So it's not got that sort of precision. And what you're trying to do really, in the end, is to work out what this, what I suppose, if you're a biologist, you're trying to ask the question is, what's the relevance of this, whether it's a disease state or in biological state. And a lot of that could be, I suspect, determined from surrogate markers like what are the enzymes turned on? What are the activities of the enzymes turned on? Actually, how relevant is it, that this particular, that the impact on this particular protein, that how relevant is just one specific interaction this protein is making by virtue of that glycosylation site. Some cases it'll be relevant, but in other cases it just reflects the fact you've turned on an entire pathway of enzymes. So I think, for example, in a disease state, you may find that one particular molecule, like aglycosylated IgE, is potentially pathogenic, I suppose. And so that may be the final cause, but actually what really matters is how did it end up in that state? What if you're going to do something about it, is there something you can do to boost the enzymes? To me, it isn't like that. To my mind, it's sort of why you want to know the answer. Now, of course, if you're a reductionist and you want to know the answer because actually you want to be able to explain every single molecular interaction, there's nothing for it but hard work. You've got to take every single protein, you've got to characterize it, you've got to measure its binding affinity to another partner, you've got to look at all the enzymes, you've got to look at the complete picture. That, of course, is a lot more work, and that may not be justified for what the question that you're addressing. I think there are some things even you could do there that might be interesting, or that might give a bit of an insight if you did decide you wanted to follow that approach. And that is the role of particular residues at particular positions. Now, in principle, and I'm sure in practice, you could make a series of mutations of your protein. You can obviously know you can knock out glycosylation sites, so you can determine what the effect on the biological activity is of knocking out that glycosylation site. You can also, and perhaps more interestingly, imagine putting in unique amino acids, perhaps not there are technologies now for putting in novel amino acids which could have a unique chemistry. And from that, you could use to build up, you could put on chemically related chemical chains, either single molecules or multiple molecules, latching onto a single site or multiple sites as you please, and seeing which one of those has the effect. And that's probably the approach that I would take if I were back in science. If I wanted to learn about this in molecular detail, I'd be saying, what chemistry do I need that I could readily add glycans to that would be seen to look like just a natural glycan as far as I can, and then would have a whole panel of these different glycans and couple into that residue, and to then look at the biological properties. So that's the only kind of insights I can give. But you know, one of the problems is that people, you know, I don't think I would necessarily have any greater insight than anyone in the field. I mean, my own insights have actually come from a deep knowledge of the field. They haven't come from grandstanding over somebody else's field by making some throwaway comments. They're very much only as a result of incredible hard work, deep thought, and I suspect you've probably got to work it out for yourself, John.
or Ghislaine. Does that answer your question, Ghislaine?
More or less, because if you mutate your glycosylation site, you change also the protein backbone, and in fact you are comparing with something different. And I think one way out would be to have, let's say, glycoconjugates with and without a sugar on, having the asparagine, for instance, for an end-linked structure at the right site, but then the structure with and without the glycan on it. That could be a solution. And also coming back to the point where you say the relevance of this, of course, all glycobiologists are struggling with the relevance of their glycoprotein they are studying. But I think the relevance is indeed, as you say, and also as John says, in the diseases. If you have a patient who has a genetic defect in the glycosylation machinery, this patient has a severe phenotype. So I think there is a big future in getting more details what the structures and the functions of these glycans are, because it yields strong phenotypes. And that is in fact stimulating all of us to keep the field going.
Oh yeah. But I think there's no question, it's a very difficult field. I mean, it's sort of just because of the heterogeneity, both of the glycan and also the sites it can go on, and the different properties it can give rise. In some cases, not even, you know, sort of you get a spectrum of properties. So it's even quite difficult to you know, you get a range of rate constants or something. So it's a real problem. So yeah, I'm afraid if you're trying to take a sort of reductionist view about what is this molecule doing, then I think it's a really hard work. But if you know it's involved in a disease, it's worthwhile.
Pauline Rupp behind me.
Yeah, but Greg, even so-called monoclonal antibodies have a spectrum of glycoforms. So I think we looked at Herceptin. It has 5 different populations which are differently glycosylated, and those different populations stimulate different arms of the immune system in different ways. So I think it is pertinent to antibodies to really understand why we have these different glycovariants. And we've only just got the technology now to look at an intact IgG molecule, so we know how the glycans
paired in the middle rather than just look at the released glycans. So
no, I couldn't agree with you more. I mean, I think I'm not trying to say it isn't important. I think it is important, but it depends on. So if you're trying to understand which particular immune system, which particular series of receptors it's triggering, then you're going to need to produce these as unique forms. And that's actually why I was suggesting that you might be able to do it by enzymology, but maybe you can also do it by direct conjugation, by taking some unique thing, selenium or something that be uniquely reactive, putting in the backbone and then conjugating into that with a variety of forms to find out rapidly how long you can make it, so you can sort of do a dipstick and titrate them, much as we used to do with site direction mutagenesis. The issue there is going to be how you deal with, for example, if you can do that's okay with one site, but then you're going to have some kind of series of groups that will allow you to do exactly the same thing with perhaps other sites around the molecule, because it may be pairwise sites that end up being important.
No, it's very challenging. I mean, we have a whole set of deletion mutants. So with CRISPR or something, we can make a lot of individual glycoforms of IgG now by genetic deletion. But your point is well made. You can't. It's still not that specific. It's just general over the entire molecule.
Any other questions behind me? Kelso? Claudine? Well, we're fast running out of time, which is a great shame for us. But where you make the whole monoclonal antibody scene feel so simple. No one's made an antibody now against COVID-19. No one's made an antibody that sticks to those spike proteins, etc. Is that?
No, they have. On the contrary, they do. There are antibodies. There's antibody combinations. The issue is that the issue you've got is, and in fact, they work very well in terms of blocking its going into cells. The problem is that what you would expect to happen is that you will rapidly select COVID variants which will negate that binding. In other words, with a vaccine, you've got multiple antibodies which are binding to the key parts of your spike. But with just one or two monoclonals, it wouldn't take much of a change to knock that one or both off. So at the moment, they've got two monoclonals, which is all different parts of the structure, and the hope is that that will give you some form of protection. And that was what was given to Donald Trump, unfortunately. And it seems to have saved him. But that was a molecule that was, it's much more expensive to roll out. And I think that there's a real danger if you did try to roll it out on a large scale, that you just simply evoke resistance in the population. Right. So it's really for those very small number of people, I think, like Donald Trump, and possibly people who perhaps are suffering from cancer and wouldn't normally make a response to a vaccine, who've been exposed to the virus, I think would be suitable recipients of antibodies. So I've been a bit, just for practical reasons, a bit negative about antibodies. You can do various calculations. You will never be able to make billions of doses of antibodies. I mean, just think of the costs and even the fermentation capacity. I mean, it's bad enough trying to do it with the other technologies, with the AstraZeneca, to get the scale up and the batch quality up. To do with an antibodies, it's just going to take far too long. So I think it's better for the majority to stick to vaccines. Our good fortune is the vaccines work so well, because actually, frankly, they could have been enhancing. They could have done all sorts of things. I mean, there's a cat coronavirus vaccine that was given that caused it actually to more likely to kill off the cats once they were infected. So, like dengue, you could, and in fact, it could still happen. We could still find a variant of COVID that is it actually, for which the antibodies we've already been making against the other forms could enhance its infection. So there are dangers as well. So what I'm hoping is they'll develop something which will be a small molecule which will deal with a polymerase, or they'll find ways of ramping up our innate immunity, so we hopefully don't have to face it with adaptive immunity systems.
So that will be the starting point for the next interview, but sadly we've run out of time now. An hour with you is always a great pleasure, even without refreshment. And it's been a pleasure now, and you've totally enthralled us, and I hope we've got a large audience here. I'm glad that me speaking to you hasn't decreased the audience over the hour. You know, it's been going up and down, so we haven't frightened them all off, and hopefully out of this large audience of 150 so people, there are some young scientists there that will be inspired by your early life. I hope they probably won't want to go to Ghana, but we'll try a virtual handshake, Greg.
Okay, right.
Where is it? Over here?
Okay, all right, that's fine.
So many thanks indeed, and have a good afternoon. Many thanks indeed.
Thank you very much, Tom. And good luck with the rest of it.
Thank you very much. It's interesting that no one asked any questions.
Super.
Thank you.
Super.
No.
Thank you. Really good, John.
But he's
Really good.
it's nice to have a Nobel Prize winner that talks. Although the last one we had, we couldn't stop Schwostek talking, could we?
Yeah.
You gave
Hi guys, welcome back to the third session. So in this session we're going to talk about glycosylation and glycan binding. So I'm really delighted to co-chair this session with Professor Pauline Rudd. I actually did my postdoctoral research with Pauline at the National Institute for Bioprocessing Research and Training, so it's really lovely to be sitting here as our co-chair. So without further ado, I'll introduce to you our first speaker. It's Dr. Thomas Clausen, and he graduated in 2009 with BSc from UMass in Boston before he relocated to Copenhagen, in University of Copenhagen in Denmark, where he did both his master's and his PhD. He's currently a postdoctoral researcher at UC San Diego in the US since 2021, and prior to that he was a visiting scholar since 2018. Where he also held a joint appointment at the University of Copenhagen as an associate professor. He's also busy and he has links with industry, and he's a chief technology officer at VAR2 Pharmaceuticals and VAR-CT Diagnostics. And today he's going to talk to us about GAGs and specifically the binding of malarial VAR2 CSA. So fire ahead, Thomas.
Thank you. Well, that was a very nice introduction. Yes, I'll apologize in advance if I'm a little slow. It's the first time I've given a talk at 6 a.m. in the morning, but I'll try to do my best. So today I am going to show you at least a partial completion of a project that I started as a graduate student, actually quite many years ago by now. And a project that has followed me through my career, both as a graduate student, PhD and postdoc in the lab of Aili Salenti, but also in the lab of Jeff Esco in the last 3 years. So it's not changing here. So just to start, I would like to put an idea in your head, and an idea that I have been working a lot with in the last few years. So I remember back in my undergraduate classes when they were teaching us cell biology, they would show us a picture of a cell membrane with a number of proteins inside, and then they would show little glycans coming off of that cell membrane and tell us that the functional components of the cell membrane were the proteins, and that everything looked very accessible. Obviously, glycobiologists, and I don't have to tell you guys, know that this is not true. In fact, this very thin membrane and the proteins are actually covered by very, very thick or can be very thick glycocalyx, and that everything that needs to get or interact with these proteins somehow need to navigate through this glycocalyx. And obviously this has profound effects on everything from infection by pathogens. We know many pathogens actually figure out how to interact with the glycocalyx in order to get to the proteins in the cell membrane, but also by other, whether it be cytokines, chemokines, growth factors, anything that needs to interact with the membrane somehow needs to figure out how to get through this protective layer. So this is, another way of viewing the membrane that is not often taught in school. So what I'm going to talk to you about today is malaria and cancer, actually. And when I started my undergraduate, I worked in the group of Aili Salenti, and they were working on trying to make a vaccine against pregnancy-associated malaria. So as most of you know, the malaria parasites infect erythrocytes. When in the erythrocytes, they put certain proteins of parasite origin on the surface. These functions as adhesins, allowing the infected erythrocyte to adhere to receptors in the host vasculature. And what this does is it allows them to sequestrate in the vascular bed so that they never reach the spleen where they would be cleared. Now, in placental malaria, pregnancy-associated malaria, the parasite protein that is expressed is called VAR2-CSA. And VAR2-CSA allows the infected erythrocyte to adhere to chondroitin sulfate in the placenta. What is interesting is that these parasites only adhere in the placenta, despite the fact that chondroitin sulfate is present everywhere in the human host. So there is a lot of questions to be answered in what is this? Why does VAR2-CSA only recognize chondroitin sulfate in the placenta, and what makes up the specificity, both on the protein side as well as on the chondroitin sulfate side? So that's been sort of a main project throughout my career to try and figure out what makes up the specificity. Now, as a graduate student, what I was tasked with was to look at this VAR2-CSA protein, which is a very large protein consistent of 6 complex domains. It's 355 kilodaltons, which means that for a recombinant vaccine, it's difficult to include the entire protein. So what we did was we truncated from either end and found that the CS binding site, the functional CS binding site, lies within one domain called DBL2, with flanking parts of the interdomains that are interdomains just means that they were we couldn't classify them as being specific, so or specific structures. So one thing that we started out with was to try and verify this high specificity, because at least in nature, it seems that these placental adhering parasites only accumulate in the placenta of pregnant women and not in other organs, despite the fact that they bind to chondroitin sulfate. So we developed a method where we take our recombinant proteins, which are a lot easier to work with, which includes that CS binding site, and we then panned it on different tissues. So here is tonsilous control and placenta. And true enough, as you can see, the recombinant protein only bound in the placenta, didn't bind tonsil, and we could show that it was binding to chondroitin sulfate by outcompeting with soluble CSA or treatment with chondroitinase ABC to remove the sugar. And we were able to show this for many organs. So here's binding to the placenta, heart, lymph node, and pancreas. And again, it only seems to bind in the placenta, despite the fact that chondroitin sulfate is present in all these organs, as we could show with a chondroitin sulfate or an anti-chondroitin sulfate antibody. So again, suggesting that somehow there's something in the chondroitin sulfate in the placenta that makes it unique. And we could zoom out even further, and we did this was all published in Cancer Cell in 2015, showing that most healthy organs did not support binding of our recombinant VAR2-CSA. Now, what's interesting is that when you look back in history, when these placental adhering parasites, which is a subtype of malaria parasites, were first discovered, they were panned on cancer cells. So, no one really stopped to ask why a malaria parasite that only adhered in the placenta also seemed to bind these cancer cells, and why we were able to select for that phenotype by panning them on cancer cells. And the cell they were using was a C32 melanoma. So we kind of took a step back to ask, so why is this? So here are infected erythrocytes binding to the surface of a placental cell. It is a cancer cell, but it's of placental origin. And you can see that the parasites very nicely bind the surface. They also bind the C32 melanoma cell, which has been known for decades. And we could show the same with our recombinant proteins here, showing flow cytometry binding of VAR2 to the BEVO cells, the placental cells, the C32, and we could show that the binding was of quite high affinity. So that to us was interesting because cancer cells in many ways mimic the mechanisms that are occurring in pregnancy, such as fast growth, proliferation, and migration and invasion. So maybe what we're really looking at is something that could be an oncofetal antigen, and that kind of it was what we started working on, and also was the foundation of the companies that were mentioned. So we then set out to really investigate this. And here is, again, binding of parasite-infected erythrocytes to a normal endothelial cell from the brain, showing that the parasites didn't really like to bind these cells, but they did bind a number of different cancer cells, breast cancer, prostate, and lymphomas. And with our recombinant proteins, we screened a lot of different cancer cell lines, and almost all of them had this unique type of chondroitin sulfate. And again, also using IHC, we could show that here's just a representation of tissues from different types of cancers showing binding to the cancer and little binding to the normal adjacent tissue in all these types of cancer. Now, so that was a very fast introduction to quite a big project. But what I really wanted to talk to you about today is, so what is the nature of the VAR2-CSA and oncofetal conjoined sulfate interaction? We call it oncofetal conjoined sulfate because it's supposedly shared by the placenta and cancer cells. So there's obviously two components to an interaction. There's VAR2 and there's the conjoined sulfate. So we and others since at least 2008, before I even started in the lab, had been trying to generate a structure of VAR2-CSA, but it had been difficult. Then suddenly 2 years ago, we and another group at the same time figured out how to solve the structure of full-length VAR2 by cryo-EM. And so this is the structure. I'm not going to go into too much of the specifics, but it shows that the classical idea that these domains were put on like beads on a string is wrong. Actually, all these domains come together and form a higher order structure. And just labeled here, you can see the proposed binding site kind of sticking out from the membrane, which is down here. So the other group that published at the same time with us actually were able to get a co-crystal with conjoined sulfate, not crystal, sorry, co-structure with conjoined sulfate. We weren't, but we along with Joshua Sharp, did something different. So Josh helped us do his method of photochemical oxidation of proteins, which means that we gave him recombinant proteins and placental CS. He then did oxidation of the proteins with and without conjoined sulfate to see if there were regions that were protected. And indeed there was, and we found this motif, which was actually corroborated in the other paper to be the actual binding site. The binding site is, true enough, sitting in the DBL2 domain with a slight contribution of the downstream DBL4 domain. And it is a patch of a positive charge going through the protein, as is what we see with many GAG binding proteins. So finally solved that, and it was published this year, which we're very happy about. So then looking at the other part of the interaction, which is conjoined sulfate. And I mean, I know I'm talking to glycobiologists, but just to be completely, what do you call it, good in introducing all these things, maybe I'll just take a minute to look at the conjoined sulfate structure. So in this case, I mean, the important thing is the conjoined sulfate are long linear saccharides of repeated disaccharide units. And while the backbone is rather simple in that it's just a repeated unit, an immense heterogeneity comes in by sulfation of hydroxyl groups along that backbone. And that generates a lot of charge, but also a lot of heterogeneity and different binding sites for different molecules. So the idea is that VAR2 recognizes maybe a novel modification or a specific sequence of sulfation. And that is, what we tried to figure out. So this is really a project that after Meet got taken up by Charlotte Split, she did far most of this work after she started in my group. And together we developed sort of what we thought was the best way to try and figure out what it is it's actually binding to. So we used our recombinant proteins. And in this, we generated these affinity purification columns. So we basically mobilized the VAR2-CSA protein on a column, and then we took different sources of conjoined sulfate, put it through the column, and then saw what was bound, what wasn't bound, and all of that. And we characterized it in a couple of different ways. So first, intact oncofetal conjoined sulfate was just purification of the intact chains. We also did a footprinting experiment where we treated with chondroitinase ABC in the hopes of what was bound in the VAR2-CSA binding site would be shielded. And this is then called the retained oncofetal conjoined sulfate, while what was digested off the column in this case is liberated oncofetal conjoined sulfate. So then this is all done in Jeffrey Esco's lab. We did LCMS to actually look at what is the disaccharide composition of these different isolates. And what we found, which is corroborated by a lot of literature prior to this, is that the main thing that was enriched is 4-O-sulfation. And we don't actually see anything else getting enriched. So as you can see that both there was a massive increase in 4-O-sulfation in what came off that column. We also did an experiment where we tried to separate different entities based on their affinity by doing salt dilution. And again, we see that with higher salt concentrations and therefore higher theoretical affinity to the column, we get an increase in 4-O-sulfation with very little of other types of sulfation sitting there in the high affinity ligand. Again, suggesting that that what VAR2 is really binding to is a 4-O-sulfate of conjoined sulfate. Now, now that we had that intact or retained chain, which is supposedly an oligosaccharide, we teamed up with John Amster and Franklin Leach, who had a method to sequence these by mass spectrometry. And they did that and found that retained oligosaccharide had a length of between dp12 to dp20, which also correlates with what has been previously described. They did not find a specific sequence of sulfation. Instead, they found that many different epitopes were in that pool. So we weren't able to sequence it, but we got a pretty good determination of what length it is and what type of sulfation is in there, which is 4-O-sulfation as well as a few others. So that didn't really tell us much because 4-O-sulfation is present everywhere, and certainly these lengths of oligosaccharides are present everywhere. So instead, we kind of zoomed out a little bit and then, took a different approach. So what Charlotte did was we obtained tissues from basically all the human organs we could get a hold of, and we isolated the conjoined sulfate and then did a disaccharide analysis. As you can see here, with all these different, so down here, the colon T1, these are colon tumors. So now we're also we're both looking at CS from placental origin as well as many normal tissues and here colon tumors as well to kind of see if there's a difference. So what we found was that there's not really a correlation between the disaccharide composition of the input material. You can see the placenta actually had quite low 4-O-sulfation compared to many of the other tissues. What we did notice was that there was a lot more chondroitin sulfate in the placenta as well as in the colon tumors compared to most of the other organs. So that could suggest that maybe it has to do with the abundance of chondroitin sulfate rather than a specific epitope. Another thing that was very interesting was that when we tried to separate by size, and I'll be cautious here in that we don't have standards to say exactly what size these are, but we can see that when we run the CS preparations on the gel, placenta CS had a proportion of higher molecular weight chondroitin sulfate chains, and the same was true for the tumors, and what came off of the VAR2 column had actually been depleted by the lower molecular weight structures and had higher molecular weight. So that suggested to us that maybe there's a thing in the length of the molecular weight of the chondroitin sulfate. So what we did, we could then look at the enzymes that actually elongate chondroitin sulfate, and sure enough, we found that a couple of the elongation factors, which is CHPF and CHPF2, is highly upregulated in quite a few cancers, suggesting that maybe there is actually something to it in that these tumors or placenta have longer chains. So to look at that, we knocked out CHPF and CHPF2, the elongation factors, and found that when you knock it out in a cancer cell, this is A375 melanoma cancer cells, we did see a profound decrease in VAR2 binding. When we look at the disaccharide analysis, there's not really much difference by doing it, a little bit here and there, but nothing that would suggest a difference in binding to this extent. There also was not less CS per cell. As you can see, in fact, the CHPF had more chondroitin sulfate, per milligram protein. What we did find is that when we look at the size distribution, the knockouts created a lot shorter chains compared to, compared to the wild types. So again, suggesting that length of the chain might have something to do with it. So in order to test this, we set up a model in which we used a lipid tail with streptavidin on it. And then we size fractionated placental C8S to isolate a low molecular weight species and a high molecular weight species. We then biotinylated it through the protein that was left on it, and then used this conjugate to generate, basically we could insert chains of different length into a cell with a very simple glycocalyx or with a thick glycocalyx. Here is red blood cells or Huvik cells. And then we did VAR2 binding. So here just
Anna, sorry, just in the interest of time.
Oh, I'm on last slide, sorry.
No problems.
So just showing the difference in the size of those fractions, that there was no difference in the disaccharide analysis, but when we tested binding of VAR2 to the erythrocytes, so both the long and the short chain, it could support VAR2 binding in the simple glycocalyx, but not in the Huvik cell, where the shorter chains, there was no binding to those cells, while the longer chains, they were able to bind. So what we think it has to do with is more about the accessibility of the ligand rather than a specific sequence of modifications on nitrogen sulfate. And with that, we had a lot of help with this project. Here's just the main people. Charlotte, who did most of the work that I showed here at the end, and then obviously Ailey, Jeff, Josh Sharp, and Jonathan Amster. And just to show you what San Diego is like in the fall, here's our last outing as a lab.
Nice, very nice. Well done, lovely work, lovely work. So maybe while we're waiting for a question to come through, I was thinking about, as you were speaking, do you think maybe it's multivalent, that, you're kind of looking for individual events of attachment, or do you think maybe that you have more than one mechanism going on and that might complicate it further?
So you mean more than one binding site, or?
Yeah. So you were talking about the 4-O-sulfate enrichment, but then you were also talking about other things. Is it possible that, you have a few different species having different affinities to the VAR2 CSA?
I think so, definitely. I think that VAR2 is probably going to bind most things that has 4-O sulfation on it, with a preference for high sulfation. And I think that what the last work indicates is that it has more to do with the accessibility in a complex glycocalyx rather than the actual specific sequence of modification. And that's what that footprinting showed, is that we did have many species in there of a certain length.
So what does that mean if you're thinking to bring it to the next step in terms of a product? Do you think you're going to need the large species to be therapeutic in order to have the correct conformational?
So do you mean on the protein side or on the chondroitin side?
On the protein side, yeah.
I think on the protein side, it really depends on what we're going to use it for, because it is clear, I didn't show all the data, but VAR2 does localize the tumors, and we can use it diagnostically to pull chondroitin sulfate out of certain samples. So I think that there's a specificity in the long chains, and we can use that little recombinant protein to find it. So I think that protein would be sufficient. I don't think we need the longer protein, because it does contain the full binding site, if that was what your question was.
Yeah, no, that's what I was wondering. And there's a comment up there saying in mother cells. I'm not 100% sure what they mean. In mother cells, question mark.
Maybe from the pregnant mother.
Calso was saying maybe stem cells.
So stem cells, we do have some data suggesting that with the loss of differentiation, that there is an increase in binding. Yes.
It means in the mother's cells.
Yeah. Oh, in the mother's cells. Okay. And there's another question here. Do you know if the native VAR2 CSA is glycosylated?
It is.
Okay, and can
Wait,
you
wait,
comment on?
sorry, no, I'm thinking of our recombinant proteins. No, so it's thought that native R2-CSA is not, but our recombinant proteins do pick up glycosylations from their production cells. Yeah, that's what I meant.
That's interesting. Okay, thanks a million for the talk. In the interest of time, I'll move on to our next speaker. Thank you very much. So I'll introduce Dr. Martina Mulinoff is graduated in 1992 in biochemistry from the University of Hanover. And she was trained in glycobiology with Professor Rita Girardi-Shan at Hanover Medical School. During her PhD and her postdoc, she was particularly interested in polysialic acid and its interaction with NCAM molecule, and has also done some extensive work looking at polysialotransferases and endosialidases. Since 2003, Martina has been a group leader at the Institute of Clinical Biochemistry at Hanover Medical School, and her interests are in sialoglycans. Particularly, she's developed knockout mouse models, knocking out polysialotransferases, and is also interested in O-acetylation of polysialic acid, particularly in bacterial species. And she joins the hunt for the long-searched mammalian sialate O-acyltransferase. Since 2019, she heads up the DFG research unit, Sialic Acid is Regulators in Development and Immunity, which connects 9 research groups across Germany. So good luck, Martina.
Thank you very much. So I hope you can see already my screen. Yes, I will give you a brief introduction into my topic. So as you all know, the sialic acids are 9-carbon sugars that are frequently found at the terminal position of N and O glycans, and they are also crucial components of gangliosides, an important class of glycosphingolipids. And due to their exposed position in the glycocalyx, they are involved in numerous recognition events in host pathogen interactions, and they also mediate adhesion and signaling processes. So in mammals, we have 20 different sialotransferases located in the Golgi apparatus that are classified in different subfamilies and create a huge number of structurally diverse sialoglycans at the cell surface and also on secreted proteins. Moreover, sialic acids can feature structural diversity on the monosaccharide level due to post-synthetic modifications of the parental sialic acid, which is 5N-acetyl-neurominic acid. So many of these modifications are species-specific or occur only on particular tissues or cell types. And we are currently interested in the biosynthesis and function of O-acetylated sialic acids, which is the addition of an acetyl group to, in principle, one of the 4 hydroxyl groups. So 4-acetylation seems to be absent from human tissues. So in humans, mu-5AC is the predominant sialic acid, and the most frequent sialic acid modification is O-acetylation at position 9 of the sialic acid, which can occur either as 9-mono or as 7,9-diacetylation. And as you can see, the O-acetylation, of course, affects the size and also the functionality of the parental sialic acid. And so O-acetylation can mask recognition sites for sialic acid binding proteins, as shown for particular members of the cyclic family, which are mainly immunoreceptors. And this is shown here by the crystal structure of CD22 or cyclic 2 by June Renio-Orbea. And she modeled an O-acetylated sialic acid into the binding site, and the acetyl group would clash with this tryptophan in this conserved sialic acid binding site, explaining why O-acetylation blocks binding by this cyclic. O-acetylation can, of course, also create new recognition sites, and we know that because O-acetylated sialic acids are used as host cell receptors for different types of viruses, including influenza C and D virus, porcine torovirus, but also beta coronaviruses of the subgenera Ambecovirus, as shown here for the human coronavirus OC43. These subgenera, these viruses have a spike protein. Here nicely shown the cryo-EM from David Wiesler's lab, showing the 9O-acetyl group sticking deep into this binding pocket. But these viruses also have a hemagglutinin esterase protein, and I will come back to this later on, because these proteins prove to be important tools to study O-acetylated salic acids. O-acetylation is also implicated in the regulation of ganglioside biology. This is best studied for the 9O-acetylated D-sialoganglioside GD3, shown here. This is displayed on various cancer cells, including melanoma, glioblastoma, and acute lymphoblastic leukemia. It has been shown to protect cancer cells from GD3-induced apoptosis, and it was also shown to be upregulated during peripheral nerve regeneration and to be implicated in the migration of neural precursor cells in the central nervous system, in particular in the cerebellum. So in 2015, we succeeded in identifying Cas1 as the key enzyme in the biosynthesis of O-acetylated sialoglycan structures. This Cas1 is located in the Golgi apparatus. So this was the work of Anna Maria Baumann in the lab. And this is a complex protein with 13 to 15 predicted transmembrane helices and a luminal catalytic domain, for which we did some homology modeling. So it seems to adopt an SGNH fold as the scaffold for a serine, histidine, and aspartate triad. And we were able to express this catalytic domain as a soluble protein to do some biochemistry. And we could demonstrate that Cas1 uses acetyl-CoA as donor substrate, and that the reaction proceeds via the formation of an acetylated enzyme intermediate, where the acetyl group is connected to the hydroxyl group of the catalytic serine. And if we inactivate the serine, this intermediate is not formed, and the catalytic domain is inactivated. We also searched for the acceptor substrate, and this came a bit to a surprise, because it turned out that the acceptor substrate is not an assembled sialoglycan, as we've seen that before for several bacterial O-acetyltransferases, but Cas1 acts on CMP-activated salic acid, as we've shown here by a combined HPLC and mass spec approach. And this means that Cas1 acts on the donor substrate of all the 20 cellular transferases and might work in concert with particular cellular transferases. To study this in a bit more detail and to analyze the role of Cas1 in a cellular context, we were in urgent need for appropriate tools to detect O-acetylated sialoglycans. And here we and others, so-called viral lectins, where we used hemagglutinin esterase proteins from bovine coronavirus, from influenza C virus, also from the porcine torovirus. And these proteins, they have two domains, a receptor binding domain that is specific for O-acetylated salic acid, and also they have an esterase as a receptor destroying enzyme to remove 9O-acetylated or to remove acetyl groups from the 9 position, which is important for the virus to detach from cells. The esterase can be inactivated by a serine to alanine mutation, and by fusing this to an FC part of an IgG, we end up with these very nice viral lectins. There's a recent study by Raoul de Groot and Gert-Jan Boons using glycan microarrays, showing that they differ also in the specificity for different O-acetylations. So the bovine coronavirus lectin very much prefers the 7,9-dioacetylated version, while these two, as was also clear from the crystal structure, are very specific and restricted to 9-mono-O-acetylated salic acid. Just to introduce these important tools to you. So we use them to screen silyl transferases, or to screen for silyl transferases that drive cell surface O-acetylation. And this shows you just our first hit, which is SD8-Sil6, a silyl transferase that adds an alpha-2-8 linked sialic acid onto O-glycans, forming this diacylyl motif. This was done here in CHO cells with the bovine coronavirus lectin. And upon expression of the silyl transferase, we see this nice cell surface staining. Here also shown a flow cytometry, and we use the bacterial silate O-acetyl-esterase to remove the staining and to show also the specificity of this staining. And using all this system, we meanwhile did CRISPR-Cas knockouts in 3 different cell types and could show that in the knockout we lose or completely lose the 7 and the 7-9, or the 9 and the 7-9 O-acetylation of sialoglycans, both on glycoproteins and on gangliosides. So in the next step, and that's the story I would like to present you today, is the work of Marlena Albers in the lab, where we ask, okay, we have Cas9 as the key enzyme, and we showed that it uses acetyl-CoA as donor substrate, but how is that acetyl-CoA imported into the Golgi apparatus? Because acetyl-CoA cannot freely diffuse across membranes. And there was one candidate protein, the acetyl-CoA transporter SLC33A1, to which we had a closer look how that one comes into play here. So SLC stands for solute carrier, so it's a member of the SLC family 33A. It shares sequence similarity to the major facilitator superfamily. These have typically 12 transmembrane helices and form 2 6-helix bundles. And we've done some homology modeling on crystal structures of distantly related transporters in the outward open and the inward open form to give you an impression how these transporters work. So why was this our candidate? Because SLC33A1 was originally identified in an expression cloning approach aiming at identifying the O-acetyl transferase because it has the ability to increase ganglioside O-acetylation upon overexpression. However, later studies demonstrated that SLC33A1 is strictly located to the ER and provides acetyl-CoA for N-acetyl transferases that modify lysine residues. And we asked the question, so is this just by accident if the protein is overexpressed that it spills over into the Golgi, or is this transporter indeed crucial for a salic acid O-acetylation? What Marlena did is she did CRISPR-Cas knockouts in CHO cells by different approaches. So she targeted exon 1 to introduce a frame shift mutation here. She targeted exon 3, and she also obtained clones where there's a large 12 kb deletion that spans several exons. And we used CHO wild-type cells for this approach because they express Cas9 and the acetyl-CoA transporter and have a very simple ganglioside, namely GM3. And upon expression of the silyl transferase SD8-Sil1, you obtain GD3 and also 9-O-acetylated GD3, which is shown here in an immunothin layer chromatography with specific antibodies for the non-modified and the modified GD3. And we have here standards showing that this antibody is specific for the 9-O-acetylated form. So in the wild type, and you see here in blue all the cases where we express the silyl transferase, only in this case we get GD3 and we get also O-acetylated GD3 formed. However, in the mutant, GD3 is formed in the presence of the sialyl transferase, but we completely lost the 9-O-acetylated form. This deficiency could be complemented by additionally expressing the transporter. And the same was true for the exon 3 frameshift mutation and for the large deletion, highlighting that the transporter is essential for the formation of 9-O-acetylated GD3. This is of particular interest because there are diseases associated with mutations in the transporter. And one is the so-called Hupke-Brendel syndrome, which is a lethal genetic disorder. So the patients die at the age between 10 months and 3 and 6 years of age. So they present with developmental delay, psychomotor retardation, hypomyelination, cerebellar hypoplasia, cataracts, hearing loss, and low serum copper, possibly due to low ceruloplasmin levels, because this is the main copper transport protein in the plasma. So there are different mutations, also frameshift mutation in exon 1, premature stop codon caused by a mutation in exon 3, and there is a missense mutation causing this alanine to proline mutation predicted in the second transmembrane domain. And there are other diseases, so autosomal dominant mutations. There's one Chinese family where the serine to arginine mutation was found to cause spastic paraplegia. And there's one patient with cerebellar ataxia that presented with this glycine to serine mutation. And here again, we ask whether these mutations affect ganglioside O-acetylation when we express these variants in our transporter knockout system. And that's shown here. So we have the CHO cells with this large deletion in the transporter gene. The knockout cells, although they present GD3 upon expression of the sialyl transferase ST8-SIA1, they don't form 9-O-acetylated GD3. The deficiency is nicely complemented upon expression of the human wild-type transporter, but the transporters that carry the mutations found in the Hupke-Brendel syndrome hardly complement the defect, and there is a much more compensation by the mutants or by transporters carrying the mutants found in spastic paraplegia and cerebellar ataxia. And we quantified this by flow cytometry, showing that the most dramatic effect on the transport function was seen with the mutations found in the Hupke-Brendel syndrome. The mutated transporters were expressed similar to the wild-type protein, and this is the truncated version found in this mutation here. We also had a look on the impact of the transporter in the formation of O-acetylated GD2. Here we co-expressed the sialyl transferase together with the beta-4 GalNAc-T1 transferase. This is shown here for the wild type. When we express these both glycosyl transferase, we get GD2 and O-acetylated GD2. The amount of O-acetylation increases if we overexpress the transporter, but in the knockout, GD2 is formed, but there's no 9-O-acetylated GD3, only if we also co-express the transporter, showing that it is crucial for both formation of GD3 and GD2. And this is also of interest because there are patients with mutations in this glycosyl transferase that present with spastic paraplegia, highlighting the impact of gangliosides in these diseases. So we could have stopped here. It was pretty clear, black and white. The transporter is essential for the formation of these O-acetylated gangliosides. But we moved on and we asked in other conditions, is it also essential for the formation of O-acetylated sialoglycans on proteins? And we used here the ST8-SIA6 again, which forms these structures on O-glycans. And we found that if we express this enzyme in CHO cells, we get nice staining of the bovine coronavirus lectin that recognizes mainly the 7,9-dioacetylated structures as shown here, but also the 9-mono with the influenza C virus lectin. when we had a look into the knockout, we still see O-acetylation. That came to a surprise, but we saw impaired 7,9-dioacetylation. So this is clearly reduced, but there is under these conditions, so no transporter, but there's still O-acetylation, and the question is, how can that happen? And this will be a major question for our future work. And what we speculate is that the transmembrane part of Cas1, which has not been very much characterized so far, might be involved also in the transport of acetyl groups across the membrane. Okay, and with this, I would like to summarize what I've presented to you. So Cas1 is the key enzyme in the biosynthesis of O-acetylated sialoglycans. We meanwhile also succeeded to generate knockout mice and confirmed that this is the critical enzyme in this pathway. But we also found that the acetyl-CoA transporter largely contributes to O-acetylation, in particular to the O-acetylation of diacylated gangliosides GD3 and GD2. And we are currently further investigating all the 20 sialyl transferase to show their contribution to the system. And I hope I could convince you that the O-acetylation machinery is much more complex than we thought in the beginning. But dissecting the pathway is important also to understand how O-acetylation is upregulated, for example, in particular cancers. And with this, I would like to thank the people involved in the work. So this is my current O-acetylation team: Marlena Albers, Larissa Schröter, Melanie Grove, Maike Hartmann, and Carina Weiss. So for the Cas1 story, we had contribution from Raoul de Groot. And I would like to thank our funding agency, the German Research Foundation, which currently funds our project in the framework of a research unit called Salic Acid as Regulator in Development and Immunity. And with this, I thank you for your attention, and I'm happy to take questions.
That was a beautiful story, very, very nice. During my PhD, some of our precursors were acetylated glycolipids, and we always, at the time, we were trying to deprotect the acetyl groups, but we always thought, if they're still acetylated, thank God you can still use them in a biological model. So I'm familiar with the trials and tribulations of acetylation. And one of those problems that we had was acetyl migration, and it's picked up by Nicholas Carlson there. It says, very nice. So what are the enzymes involved in 7-acetylation and sulfation? And his second question is, can the acetyl groups migrate and sulfate? So if you could ask or answer any of those questions.
So, so currently we believe that Cas1 can transfer both the acetylation in the 7 and in the 9 position. And what we saw is that we will need the luminal catalytic domain and the transmembrane part to get 7,9-dioacetylation. The acetyl groups can indeed move from the 7 to the 9 position if the 9 position is free under slightly alkaline conditions, and it can be also enforced by elevated temperatures, which is really also another hurdle in analyzing these O-acetylated sialic acid structures. Yes.
Very nice. And we have, and to the other enzymes involved in O-acetylation and sulfation. Oh, you mentioned that already. Sorry. Yeah. For Ulla, great presentation. Thank you. And do you know of any acceptor preferences of Cas1 sialic linkage and/or other types of glycoconjugates?
So, as I mentioned, Cas1 acts on the CMP-activated sialic acid. So it does not act on the assembled sialoglycan. So it works together with particular sialyl transferases to create then O-acetylated sialoglycans with different linkages. So this is the picture that comes out of our study here.
And then you mentioned for the transporter that you were talking about with, is it Huckel-Brundle syndrome? So in terms of the treatment types, will they be targeting that transporter, or are there any current treatments?
No, not as far as I know, no.
And is that something you're considering?
I think we need a bit more work here to know exactly also the structure of the transporter, of course, but that would be also an aim of the future, yes.
Okay, and there may be further questions, but I can't see them on the screen in front of me, so.
No, I think.
Okay.
Yeah, no problems. Okay, so without further ado, I'll pass you over to Pauline for the next speaker. Thank you.
Thank you. So.
Oh, hello. Hi.
Hello.
We have you, but not your slides.
Yep, they're coming right up.
Excellent, excellent.
There we go.
Okay, so actually one of the really basic aims of the Jenner meeting is always to have a lot of speakers for early stage researchers and to encourage the next generation. And I have to say this meeting is so impressive because all of the talks have been really excellent, and I think the future is in very safe hands with all of your contributions. And I've been looking at some of Elle's work, and I believe that you're exactly the same. We're really looking forward to your talk. So Al got his PhD in microbiology and immunology from the University of British Columbia, and he did his PhD in microbiology and immunology at the same place at the University of British Columbia. And he then went to England, and he worked with Gideon Davies, which is a wonderful opportunity, I think, to have been able to work with Gideon. And after that, he became a full professor in the Department of Biochemistry and Microbiology in the University of Victoria. He's got many interests, but particularly around the area of structural biology, glycobiology, and its application to the analysis of protein-carbohydrate interactions in enzymes that modify glycans or proteins that non-catalytically bind carbohydrates. And particularly he's interested in host microbe interactions. And he's received a number of awards over his time: the Stacey Memorial Fellowship, a Michael Smith Foundation Scholar Award, and now presumably he's living from a Tier 2 research chair. So we're really looking forward to your talk. Glycopeptidases, unique catalysts that recognise post-translational glycan modifications as a requirement for hydrolyzing peptide bonds. It's all yours.
Thank you. I'm not sure I can be considered an early career researcher anymore. I feel a little long in the tooth, but I appreciate that. That's wonderful. Thank you. Well, good morning, everyone, or at least it's morning here. I'm coming to you from the west coast of Canada through the miracle of technology. And so I'd just like to begin thanking the organizers for the opportunity to speak to you today, and a slight apology for providing this really long and dry title. It was one of those situations where I was asked for the title, I delayed, and then I panicked and just provided whatever came to mind. And so really I should have provided this. It's a little bit more catchy, and it regards what I'm going to talk about today, which is peptidases that literally have a sweet tooth. And so I'd like to begin this by a discussion, or beginning a discussion of this particular question, which is: how are hairy glycoproteins hydrolytically degraded by enzymes? And just as an example, we all know that glycoproteins perform a variety of very important biological functions. And I've provided my main example here as mucin, which are highly glycosylated proteins whose biological function is typically to provide barrier functions at epithelial layers, and they're coated with sugars. Now, of course, the degradation or turnover of glycoproteins is part of normal processes, and it's part of also pathogenic processes. We also know that the glycosylation, more often than not, tends to stabilize these proteins or make them resistant to degradation. And so this is where we get to this question of how are hairy glycoproteins degraded by enzymes when they're protected? Now, the obvious answer, the answer that seems obvious to me at least, is we have glycoside hydrolases, or enzymes that give the protein a haircut first and expose the peptide backbone, and then proteases go to work. And in fact, this was the preconceived notion that I had when we stumbled into this. But what I want to talk to you about today is an aspect of this. Surely that is a component of how they're degraded, but I want to talk about an aspect of this, which is actually far more interesting, and we backed into it quite by accident. And so the story begins when I started my lab about 20 years ago, and I became interested in this bacterium Clostridium perfringens, and it gained some fame as a cause of gangrene, particularly during the World Wars. However, it's actually a normal commensal of the human and animal gastrointestinal tract, and it's an opportunistic pathogen. So under certain conditions, it's a major cause of gastroenteritis. And so it inhabits this environment, this mucosal layer. And so we spent many years investigating this interaction of Clostridium perfringens with things that might mimic the mucosal layer. And we ended up studying a variety of different enzymes with complex molecular architectures, whose role was to destroy glycans that would be present on glycoproteins like the mucins in this environment that it inhabits. And so we spent well over a decade doing this in total. And during this time, one of the tools that we used is to catalog what these enzymes are. And so this is basically a one-dimensional schematic of what these glycoside hydrolases look at. So just to break this down for you, the gray pieces here are the domains of these proteins that actually perform the catalysis, so the hydrolysis of a glycosidic bond. And what you'll notice is that they're fairly large and complex, and they have these other ancillary domains. And it's not terribly important, but you can see a column here where these things all have a variety of different activities. And this is all consistent with degradation of complex animal glycans. And so our goal was to study how these things work together to degrade glycans. Now, one of the other interesting components of this program was to find enzymes, new enzymes that might have other activities. And so one of the approaches that we took was to search for ancillary domains in proteins of unknown function. So what you'll notice here is in blue and red and green, these are carbohydrate binding modules. So these are modules that facilitate non-catalytic interaction of the proteins with glycans. And so these made good markers for things that might interact with glycans. And so we used this approach of searching for them. And we came up with a couple of interesting examples in the genome of Clostridium perfringens. And this was actually a bit of a head scratcher. So here are the CBMs in red and blue. And in the middle here is a domain that was presumably the catalytic domain, but it didn't really look like a glycoside hydrolase domain. It looked more like a protease. So it had a gluzinsin type motif in it that would be consistent with proteolytic activity. And so that was the hypothesis that we started with. And we tested every typical protease substrate you could possibly imagine and failed miserably to support that hypothesis. We then backtracked and thought, okay, well, perhaps these are very strange glycoside hydrolases, and tested every possible glycoside hydrolase substrate we could think of, and again, failed miserably. Eventually, we made what you might think of as a breakthrough, and we found activity on bovine submaxillary mucin. What this is this is an SDS page gel that is stained with a periodic acid Schiff stain. So it's staining for the carbohydrates on bovine submaxillary mucin. And what you can see here is when we have wild type peptidase or protease, that's ZMPB, this protein here, it destroys this. Now, if you looked at the Kamasi staining, you'd still see stuff here, but what you see when you stain it with periodic acid Schiff stain is it disappears. And so this was at least a step forward that we could identify some kind of substrate, something that this was active on, but it wasn't particularly informative in the sense that we couldn't tell whether it was active on carbohydrates or protein. The real breakthrough came when the postdoc who was working on this, Liz Ficko-Blaine, solved the structure. We're a structural biology lab, and so she solved the structure of the catalytic domain of this protein, and it looks like this. And the salient feature here is this thermolysin-like structural motif in the center here, and then this catalytic center, which is the typical gluzinsin catalytic organization with the catalytic glutamate there. And so this definitely looked like a peptidase, but this wasn't really the aha moment. The really aha moment was when Liz did the unthinkable and did the experiment that her supervisor said would never in a million years work. She soaked crystals of this protein in what was basically cow snot. And there was no reason that this should have worked, but it did. So she soaked crystal for 2 weeks in this. And the result was something that was so stunning to us, we actually had a hard time interpreting what the electron density was in the active site. After a bit of pondering, we eventually realized that we could model in this glycopeptide, which is this alpha-2,6 sialylated core-3O glycan in a short piece of peptide. And what's important here is this is the bond that appears to have been cleaved by the enzyme. And we can see this a little bit better in this slide, where here's the peptide. And this is the catalytic center there. And so this nitrogen on the terminal threonine is poised perfectly for catalysis here. But the real stunner came when we turned this around and looked at what was happening with the glycan. And you can see a series of very specific interactions between the glycan and the enzyme active site. And this glycan packs up nicely against the surface. So what this looked like was an enzyme that was cleaving the peptide backbone, but was actually recognizing the glycan as well, which was quite a surprise. Now, armed with this information, and around the time we were doing this, we somewhat got beaten to the punch in that there was a paper published which reported activity of this enzyme from Bacteroides theta iota omicron, which they reported to degrade mucin. It was primarily a bioinformatics paper where they reported some features of these. And the interesting thing was that this family that they defined incorporated several families from the MIROPS peptidase database. And our enzymes fell over here on this wheel of the species. That year as well, there was another example that was published and reported to degrade O-glycosylated surface receptors. So rather than be discouraged, we decided to use this to our advantage, and we incorporated BT4244 and IMPA as model systems that we could study. And we put these into our structure function pipeline. And the most salient result that we got here was when we tested these three enzymes on custom-made glycopeptides, the naked glycopeptide, and then a series of three different structures. And what you can see is, first off, none of them are active in the absence of the glycan, supporting the fact that these things need a glycan on these peptides. The other interesting thing was that they were all different on a different pattern of glycopeptides. IMPA is a beast, whereas ZMPM was a little bit more specific. The other key finding through mass spec was that all of them cleaved the bond, the peptide bond, immediately N-terminal to the glycosylated residue. And so this was a common feature of all three. And so synthesizing this with all of the other structural information we had, we could propose a model on base of how these peptidases work. And so it incorporates this concept of how peptidases accommodate their substrates, where there's S subsites that accommodate P residues in the peptide or protein, and there's cleavage between P1 and P1 prime. But in this case, we have P1 prime, which actually has a no glycan on it. And there's specific subsites that accommodate those residues. And these appear to be different from enzyme to enzyme. So there's variation in what glycans are recognized. And so this leads to the question that we're exploring now is, do both peptide sequence and glycan structure influence O-glycopeptidase activity? You can imagine that this is quite a monumental combinatorial challenge. And so this story has expanded into a saga. And so to explore both the glycan diversity, or the ability for these things to recognize different glycans, as well as the potential to recognize different peptide sequences, we're now looking at upwards of 30 of these different enzymes. What I want to note is some of them fall into MIROPS M60, that's these branches, and then others fall into other MIROPS families. Now, these are all demonstrably related at the amino acid sequence level, but there's a continuum of relatedness. And if you take the extremes, you get percent identity as low as 10%. So we've got a lot of sequence diversity to the point where you could almost say they're unrelated. We also chose these examples because there's expansion of some of these enzymes in particular bacteria. In particular, you see the BC, the Bacteroides, there's 12 examples of this in here. And if we work under the premise that we don't have redundancy, expansion to cause redundancy, then you might hypothesize that these things will likely have different specificities. So this is where we're going now, and we're beginning to flesh this out. This, of course, like I say, is a saga, but it gets even more complicated than just recognition of glycans in the active site. So I'm pointing to this giant structure here because this also was a surprise. So this is ZMPB showing you again, and this is the 1 or 2-dimensional modular architecture of this enzyme with this orange domain here, which is the peptidase domain. We've managed to solve the structures of all the different pieces, and this took many years, and part of the problem was that big pieces of it were difficult to crystallize, but we managed to do it. And by overlapping all of these structures, we can generate a model of the whole thing. And then we can validate the flexibility or lack of flexibility by using a variety of techniques like small angle X-ray scattering and normal mode analysis. And in fact, this is fairly inflexible protein. So this is likely a good model of what the protein looks like in solution. And so why is this important? Well, here's the peptidase. We have these ancillary domains here, which non-catalytically interact with glycans, and we verified this experimentally. When we look at this structure from a different angle, what we see here is that these active sites or binding sites all fall roughly in a line, and in fact, these are sitting on the plane of the screen that you're looking at right now. So they're all on the same plane and on one line. And this seems particularly relevant to us when we thought about some recent data probing the structure of mucins, which of course the structure is somewhat of a mystery, but this low resolution cryo-EM study revealed that, at least in this particular protein, we have these mucin domains which are on repeated 50 nanometer centers. And so these blobs here are thought to be particularly hairy glycosylated regions, with the intervening region still being glycosylated, but a little less so. And so there's this repeating motif. It was quite interesting to find that these functional portions of this enzyme were actually also on 50 nanometer centers, or multiples of 50 nanometers, whereas the active site was on almost exactly half of that, suggesting or evoking this idea that this protein sits down nicely separated to interact with the domains where the mucin domains, where the catalytic site would cleave somewhere in between in the slightly less glycosylated region. So it's a very intriguing correlation between the structure of the protein and the substrate. And so returning back to this question, how are hairy glycoproteins hydrolytically degraded by enzymes? 20 years ago, we started with this idea that glycoside hydrolases trim down glycans, and we didn't work on the peptidases, but you can throw this in. And certainly this is one mechanism to reach glycoprotein degradation. What I can't tell you is how efficient it is. What I can tell you is that these O-glycopeptidases are very efficient at degrading glycoproteins. Many of them completely obliterate the glycoproteins that you give to them. And this is my little note to myself. Of course, I'm biased towards bacteria. I'm a trained microbiologist. But I think that this phenomenon is going to be far more widespread than we think. So this is a table I borrowed from a nice review by Stacy Malacher. And this is the group of enzymes that I've been talking about. There's a variety of different enzymes that fall into other families which have recently been discovered to be specific O-glycopeptidases. And I think as we go on, we're going to find more of these in other organisms, including animals or eukaryotes. In fact, I think there's many matrix metalloproteases that display the features of O-glycopeptidases as well. And so just by way of summary here, how do we cope with hairy glycoproteins? I think it's a fairly complex process, which involves glycoside hydrolases, normal peptidases, but I think O-glycopeptidases are going to be found to play a major role in the turnover of these glycoconjugates. And then just to finish up here, I want to tantalize you with this concept here that what we have is a superfamily of enzymes and a variety of different families as well, where we have specific recognition of glycans and cleavage of the backbone. And I think we're going to find that some of them are very specific. And does this give us a tool, something like restriction enzymes for glycoproteins? And already these tools are being used in mass spec analyses. And so with that, I'd like to acknowledge the players and the past, the present, and the future of this this research program. There's many of them to thank, but some special shout outs to Liz, who I mentioned, and Ben Pluvinage has been playing quite a strong role more recently in the project. And so with that, I'd like to thank you and invite questions.
You just started to talk a little bit about its application in mass spec. I mean, do you think you could develop this into a major tool for analysis of mucins?
Sorry, the audio kind of cut out a little bit there.
I was just wondering, you just touched on it at the end. You know, do you think that this could be developed into a major new set of tools for mucin analysis? Which, you know, some of the problem is that once you remove a sugar or a part of it, the conformation changes, and so then you have another enzyme coming in. But it seems like impenetrable to actually understand if there's any order there, or whether it's completely random process where enzyme is just by serendipity, some enzyme comes in that's useful and chops it up. I mean, do you think there's any pattern in these enzymes, and do you think you could use that as a tool in glycopeptide analysis in mucins?
Ultimately, I think yes, and people are using them already to study glycoproteins. So they'll it simplifies the problem where you can break down your glycoprotein specifically with these enzymes and then examine the products by mass spec. I think once we understand more about their specificity, how these tools can be deployed will become clearer. And I mentioned Stacy Malacher. Her group is actually, this is one of the things that she's been working on, is actually how to use them as tools in mucinomics, as she calls it. So I would keep an eye out for the work that she's doing in that respect.
Yeah, we've come a long way from the bottle brush model, haven't we, really?
Yes, indeed.
There's a question here from Jørgen Larsen. Thank you very much for an interesting talk. Do these peptidases show a specificity for serine, threonine, or lysine O-glycosylation? Oh, tyrosine, sorry. Tyrosine O-glycosylation.
All of these seem to be equally active, whether it's serine or threonine that's glycosylated. The other potential O-glycosylations, to my knowledge, haven't been tested. We certainly haven't done it. I don't think other groups have. But based on the structural analyses that we've done, it looks like they will be very specific for serine or threonine O-glycosylation, at least these enzymes.
And presumably you're looking at the interaction of the microbes with the new epitopes that will be formed once you get these cleavages. Is that something you're interested in?
There's lots of interesting things to do. There's only so much one can do, unfortunately. But one of the reasons for giving talks like this is hopefully to get other people interested in it, and maybe people will start using these enzymes to probe questions like that.
That's right. Thank you very much. I mean, this is such a difficult area to work in. I'm full of admiration. Thank you very much indeed.
Thank you.
So now I think we're going to be entertained by Ryan Flynn. Is that right? Hello, Ryan.
Good afternoon.
Good afternoon. Would you like to load your slides?
Just going to get that started here. All right. I think everyone can see it.
Okay, so Ryan is a native of New Jersey. He completed his undergraduate training at MIT with Phil Sharp and subsequently moved to Stanford, where he worked with Howard Chang, developing methods to study RNA-protein interactions. He then won the Weintraub Graduate Student Award, and as a postdoc, he decided to change fields to learn both chemistry and glycobiology with Carolyn Bertozzi, which was a very smart move, I think. And he went to her lab at Stanford as a Damon Runyon Cancer Research Postdoc Fellow. And at the beginning of 2021, he opened his own lab, went to the Boston Children's Hospital in the stem cell program and the Department of Stem Cell and Regenerative Biology at Harvard University. So his lab is currently focused on advancing methods and mechanisms surrounding the glycoRNA molecule, which operates at the interface of RNA biology and the secretory pathway. So Ryan, we're really looking forward to your talk, where the title's there, GlycoRNAs, Bridging the Gap Between Glycobiology and RNA Biology. Thank you very much.
Yeah, and thanks everyone for coming either in person or or via Zoom. I'm excited to share the work we've been doing. So as mentioned, I'll discuss the results of the glycoRNA and hopefully have some time for questions at the end for outstanding questions or new ideas that we've been thinking about. But to begin, I just want to put a little bit of a frame of reference on where I came from to come to this question and start to address it. And as mentioned, I sort of trained as an RNA biologist. And so when I think about RNA, I actually think about RNA as actually a regulator of biology. And so I think we're all now, of course, familiar about its mRNA, its coding potential. Probably many of us got mRNA vaccines, and that's really useful. But RNA does a lot of other really cool things. So RNA is actually the central scaffold and catalytic core of the spliceosome, which cuts and religates RNAs during their biogenesis. RNA is also a scaffold in the ribosome, helps to decode the actual sequence information in the mRNA, and then is also the catalytic mechanism to produce proteins. And of course, RNA can also fold into really complex structures to sense different environmental statuses, like the concentration of small molecules in the context of riboswitches, which can then change gene expression. And so the idea that I'm just trying to communicate here is that RNA is this incredibly diverse and versatile molecule that is used across all of life. And so I think it's a really exciting molecule, and was thinking about ways that we could learn new functions for RNA. And so when I was thinking about what RNA could do, one question is often, where are these molecules? So form can often follow localization. So if you're in the kitchen, you might be doing something than if you're in a living room, even if you're the same person. So same thing is true for RNA. And so then the question was, where is RNA? Where does RNA go inside of a cell? So of course, RNA comes from the nucleus, it comes from our DNA. And so there's nuclear mechanisms of RNA biology. It's transported to the cytosol for translation and folding and regulation and degradation. So there's cytosolic mechanisms. There's translation on membrane organelles like the ER for the production of membrane and secreted proteins. So there's on membrane mechanisms of RNA. And there's of course a lot of work now being done where we've learned that RNA can be packaged into vesicles. But what was interesting and sort of specifically important in my framing of this is that these RNAs are actually, from a topological sense, still cytosolic. So they still are contained within the cytosolic side of these membrane vesicles. And so when I was thinking about this again, I wanted to understand if there was ever a case where RNA could go inside the luminal spaces of membrane organelles or membrane vesicles. And when I wondered about this, I kind of realized I didn't know much of the biology or the function of these organelles. This won't, of course, be a surprise to this audience, but these organelles are really important for making glycoconjugates. And so a high-level view of this would be that when cells want to make these conjugates, they produce and import carbohydrate polymers, glycans. They bring together lipids and proteins and can then form these glycoconjugates, glycolipids or glycoproteins, and together through the serial action of a lot of these organelles and different chaperones can move them through the vesicular network to be presented on the cell surface or secreted. And so the luminal spaces of these organelles are really important for this biogenesis and trafficking again. And I kind of wondered, again, would RNA or could RNA be a part of this process? And it seemed to me that whenever you got a lipid or a protein in this space, it became a glycolipid or a glycoprotein. And so I thought, kind of as a trivial or a simple view, if RNA also went inside these luminal spaces, maybe it would also just get a glycan. Not thinking too deeply about it, I just thought maybe this would be an interesting way to ask if RNAs could ever get into the luminal spaces. And I thought to use glycans as actually the readout for this localization change, this transport into the space here. And so we took a chemical biology approach to actually ask this question and used these azido sugars. In our case, we used mainly azidomannase, so an azidomannase. So we used mannase, cartooned here in this purple diamond. And we did a pretty standard experiment where we just took cell culture cells, added mannase, and let it grow on the cells. And then rather than extracting proteins or lipids, we instead collected the RNA fraction of cells. And through, of course, the click chemistry technology that Carolyn helped to sort of pioneer with the copper-free structures, we could assay for the presence of any of these mannase molecules in our RNA sample. And so if we did that and then ran our RNA samples on a gel, we could ask, did we see any signal, and where did the signal migrate? What did it look like, if there was any, with the RNA species from a cell? And so just as a particular example of many, I'm showing you here just a total RNA gel, just to kind of get a sense for what the RNAs are looking like. And so if we run out the total RNA from these two cell lines, HeLa cells, a cancer cell line, and ES cells, embryonic stem cells, you can see that you get these three major bands, the 28, 18, and small RNA pools of RNA. And of course, because this is RNA, staining for total RNA with the cyber reagent, you can totally destroy the signal by adding RNase. So that's not surprising. But what was surprising, of course, is that if you transfer this to a membrane, an image for that mannase reporter, we saw signal. We started to see biotin signal as imaged with the streptavidin reagent. And it was really robustly expressed in this high molecular weight region above the 9 kb marker of the ladder. And there was differential expression. So the HeLa cells looked different than the ES cells. And I think critically, the signal was fully sensitive to nuclease, so fully sensitive to RNase, suggesting that there was some glycan RNA biomolecule that was sort of maybe unknown before and that we should begin to start to address. And so, kind of cutting a little bit of the data short, but we did a number of things to try to prove out that this was kind of making sense to us. But one of the major things we wanted to do was to understand which RNAs were we actually labeling. We can prove in this blot that the signal is dependent on RNA, but which ones were actually being modified. So instead of blotting with the streptavidin reagent, we used streptavidin beads to pull out this signal and could sequence those RNAs. And so we did this from both of the cell lines. And I'm going to show you a sort of a comparison of the enrichments from both of those cell lines. And what we found was actually that a select group of small non-coding RNAs were those that were modified. And so just walking you through the plot on the right here, if you look on the x-axis, you're looking at the enrichment of RNAs that are glycosylated in the ES cells. And on the y-axis, you're looking at the enrichment in HeLa cells. And so immediately you can appreciate a sort of diagonal line going up and to the right, which suggests that the enriched RNAs are quantitatively the same or similar between these two cell lines. And so this was sort of interesting because these are very different cells. They're grown in different media at different densities. And yet the cells seem to pick the same RNAs, again, in a quantitatively similar manner across these cell states. And for some specifics, of course, we're looking at these types of RNAs like SNO RNAs, small nuclear RNAs, and yRNAs. And these are highly conserved, highly abundant, non-coding RNAs that are found to be important for a variety of sort of basal cellular activities, but were selectively enriched in this context. And so, again, just to summarize, what I've basically told you so far is that there's a series of these small RNAs that are coupled to a glycan. But right now, I've been making a lot of assumptions about what this biotin signal is, what glycan it could be, by just assuming that the man-NAS is behaving like we think it should in our assay. And so in some work I won't go into detail we kind of could narrow it initially down away from O glycans to this signal being generated through N-linked or N-linked like glycans, and could prove out that actually the signal we were seeing with the man-NAS reporter was, for example, sialidase sensitive, which suggested that it was actually looking at an oligosaccharide rather than a man-NAS in some way, or a sialic acid in some way, sort of directly coupled to the RNA in some other novel way. But to start to like confirm a little bit around this N-glycan idea, we started to use some well-established chemical inhibitors of the different steps in N-glycan biosynthesis. And so, as everyone here probably knows, the OST is the major catalytic complex in the ER to produce N-glycans on proteins. And if we inhibited OST with a small molecule, we could, in a concentration-dependent manner, totally reduce the amount of glycoRNA signal, as seen in this top plot here, imaged in streptavidin. And if we go sort of later in the process down into the Golgi and start to inhibit monosadases with either kifinesin or with swansinine, you can see an interesting pattern arise where both of these second and third drugs are able to reduce the amount of glycoRNA signal that we get, but they also start to change the molecular weight. And it's interesting to think that the ability for these monosadases to actually operate and change the glycan configuration, the actual oligosaccharide structure, may indeed communicate to us in the blot how these signals are migrating in the gel. And this is where we started to first get our understanding that the migration of this glycoRNA signal is highly dependent on the precise glycoforms that we're getting coupled to the RNAs. Now, to move into a little bit more detail on the glycan side, we eventually were able to optimize a PNGSF release assay where we could analyze the RNA, the glycans released from RNA with LCMS analytics. And what we did was we took 3 different cell lines, isolated the peptide fraction or the RNA fraction, and then produced PNGSF released glycans and analyzed for the glycan compositions. And we could cluster those compositions in a principal component space. And what we noticed was that the RNA glycans in blue clustered really close to each other and conveniently, sort of for us, away from the peptide glycans. So this is telling us that the composition or the preference of glycoforms on peptides is different than that on RNA. And when we looked deeper into what were differences, what were some specific differences that we could think about and try to understand, we found that in many cases, the RNA glycans contained either more fucose residues in red or more sialic acid residues in purple here. And this was interesting, of course, because these sort of point directly to specific types of ligand receptor interactions that could potentially speak to functional outcomes for what the RNA glycans are doing. So this led us to a sort of an updated model of what a glycoRNA might look like, where we have these more elaborated, more complex N-linked like glycans with fucose and sialic acid. But I think one of the last things that we were initially thinking about and I want to describe here is again, where are these things? I started off by saying that the localization of molecules can really influence their function and vice versa. And we thought that maybe glycoRNAs might be in a new place, and that could impact either the glycan or the RNA in its sort of functional role. And to begin to address this and think through where these could actually be, we try to take a page out of where all the other glycoconjugates go. So we figured, at least initially, that glycans may be driving a lot of the localization of these glycoRNA conjugates. And so, of course, glycoconjugates often are chaperoned, like I mentioned, to the cell surface or the extracellular space. And so we wanted to set up an experiment where we could assay for the possibility of RNAs and glycoRNAs being on the cell surface. And so we set up the pretty similar experiment to that very first one I mentioned, but stopped one step before just homogenizing the cells, and thought that if we could cut all the sialic acid or most of the sialic acids off the cell while it's still alive, we could assay for extracellular accessibility of the glycoRNAs during sort of live cell growth, right? And so what we could do is treat our cells with the MANAS reporter just after they're cultured for about 24 hours. We could treat them for an hour with sialidase while they're still alive, use the membrane as a barrier so that the sialidase isn't getting inside, and then wash everything away, perform our RNA extractions and our blotting. And the expectation would be that if indeed RNA was on the cell surface, if the glycoRNA was on the cell surface, this sialidase treatment would destroy some or much of our signal. And that's actually, like, remarkably, honestly, what we saw was that if we did take cells and we treated them with sialidase while they were still alive, we really dramatically reduced the amount of glycoRNA signal we could recover. And this was really reproducible across a number of cell lines, whether they were adherent or suspension. And what was kind of interesting was that in many cases, not only did we get sensitivity to the sialidase during growth, but it reduced the signal near to zero. And so it showed that there was likely the bulk of the signal we were looking at is actually presented on the cell surface rather than inside the cell. So one hypothesis, of course, that comes from this is that, or sort of like just the logical conclusion of this is that glycoRNA is on the cell surface. And what that would mean experimentally is that you should be able to do a pretty trivial experiment where, rather than using this blotting technique and having to treat cells with the MANAS, you could just use an anti-RNA antibody to perform a flow cytometry experiment and see these RNA ligands on the cell surface. And so that's what we did. And what we could find was that if you took cell cultures, like in this case, we were using HeLa cells, and stained them with an anti-RNA antibody, you could get a shift in the population. And so some of the cells were actually expressing in this population what appeared to be RNA ligands on their surface. And this signal was really, importantly, it was sensitive to RNase. So showing that if you treated the live cells with RNase, you could cut those ligands, and that this RNase activity was recovered if you could previously inhibit the RNase A, showing that it is actually the enzymatic activity of the RNase A that was removing this signal in the third plot. And so the last kind of functional piece of data that I want to show and kind of hopefully think more clearly about what we think they could be doing follows this idea, but begins to think about other aspects of this molecule that we could assay given the model on the right. So you can see here that again, these glycans, many of them have sialic acid moieties. And so we thought that thinking through how these glycoRNAs may or may not interact with Siglec receptors could be very interesting. And Siglecs, of course, are a large family of glycoreceptors focused on recognizing sialic acids, and they're highly expressed and selectively expressed on specific immune cells in the body. And they, of course, read the presence or absence of sialic acid and then communicate activity changes to the cells that they're expressed on. And so we bought recombinant versions of all of these Siglec receptors and began to set up flow cytometry experiments, staining cells with these Siglecs with or without a pretreatment with the RNase, with the idea that if there were Siglec ligands that were glycoRNAs, RNase treatment would reduce their binding ability. And what we saw was that in most cases, actually, like Siglec 7 here as an example, there was no change. So there was robust binding to the cell surface, but RNase had no impact on the binding, suggesting that these were likely all glycolipid or glycoprotein ligands or other glycoconjugate ligands. But what was really exciting and honestly pretty unexpected was that Siglec 11 and Siglec 14 had the ability to bind the cells while they were alive, but this binding was highly sensitive in the red outlined traces, shifting to the left with the addition of RNase. And this suggested to us that at least with these two Siglecs on HeLa cells, their ligands may be, at least in part, glycoRNA, cell surface glycoRNAs. And so just to sort of conclude and happy to take questions afterwards, I think our data starts to point to the idea that not only do we have glycoproteins and glycolipids on the cell surface, but there are these new class of molecules called glycoRNAs. And these are composed of a series of small non-coding RNAs that are highly conserved across all cells and many life forms. The glycans on RNAs contain sialylated and fucosylated N-glycans, N-linked like glycans. These RNA glycans are expressed in many cell contexts, and so we've been looking very carefully in human, many eukaryotic systems, so human, hamster, mouse. We've been expanding our work into zebrafish recently. And this, of course, as I mentioned at the end, the sort of developing evidence and more work ongoing on its presentation, expression on the cell surface, on the external surface of the cells. And of course, we're working very hard and making some progress on understanding the precise nature of the linkage between the RNA and the glycan. This is a sort of a critical piece of information that we are working on at the moment. And so just to finish up, I'll thank Carolyn, my mentor as a postdoc, who was an excellent partner in figuring all this out and working on this project. The people on the left were people who collaborated with me at that time. And now, as mentioned, I've opened up my lab here at Boston Children's. We're all focused on figuring out why and how these things work. And yeah, again, happy to take questions or talk through other ideas. And thanks for everyone's attention.
Oh, that's such an exciting finding, really. It's a brilliant piece of research. There's a question here from Yongbin Choi. I'm sorry, I'm sure I'm not pronouncing that properly, but he says, she says, such a great talk. I'm curious as to how the glycoRNA can be stable in the outer membrane.
Yeah, so, I think there's a couple of things we've been thinking about. Cells secrete proteases, cells secrete DNases, cells secrete RNases. People feel like RNA is very labile, but there's actually turnover of all the biomolecules. And it's a question of dynamics, which we don't yet understand. And so I think that RNA may not be necessarily special in its some sort of ability at some kinetic level to be existing there, but also in general, small RNAs are relatively nuclease resistant. There are many fewer double-stranded nucleases than there are single-stranded nucleases. We think that these RNAs are bound by proteins. Most RNAs are bound by proteins in all contexts, and so protein-bound RNAs would be more stable. And also the glycan itself would afford additional nuclease or metabolic stability, because it would block binding and recognition through other means of turnover. So, in short, these can be digested, these can be turned over, and they're probably not infinitely stable. But I would say they're more stable than, for example, an mRNA. And you can just think about an mRNA will break if there's one point of failure, right? If it's not the entire many kb long RNA, you don't get your protein. But these are structural RNAs. And so even if you were to nick it, even if a nuclease came and cut it, it may still adopt the same structure or a similar structure. So I think there's some wiggle room around the templates that we're working with, but it is the case, of course, that biomolecules are labile to their metabolic pathways or catabolic pathways.
Yeah, thank you. We've got a lot of questions. So Gizem, I think you have.
Yes, thank you for this very nice talk, and I think I must agree with your comment that you just gave about stability imposed by eventually an attached oligo or monosaccharide. In fact, that is also the case with proteins. In fact, Pauline Rudd has been one of the pioneers showing that, in fact, with ribonuclease, the attached oligosaccharide, an N-link sugar on asparagine 34, if I recall correctly, was in fact stabilizing the protein. So your point is very well made. Also the point about the different variations in RNA structures. You have single-stranded, you have double-stranded RNA, and in your cartoons, in fact, you show your substrate RNA as a partially single-stranded, partially double-stranded structure. Is that the real structure? And then also, of course, would it, in your view, make a big difference whether you would have a monosaccharide attached or a larger structure, disaccharide, trisaccharide, a monoantennary, a multi-antennary? Can you speculate? Because this is an evolving field, and of course, at this moment, there must be much speculation. And please speculate a little bit to make us even more excited about this.
Sure. Yeah, thanks for the question. So the structure, the structure in the cartoon is the known structure, secondary structure of a yRNA. And as cartooned, the glycans are positioned in places we see modification stops in our sequencing. And so I do think that will eventually be a reasonably accurate view of what a glycoRNA could look like. Questions that we're thinking about that we don't yet know are, do RNAs have one or two or many glycans at the same time? What's the valency of a glycoRNA? And what is the combinatorial, right, modification set? So in our mass spec, we see, mostly like larger oligosaccharides, bi, tri, and tetra and tenerary N-linked glycans, N-linked-like glycans. We don't see very many small oligosaccharides or glycans. We didn't find robust evidence yet for O glycans. So at least to date, we're thinking that these are larger carbohydrate modifications. But I think one of the things that we have to be careful about is that all of the data I just presented is looking at sialic acid, and you'll know that not every glycan has sialic acid. And so we have to think very carefully about how to develop new tools that are either less biased than MANAS, but still read out sialic acid, or more general, that can see other types of carbohydrate structures. We've used other metabolic reporters, which also give signal like GalNAS and GlycNAS, but those signals are not always RNase sensitive, which was going to be complex to describe in the first iteration of this work. And so there's a lot of work to be done, even with existing probes, and then also to develop new probes to think through other possibilities.
Thank you very much. Actually, there are a lot of questions here, so I hope you can really communicate with people over the email or something after the meeting, because we've really only got time for one more. And Chloe was asking if she could have a question.
Yes, thank you very much. It was very, very great talk. do you think we can speculate also on finding this type of glycoRNA in exosomes?
Yeah, it's a great question. I think I would bet yes. The question will be, is it inside the exosome or on the surface of an exosome? What's the topology? And then how good are you at purifying exosomes? I'm very bad at doing that. And so I haven't done that really. But I think that it's possible they'll be associated with them in some way.
Oh, that's great. Could I just ask one very quick question, because it was actually a question I had. It's from Sharif. How does the oligosaccharide transferase recognize the RNA? Do you have any?
It's a great question. I think to date, our data suggests that OST is important, but we don't yet have data to suggest that OST is the direct catalytic enzyme that operates on the RNA. There are multiple explanations for the way our data work to date. The catalytic pocket, the active site is actually in OST and STD3A and 3B. It's quite big. It has a fairly open face, and there are some positive residues. And I'm not a structural biologist, but it seems plausible to me that it could accommodate an RNA polymer. But we don't have any interaction data yet. We don't have any structural data yet for that. And so we're working with genetics and with co-IPs and things like that to try to understand what is the direct glycosyl transferase and how does that process work, and is it modifying native RNAs or modified RNAs which then get the glycan. There's a lot of possibilities, but I think it's plausible that STD3A or 3B could directly modify the RNA, but that is not something that we know yet with certainty.
Oh, thank you very much. That's a really stimulating end to the session. Thank you. Would you like to say anything?
Yes, just to let you know, we're taking a little break, so we'll be coming back at 5 p.m. Yeah, so 10 minutes, 13 minutes. Okay, thanks a million.
She doesn't hear.
Yeah, it's live now.
Hello, Mia.
Yes, I can hear you.
Okay, can you please upload your presentation?
I will.
So good afternoon and welcome to the session 4 of our meeting named Translational Glycobiology. This is a critical section because, of course, doing very good basic science, doing very good clinical science is one thing, but translating it into practical things, as we heard about the monoclonal antibody and the phage display system from Sir Greg Winter in the session 2 is another thing. So first of all, we welcome this afternoon Dr. Mia Huang. Dr. Mia Huang is a glyco-engineer working in the Department of Molecular Medicine at Scripps Research in Jupiter. She is joining in from Florida at a very early time, at least for her. The Jenner community is pleased to listen to your contribution, which is by now already uploaded, and we look forward to hear exciting things about your science.
Thank you very much for that introduction. And although it is quite early here, I've been here since 5 o'clock in the morning, joining the first parts of the symposium. The lineup has just been fantastic, and I've been awake the entire time. So thank you for the opportunity for me to present my work. I'd love to show you what we've been working on since we set up shop at Scripps about 3 years ago. So as has already been beautifully demonstrated earlier today by multiple investigators, the glycocalyx is replete with different kinds of protein glycoconjugates, such as mucins and other kinds of glycoproteins. The targets for our discussion today are proteoglycans, like syndicans and glypicans. These are protein glycoconjugates that are known to regulate biological events such as the spreading of cells on some sort of protein substrate, or the differentiation of embryonic stem cells into different lineages. We know well that these proteoglycans, as protein glycoconjugates, are composed of a macromolecular glycosaminoglycan, or a GAG chain, that's variously appended to a core protein shown here in black. What's even sort of underappreciated about proteoglycans, however, is their additional architecture, is that apart from glycosylation states and the number of glycosylation sites being present on the core protein, they can also exhibit differential regulation based on their localization on their cellular surfaces. And so, in fact, due to the structural complexity of these types of molecules, we often see these GAG molecules studied away from these protein components. However, we really do believe that it is of paramount importance to study these entities in the context of each other, as a polypeptide backbone of your proteoglycan can dictate how these glycosaminoglycans are oriented at the cell surface. And these glycosaminoglycans, in turn, as has also been beautifully shown by Thomas earlier, can also exhibit their own kinds of interactions. And so these proteoglycans are perhaps much more well known to serve as co-receptors for growth factors such as FGF2 and its corresponding receptor, FGFR, via these long glycosaminoglycan chains, where the formation of these ternary complexes between FGF2, these heparin sulfate GAG chains, for example, and FGFR are well known to activate various signaling transduction pathways and affect cellular phenotypes, such as differentiation of stem cells. Through the work of Linda Shea Wilson and many others, we now know that these HS or heparin sulfate GAG chains can encode specificity for binding different growth factors like FGF2 via these differential sulfation patterns, shown here as 2-sulfate, 6-sulfate, and this is analogous to what Thomas was mentioning earlier as a 4-O sulfate or chondroitin sulfate. And so, as you can see here, the GAGs on proteoglycans perhaps represent the most heterogeneous portions of proteoglycans. And these molecules have absolutely warranted our attention. But we do believe it is time to integrate our focus on GAG chains and GAG sulfation patterns with other parts of the proteoglycan glycoconjugates. And so despite 50 or so years of information that we've gathered around proteoglycan-mediated biology, there remain a number of outstanding questions regarding the structure-activity relationships of these molecules. For one, does the core protein itself really play a role in biology? There seems to be very little homology among different types of core proteins found in syndicans and glypicans and other types of proteoglycans. Is that really important? How do you distinguish between a glycan-mediated function of a proteoglycan and a core protein-mediated function? Is there really a requirement for chain valency? So you'll often find that these proteoglycans have anywhere between 1 to 5 of these GAG chains per protein. But people have also found that there are these half-time or part-time proteoglycans where some glycosides are left completely absent. What about these GAG sulfation patterns? We know that they're important, but how do we study them within the context of the entire proteoglycan? And also structural conformation. I think people have seen or believe that these molecules are largely intrinsically disordered. Is that really true? I think that providing large amounts of these proteoglycans with defined sulfation patterns and very homogeneous core proteins is going to be ideal in order for us to be able to address what the structural conformations of these molecules are in vitro. And furthermore, as I alluded to earlier, is membrane presentation required? So this is just a cartoon illustrating the effect of what happens when you have a soluble proteoglycan being chipped off of the cell surface. So proteoglycans often have a Shed site that's proximal to the membrane, and these are substrates for matrix metalloproteinases that can go on and cleave and generate these soluble ectodomains. Now you can easily imagine that because of these soluble molecules, they can actually compete for growth factors or other kinds of proteins like cytokines that are present now on that cell membrane. And so you can actually have a differential regulation. And so given all of these problems, how do you begin deciphering them one by one systematically? And we wanted, so our task here in my new lab was to be able to create a platform to be able to dissect these kinds of questions. Speaking of GAGs, however, we know that heparin sulfate found in cells is very different from what we all commonly use as a tool like heparin, and even modern synthetic techniques can only make oligosaccharides that are, about less than 50 residues. That's probably generous. Despite our limitations here, heparin being highly sulfated, much more than native heparin sulfate, we still like to use it as a tool because of its readily available. But we do have to be aware that heparin does not have the correct domain-like arrangement of the sulfation domains found in native heparin sulfate, and they are much smaller in size compared to heparin sulfate. And so in order to really study native glycosaminoglycans, how do we go about doing this, especially within the context of the entire proteoglycan? Chemistry techniques are not going to get you to the correct kind of GAG structure. It'll be hard to get you to the core protein. Chemistry will be hard to look at membrane presentation. And in fact, a combination of all of these enzymatic and genetic techniques, neither of these alone are going to suffice to be able to get to these outstanding questions in proteoglycan biology. And so our solution to this was to basically combine all of these methods into one. And so I'd like to present to you today a platform to be able to chemically edit proteoglycans. So what we basically do here is to take a core protein and use unnatural amino acid incorporation to tailor glycosylation sites. We use metabolic oligosaccharide engineering to be able to generate these recombinant sulfation-defined glycosaminoglycans that do indeed mimic the correct domain arrangement of native heparin sulfate, and use lipid engineering to be able to modulate how these molecules are presented on the cell surface. And so to start off, we use E. coli as our host cell to generate these unnatural amino acid modified core proteins. What we basically do is co-transform E. coli one with a plasmid that encodes for your proteoglycan ectodomain, and where sites of glycosylation are, we mutate that into a TAG amber codon. And so under normal circumstances, TAGs encode for a stop in a translation, but in the presence of an orthogonal tRNA and tRNA synthetase, and with an unnatural amino acid like parapargyl tyrosine, the TAG codon can then encode for these unnatural amino acids. So here we've modified the entire family of the syndicans with these PPY or parapargyl tyrosine amino acids at sites of glycosylation. You can even see here that we can add them in anywhere from 1 to about 3 sites. We haven't really tried further beyond the 3 sites of glycosylation per protein. And these subscripts here indicate the sites of the unnatural amino acid incorporation. We've also made them with these Apex2 fusions, and I'll show you what that's useful for later on in terms of proximity tagging. And we actually append the ectodomains with C-terminal polyhistidine tags as a way to purify them upon expression, but also to engineer these molecules at the cell surfaces, and I'll show you that example later. Here's just a gel showing you that we can make these proteins quite efficiently. If we use copper catalyzed click chemistry with a fluorophore, this basically, the presence of the fluorescence in these bands tells you that there are indeed these alkyne amino acids present in these proteins. So armed with a core protein in hand, we wanted to be able to engineer these recombinant glycosaminoglycans. And so glycosaminoglycan biosynthesis in cells happens by with translation of the core protein. It makes its way through the ER and the Golgi, and the first thing that happens to it is this modification with a xylose monosaccharide. And so Jeff Esco in the early 90s had actually shown that you can take xylose monosaccharides appended with benzene or any aglycone, and these molecules could serve as decoys or inhibitors of proteoglycan biosynthesis. So you would be left with a proteoglycan with an absent or shortened glycosaminoglycan chain. Our take on this is that maybe we could append a little azide motif at the aglycone side, and the cell would still produce these truncated proteoglycans, but in turn we would get functionalized recombinant azide modified heparin sulfate or chondroitin sulfate if you wanted to. And so in our modeling studies, we've actually shown that the azide group doesn't affect much the ability for beta-4 GALT7. This is the enzyme that's responsible for elongating that xylose for its ability to accept these kinds of molecules. And so what we then did is to take a panel of these recombinant xyloside molecules, threw them in a panel of sulfation mutant cell lines. This is provided by Jeffrey Esco, but there's actually a library of these different sulfation mutant cell lines now with the Clausen group and also the Wang group here in the US. And so incubation of these xyloside molecules with this panel of sulfation mutant cell lines allows us access to a library of these differentially sulfated heparin sulfate or chondroitin sulfate. I'm only showing you the HS here. Consistent with the nature of native heparin sulfate, we see that majority of these disaccharides are composed of this desulfated motif, in contrast to something like heparin, for example, which is primarily these trisulfated motifs. So armed with azide modified glycosaminoglycans and the alkyne modified core proteins, we can then use copper catalyzed click chemistry to append them together. And so I'll show you this data here. So if we look at anion exchange chromatography, you'd expect that the presence of these negatively charged sulfate groups would allow the core protein to then migrate towards higher retention times. So shown here in black is your core protein alone. If you add in one site of glycosylation, then you see the monovalent versions and the divalent versions and the trivalent versions increasing in retention time. And this is consistent also when we look at size occlusion chromatography, where the trivalent versions of the syndican ones migrate the fastest. So now that we have almost milligram access to these conjugates, we wanted to really solve whether these molecules were ordered or not. So people this kind of prediction using AlphaFold for what Syndecan1 looked like kind of made its way into Twitter. Obviously, this doesn't have the glycosaminoglycan chains, and you can see that this is largely disordered. And so we wanted to see whether the glycan chains actually affect the solution conformation of Syndecan1. And to do that, we use circular dichrosine spectroscopy. And so what you see here now is the Syndecan1 ectodomain that's deglycosylated in blue-green here is actually quite disordered. And even addition of the one chain of glycosaminoglycan also doesn't do much to help its order. In fact, it's still largely a disordered molecule. One thing that's also really interesting to have with these molecules is to start looking at binding assays. So here, particularly, we're asking whether the chain valency, the number of GAG chains per proteoglycan, is actually important in enhancing binding of different growth factors like FGF2. So I'm showing you binding data looking at the deglycosylated Syndecan1 having very little affinity towards FGF2, the monovalent Syndecan1 with some affinity, but it's really the divalent Syndecan1 with 2 GAG chains on it that actually has really strong affinity for FGF2. So the binding screens are really nice, but what we would like to do is to be able to study these molecules in live cells. So to do that, we can take our constructs, for example. You can have a library of these Syndecan1s with different kinds of glycosylation patterns. And we can then take any mammalian cell that we want, basically any cell with a phospholipid membrane, append that with a cholesterol with an NTA. And so by virtue of the histidine and nickel NTA complex, you can then get de novo display of your conjugates at the cell surface. And so this is really nice, especially for cells that really refuse to be transduced or transfected. And also, if you want to be able to do genetics on proteoglycans on cells, what you actually often find is that overexpression of proteoglycans tends to produce hypoglycosylated proteoglycans if you're not careful. And doing this by doing some engineering outside of the cell first, you can really control the glycosylation that these molecules have. So as a quick test, we were able to test our molecules on a simple mouse embryonic stem cell differentiation assay. Over the course of 1 week, we monitored the differentiation of mouse embryonic stem cells by adding in the cholesterol NTA first for an hour, and then subsequent incubation with the constructs that we want. And then after 6 days of differentiation, we monitor for markers of pluripotency like NANOG, or markers of differentiation like SOX1 or beta-3 tubulin. I'm showing you images here for cells that are devoid of heparan sulfate, so they won't differentiate on their own. So as you see here, after 6 days, these HS deficient stem cells still have really high levels of NANOG, a pluripotency marker. If you add in the cholesterol NTA alone, the cells remain pluripotent with high levels of green NANOG. If you add in the core protein alone, again, still really high levels of NANOG expression. In fact, it's only when you see, when you added in a heparin conjugate, or actually the GAG glycosylated versions of Syndecan1, that you lose NANOG expression, indicating that these cells are differentiating. And this is consistent with the affinity of FGF2, which is the main driver for mouse embryonic stem cell differentiation. The requirement for GAGs is consistent there. I mentioned the idea of using these Apex2 fusions of these Syndecan1s earlier. So what we've basically done is to also take the same mouse embryonic stem cells and display them with these Apex2 tagged Syndecan1 proteins. And then upon subsequent addition of biotin phenol and hydrogen peroxide, your Apex2 ascorbate peroxidase actually catalyzes the formation of highly reactive biotinyl radicals that can go on and biotinylate whatever is nearby this entire complex. So it's almost like an in situ proximity tagging. So in this microscopy image that I'm showing you, we've begun performing some of these experiments, looking at whatever is biotinylated after this protocol. So if you see purple, that's basically biotinylation happening, or we're staining the interactors of whatever proteins we have. In this particular image, we actually have membrane anchored Syndecan1, but it is deglycosylated. If you have a heparin conjugated Syndecan1, then you see some qualitative differences. And also with now in this case, if you don't add cholesterol, but you only have a soluble version of the syndecan-1, then you actually also get some changes by microscopy. And so we're performing proteomics right now to be able to decipher these kinds of changes, perhaps to be able to elucidate what's changing in the interactomes when your syndecan-1 is a soluble protein versus a membrane bound, when it's glycosylated or when it's not. In another application of these molecules, we were really curious to see whether we could prove the dogma in that syndecan-1 mediated cell spreading relies on the ectodomain. So in contrast to a GAG mediated function earlier, now we're looking at a potentially a core protein mediated event. And so using our molecules, we also surveyed the binding of alpha V beta 3 integrins to these core proteins. We did see some binding, but really we required the glycosylated versions. And if we saw an enhancement in binding of alpha V beta 3 integrins to these molecules. But this is obviously a heparin, so it's very, very sulfated. And when we added in our HS modified syndecan-1, we see affinity somewhere in the middle here. So this is an interesting observation on our part that GAG chains actually enhance the affinity of syndecan-1 to alpha V beta 3. And so using our same cell surface engineering platform, we wanted to see if this was phenotypically true as well, to look at cell spreading. This is what happens to wild type breast cancer cells when you add them onto vitronectin and stain for phalloidin, a cytoskeletal marker. They do spread on a substrate like this. If you knock out syndecan-1, the cells refuse to spread because you require syndecan-1 for spreading. As expected, when you added in deglycosylated syndecan-1, you don't see any cell spreading. However, even when we added in glycosylated syndecan-1, we really don't see any cell spreading when these molecules are added in the soluble manner. In fact, it's only when we use glycosylated and membrane anchored syndecan-1 that we begin to rescue this kinds of effects. So we were really encouraged by the use of this integrated platform to be able to decipher a lot of these proteoglycan mediated functions. And so just to summarize, I hope I've shown you that these proteoglycans are really an interesting multivariate puzzle that deserves to be studied all together and all at once. We can use unnatural amino acids to modify proteoglycan core proteins, use metabolic engineering to generate these recombinant GAGs, and then use copper click chemistry to be able to stitch them together. And that we can use the polyhistidine tags, not just for purification, but also for engineering at the cell surface. And that this platform is really useful if you're really interested in deciphering the little intricate details of proteoglycan architecture simultaneously, but also in a really defined manner. So with that, I'd like to thank you for your attention and like to thank the funding agencies and my group for this work.
Thank you. Thank you very much, Dr. Juan. This is in fact an excellent platform for synthesis by what I would call a combination of chemical and biochemical methods. Because we are in a session about translational glycomedicine, I would first like to ask you how do you see the future of this, let's say, editing of proteoglycan architecture? Let me rephrase it in a different way. An architect may be interested in bricks, building blocks, but the ultimate goal is to make a building. So what do you envisage as the disciplines where the application of your glycoengineering technology will break through? Will it be in oncology, neurobiology, endocrinology? How you see the future of this glycoengineering technology evolving?
Thank you for letting me speculate on that. So one of the things that we're really working on is in the area of neurobiology. And this recent finding that this important synaptic molecule called neurexin was found to be a proteoglycan. That finding, that discovery just blew my mind. They actually found that neurexin has a single glycosaminoglycan site on it. And so the interactomes on why the GAG chains are there are still relatively unknown, we've been really satisfied by knockdowns and knockouts without really paying attention to this gag chain. And so I think that the orexin is a really important regulator for psychiatric and neuropsychiatric disorders. And so I'm really interested in elucidating the role of the gag chain, gag sulfation, the type of gag also that's appended to orexin as a way of modulating the biology of this proteoglycan and being able to pave a pathway for some sort of drug discovery in that area.
Thank you. I'm just looking around whether there are questions here in the audience. I have another one, in fact, but I'm looking first around. So the other question is, and this is just to put the nomenclature right. People speak very often about the enzymes proteoglycanases, and many novices in the field think that these are glycanases, but in fact these are not glycanases, these are proteases. And rightfully you mentioned indeed the matrix metalloproteases. So proteoglycanases in fact are stromolysins, are molecules that cleave the protein backbone and have been studied quite a bit in cancer research as stromolysins. And also in rheumatology, for instance, in particular matrix metalloprotease 3. With your platform, are you also planning to make glycoengineered mutants that are, let's say, resistant to the cleavage by, let's say, stromolysin 1 or matrix metalloprotease 3? Is that also one of the things?
Yeah, we are actually. I feel like my moves are very predictable at this point, but yes, we're actually finding something really interesting in terms of the recognition capabilities of MMPs and the presence or absence of these gag chains too. So we're not quite ready to report that yet, but definitely very interesting area as well, especially for glypicans, where the gag chains are quite near the Shede site.
Okay, looking around still. If there are no more questions, we thank you very much. We clap hands and keep up the good work.
Thank you.
As a second convener in this session about translational glycobiology, it is my privilege to introduce briefly to you Dr. Thomas Madsen. He is joining us as a member from the Copenhagen Center for Glycomics and the University of Copenhagen. I think that O-linked glycosylation is at his heart. So we are pleased to hear all the news, and in particular the translational medicine news about the glycosylated neuropeptide. Please, go ahead.
Thank you very much. You hear me loud and clear? You see the slides?
Yes, we can.
Perfect. Yes, so I'm Dr. Thomas Daubjerg Madsen now from the Copenhagen Glycomics Center. I'm very honored to be invited to speak here today. I'm probably one of the youngest speakers in the symposium today. So I want to talk a little bit about our work on glycosylated neuropeptide hormones today. And just to go from square one, just see here, need to click on the screen, I think. There we go. So neuropeptides and peptide hormones are signaling peptides that normally maintain homeostasis. The prime example is insulin that's secreted from the pancreas to the blood when you eat a candy bar in response to the heightened blood sugar, and returns the blood sugar to homeostasis by telling another organ that it needs to take up the excess sugar. Now, there are more than 200 examples of such signaling peptides. For instance, could be calcitonin that regulates blood calcium, or secretin that regulates intestinal pH, even galanin that regulates your memory and pain perception. Now, galanin is a classical neuropeptide in the sense that it works in the brain between neurons, where the others work in the bloodstream between organs. But most neuropeptides also work in the bloodstream as peptide hormones and vice versa. So I'm going to use the term neuropeptide throughout the presentation. Because these peptides are small and have very specific targets physiologically, they're very interesting as therapeutics. And you may all know that insulin is the best weapon against diabetes we have these days. Other examples include somatostatin that's used to treat improper growth. And there's a huge potential of using neuropeptides in treatment of CNS conditions such as addiction. However, there's a bunch of challenges associated with using the neuropeptides as therapeutics. First of all, they have extremely short half-lives, less than 10 minutes. So no matter how much you will put in bolus in a patient, it will be gone within an hour. Furthermore, many of the neuropeptides in nature have distinct but multiple functions, pleiotropic functions. That's not always what we want when we want to treat a specific disease with a certain pathophysiology. And lastly, we cannot really access the CNS with these peptides because they don't cross the blood-brain barrier. So this large potential they have, we cannot utilize as research is now, which is a shame. So the way current therapeutic strategies go around these challenges is by chemically modifying the peptides. And it has been used to a great success, but we do have some issues with immunity, immunological response against the neuropeptide analogs. I think it's 10% of the patients treated with octreotide against gigantism that raise a response towards the treatment. So wouldn't it be nice if nature itself came up with these modifications that solve the challenges? So here we've just summarized the different PTMs that are found on neuropeptides, with the most prominent being the C-terminal amidation, where half of known neuropeptides contain this PTM. In many cases, this is crucial for the bioactivity, although not in all. What we're going to talk about today is the O-GalNAc type glycosylation that has been pretty underrepresented in the PTM field of neuropeptides until we came in and found that 1/3 of neuropeptides carry this modification, making it pretty widespread among neuropeptides. So before we go into the data, I just quickly want to go over the biosynthesis of the neuropeptides that are produced in the ER as proneuropeptides that are inactive. In the Golgi, they get glycosylated, and specifically the O-GalNAc type glycosylation is initiated by 20 different GalNAc transferases that puts on the first glycan. And from there, you can get a wide variety of different structures, making this ultra diverse and complex. Now, our approach was to just target the CoR1 and silyl CoR1 structures because they're the most abundant in the circulation. But by no means are the glycans and neuropeptides restricted to these. We have no proof of that. After they get decorated with the glycans, they continue in the secretory pathway into the dense core granules, where they get separated from the proneuropeptide and activated. And then they await a stimulus for secretion. That could be the heightened blood sugar in the case of insulin. So the way we went about identifying glycans in these neuropeptides was using glycoproteomics. We have a pretty well-established LWAG strategy at the CCG where we enrich for glycosylated peptides, and we improved on that by choosing neuropeptide-rich tissues as starting material, as well as enriching for the low molecular weight peptide fraction before digestion and revealing of the CoR1 epitopes that we would enrich with lectin-weak affinity chromatography. By doing these 4 steps, we reduce the complexity enormously, only having small peptide fragments from smaller proteins present in the sample and only the glycosylated variants of them. So introducing that into the LCMS, we can get a really deep glycoproteome of the low molecular weight fraction, which turned out to be a really good deal when we're looking for glycosylated neuropeptides. We found around 400 glycosides on proneuropeptides, of which half of them belong to 91 mature neuropeptides. And if we divide them out on the respective neuropeptide families, that is, one family is a bunch of neuropeptides with similar sequence, so that 1/3 was glycosylated, using the NetOGlyc algorithm to predict glycosylation based on peptide sequence, we could see another third is predicted to carry glycosylation. And the last we cannot really say anything about at this stage. So with these data, we go on and claim that neuropeptides actually group a class of O-GalNAc type glycosylated biomolecules. So what do these glycans do? And are they in any way interesting therapeutically? Like, how do we go on from here with 400 different sites? Our approach was to look at the glycans and categorize them based on where they're positioned on the proneuropeptide. And what immediately caught our eye is that many of the glycans were positioned in what is known as the message region. That is the region of the peptide that fits into the receptor. So the glycan would sit here, right at the border of the receptor interacting region, illustrated by the blue circle here. And many well-known neuropeptides had this phenotype in their glycosylation pattern. So we chose 3 families, the glucagons and neuropeptide-wise and the natriuretic peptides, to continue and investigate what does this type of glycosylation do. What was very special in these 3 cases is that the glycans we would find seem to be conserved in the same position, despite that the neuropeptides would have widely different functions. So for the glucagon family, we found half the member carrying a glycan on threonine 7, and the other half of the members we didn't identify, but a threonine and a serine was conserved at this very position, telling us that there's a conservational pressure on keeping this site available for glycosylation, despite the different functions of the peptides. The similar case we saw for the neuropeptide-wise threonine 32 and the natriuretic peptides inside a disulfide ring of this class of molecules. So we went ahead and tested the glycosylation of these peptides in traditional receptor activation assays, where we would express receptors in a cell line and then treat the cell line with increasing amount of the neuropeptide with or without the glycan. So we have an example of ANP here that regulates blood pressure. In the black line, we have the non-glycosylated peptide that will activate its receptor around 1 nanomolar. When we put on the glycans, we need 10 to 100-fold more neuropeptide to achieve the same activation effect, telling us that the glyconeuropeptides are less potent than the non-glycosylated. This turned out to be the case in all neuropeptides tested, both from the glucagon family and the neuropeptide Y family. But we know that glycosylation normally infers stability. So another thing we really wanted to investigate is how long is the circulation time of the glycosylated neuropeptides compared to the non-glycosylated. So to investigate this, we did a traditional half-life measurement study in rats where we injected the neuropeptides with or without glycans and followed its degradation over time. And here we see GLP-1 non-glycosylated. There's a half-life of 2.5 minutes. And as expected, it's gone within the hour, very short half-life. Now, when we put on the core-1 glycan, the disaccharide, we double its half-life. So it's more stable, stays in the circulation for a longer time. But most strikingly is when we put on the negatively charged sialic acid on top. This increased the half-life almost 10-fold and was still readily available after 60 minutes post injection. So now we know the neuropeptides are less potent but more stable, suggesting that they will have a longer but less pronounced effect when injected into to animals. So to pursue that, we set up an animal setup with rats where we used ANP as a model molecule, either glycosylated in one or another position with a sialylated core-1 glycan. So ANP, as I said before, regulates blood pressure and also induces natriuresis. So it basically makes you fill your bladder. So the way we went about it was to inject the rats with a peptide with a constant infusion over a 60-minute period, after which we continue with a 30-minute clearance period. And if we look at the data, you can see that the non-glycosylated, as expected, lowers the blood pressure rapidly and very pronounced within the infusion period. And when we begin the clearance period, it rapidly returns towards normal blood pressure again. Whereas the glycosylated compounds had a less pronounced response. But after the clearance, or in the clearance period, it did not go back to normal at the same pace as the non-glycosylated, and one of the compounds even lowered the blood pressure even further, confirming our hypothesis that these glycosylated neuropeptides are longer acting but less pronounced versions of the non-glycosylated. Now, what was really interesting is that when we went on to look at the urine volume produced, or the natriuresis, we see that the non-glycosylated variant really induced a lot of urine production, whereas the glycosylated variants had nowhere near as potent an effect on the natriuresis. So this is really interesting and kind of tells us that the glycans could have an important role in directing which of the pleiotropic functions a neuropeptide would have. So ANP is currently used in Japan for treatment of acute heart failure, of hypertension basically. And one of the main side effects is hypotension. Again, I think it's 10% of the patients who experience this. It's basically an overtreatment because the blood pressure goes too low for what you want. And now we have a version that's less pronounced but longer acting, which could be easier to titrate in for the patient. Furthermore, it doesn't fill the patient's bladder. So in the case of overtreatment and the blood pressure goes so low that the patient faints, we wouldn't have filled his bladder. And that's nice if you don't want to wet yourself. Now, this is just one example of the many glycosylated neuropeptides we can look at. And it shows really promising results for the 90 other neuropeptides that we might be able to produce a new generation of therapeutics based on natural designs with the glycosylated panel that we have identified in these studies. So in conclusion, we have established a new class of glycoconjugates known as the glyconeuropeptides. And when we put the glycan in the receptor interacting region, it has improved stability, but it's less potent at receptor activation. Now, this is just one of the many categories of glycans we can look at. And we hope to get into human studies soon to prove that these glycosylated variants actually are superior therapeutics compared to the non-glycosylated. Furthermore, I think all the other glycan categories could regulate biology in terms of how they're packaged inside the cell during their biosynthesis, how they're matured with prohormone cleavage in the dense core granules. And the body of work I'm really following closely these days is the work of Dr. Robin Polt that has 20 years of research claiming that glycosylation of peptide hormones improve blood-brain barrier crossover. Now, he has done chemically synthesized glycans on the neuropeptides, but now we have glycans and neuropeptides designed by nature itself. So this could actually be a way for the body to deliver neuropeptides from the bloodstream to the brain. Although this is all future studies, future perspective, so got to keep an eye out, huh? With that, I would like to thank you for your attention, and especially I would like to thank the Copenhagen Center for Glycomics team, where Lasse Holst Hansen has done a great deal of the work, really fantastic. And I would also like to thank the Department of Biomedical Sciences at the University of Copenhagen for helping us out with all the physiological studies.
Thank you very much, Dr. Thomas Madsen. This is indeed an excellent illustration of translational glycobiology. In fact, many questions pop up. Let me first say that I liked very much the approach going from an unbiased, very broad spectrum analysis to particular examples, and you tackled the example of atrial natriuretic peptide. Do you have any insights into one of the other favorites that you named, namely neuropeptide Y? or is that ongoing or planned?
So it
Because
is ongoing.
neuropeptides, of course, make us immediately think of brain activity and blood-brain barrier crossing. Can you comment on that?
So this is the issues that we are tackling currently, and we don't really have any data as of yet. I can only refer to Robin Paul's work that is really thorough, but neuropeptide-wise, one of our main targets, and we have the glycosylated variants lying around just waiting to be tested in exactly this setup.
Yeah. One of the nice things here that you developed, in fact, and that is if I compare it to technologies that have over and over been used for looking to the functions of N-linked sugars, there people usually relied on in vitro mutagenesis, where they mutated out the asparagine to get rid of the the N-linked sugar, but they replaced it by an alanine. I think the nice thing about your system is that you have the naked aglycosyl peptide compared to the glycosylated one without any mutation. And I think this is really the way to go. And I had expected in the past that this would first happen with N-linked sugars, because O-linked sugars were too complex with all these repetitive serines and threonines very often occurring, where people did not have the foggiest idea about site occupancy. So I think this is already one of the wonderful new developments of your work. And now I'm in fact looking back if there is any question. Professor Axel?
Hi Thomas, nice talk. John Axford here. If you are going to use this as an antihypertensive in patients, how is it given to a human? And is there potential for design and modification depending on how high your blood pressure is? Can you change the glycosylation to affect potency?
So it's actually given to the patients in the clinic the same way as we delivered it to the rats. So it's infused over a longer period of time, most likely because of the short half-life. You cannot really, it doesn't help to give one big bolus at the start. You'll just kill the patient within 10 minutes. So that's how it's given. We would have to look further into the right glycovariant to give. Like for starters, the sialic acid variant has the longer half-life and is probably the primary form in the circulation. So if you want to go for the purely nature design, I would definitely start out with that one. But
so it's probably going to
I mean, it's open.
So it's probably going to be used as an emergency medication, isn't it? I would think for people with blood pressure that can't be controlled in pregnancy, for example.
Yes.
No, I'm okay. I think Pauline had a question.
Yeah, it's Pauline Rudd. I was just wondering if you had any ideas of the mechanism by which the sugar stabilizes the peptide, because we had a lot of interest in that at one point. We were looking at ribonuclease B, and we did some NMR studies which showed that it was residues all over the protein that were affected by the addition of the sugar. And we also looked at the breathing rate of the protein. So, you know, how when the breathing rate slows down, the interaction rate is slower. But I mean, you can't really do that on a peptide. So I'm just wondering, you know, how you would try to understand the mechanism.
So we did some few in vitro studies with purified peptides. And just to give you a brief summary of the results, we see, of course, the closer to the enzymatic site of cleavage, the bigger the protection. But what is interesting is that the sialic acid, you can put the glycan with the sialic acid in the other end of the peptide compared to the cleavage site, and it will still protect. So the core one won't really protect, but putting on the sialic acid suddenly gives you an effect. So I think since they are so small and the enzymes are so big, you probably just have steric hindrance, electrostatic repulsion of some sort. That would be my guess. There are also a certain class of cryptases that degrade neuropeptides that have to fit in the whole neuropeptide in the binding pocket before it leaves. And putting on a glycan dramatically increases the size of the peptide. You know, that's small, right? So a glycan is enormous compared to it. So I basically think it's steric hindrance for the enzyme recognition. There's a whole discussion on clearance or renal filtration, where I believe that the sialic acid would prevent renal filtration, although I do not at this point have any proof of that. But that could explain why we suddenly get a 10-fold increase in half-life in our circulation study.
That's interesting. Would you think it was worthwhile doing some NMR experiments to look for, or proton exchange experiments to just look for the accessibility of the surface of the protein? Or?
So since it's basically just an alpha helix, there's not much to expose, I guess. Or you would do it in together with the enzyme?
Yeah.
I guess it would be interesting to see. We did a few CD spectroscopy experiments where we could see that the alpha helix of the peptide itself unfolds when we put on the glycan. And this, like the whole biodistribution project led by Dr. Robin Paul, claims that this unfolding of the alpha helix can modify its ability to interact with membranes. And it's a whole field I'm digesting these days.
Okay, thank you, Copenhagen.
Thank you so much for the nice words and inviting me.
Okay, so after this great presentation we have, and actually after this great day of glycoscience and medicine, we have this last presentation. We have from Buenos Aires Professor Gabriel Rabinovich. So Professor Rabinovich is director of the Laboratory of Glycomedicine at the Institute of Biology and Experimental Medicine, senior investigator of the Argentina National Research Council, and professor of immunology at the Faculty of Sciences, University of Buenos Aires. So among many memberships, Professor Gabriel Rabinovich is member of the U.S. National Academy of Sciences, and his work has been focusing on understanding the function of glycans and glycan-binding proteins in cellular processes relevant to immune regulation in health and disease. And today, Professor Gabriel will present galectins, soluble glyco checkpoints at the crossroads of inflammation and cancer.
Hello, good afternoon. Can you hear me well?
Yes, yes, we can.
Good afternoon from Argentina. I'm so happy to participate in these general meetings. It's a huge honor to participate, and I am very happy having spoken at Paris and also in Dubrovnik. I feel part of the Jenner family. So what I will speak today, I summarize early discoveries and also recent findings from our lab that support the translational applications of our discoveries to clinical therapy, particularly because we are in the translational glycoscience session. So the topic I will speak today will be about galectins and glycans as soluble glycocheckpoints at the crossroad of inflammation and cancer. And I try to summarize here in my first slide my talk that we have a taste of Argentina. It will be sweet, like dulce de leche, and it will involve two pairs, galectins and glycans, like the two pairs required to tango. So in the past 2 decades, we have witnessed a revolution in cancer immunotherapy. Although William Coley was the first that proposed that an immune system is important to kill and eliminate tumor cells in 1893, it took almost 120 years until these discoveries and this hypothesis became true for the benefit of cancer patients. And it was particularly related to the identification and discovery of immune checkpoints, which blockade increase overall survival and progression-free survival in a large number of patients with different tumors. But what are exactly immune checkpoints? Immune checkpoints are inhibitory pathways that limit the duration and amplitude of immune responses. They can act early during the interaction of antigen-presenting cells with T cells in lymph nodes. Here you can see dendritic cells and T cells, and they are mainly illustrated by CTLA-4 and PD-1. And they are also evident in the interactions between T cells and tumors in the tumor microenvironment, as you can see here in PD-1. But PD-1 and CTLA-4 are not the only immune checkpoints. Recently, it became important, the studies of LAG3, TIM3, BTLA, TIGIT, and VISTA. And what is important is that immune checkpoints can operate in the whole tumor microenvironment, modulating the function not only of T cells, but also dendritic cells, natural killer cells, macrophages, myeloid-derived suppressor cells, and even endothelial cells and fibroblasts in the tumor microenvironment. There are 4 hallmarks of immune checkpoints in general that characterize an immune checkpoint. They are often co-opted by tumors as a mechanism of T-cell resistance. They promote resolution of autoimmune and chronic inflammation. They contribute to immune tolerance at the fetal-maternal interface, and they control resolution of microbial infections. But we will focus today on glycocheckpoints, what we call glycocheckpoints. So glycans, as you know, have been widely studied as glycobiomarkers in the tumor microenvironment. But recent evidence suggests that they can also function as glycocheckpoints that suppress tumor immunity by attenuating and resolving inflammation in the tumor microenvironment. To illustrate this concept, sialic acid present in tumor cells can interact with Siglecs on the surface, present on the surface of innate immune cells, including dendritic cells, natural killer cells, monocytes, and macrophages, and suppress activation of these cells. Another example is the GalNAc antigens or the TN antigens that can interact with the macrophage galactose lectin with MGL to suppress activation of innate immunity, and fucosylated antigens that can interact with DC sign also to regulate activation, proliferation, and differentiation of macrophages and dendritic cells. So all these protein-glycan interactions can suppress immune responses in the tumor microenvironment. Particularly, we will focus today on galectins. Galectins are, in contrast to Siglecs or C-type lectins that are cell surface associated glycan binding proteins, galectins are secreted from tumor cells, from stromal cells, and from different cells in the tumor microenvironment, and they can interact with LAC-NAC residues, with galactose, beta-1,4, and acetylglucosamine. So what are exactly galectins? Galectins are a family of soluble glycan binding proteins that preferentially recognize N-acetyllactosamine, galactose beta-1,4, galactose beta-1,4 and acetylglucosamine units present in complex and glycans and also in core 2O glycans on different cell surface receptors. They are classified into 3 different structures: prototype galactins, chimera type galactins, and tandem repeat galactins. We will focus today on prototype galactins, particularly on galectin-1. In response to stress, galectins are secreted through an unconventional Golgi independent pathway, and they can form different type of lattices when they act extracellularly. Like here, they can breach different glycosylated receptors. They can form multivalent interactions with these glycosylated receptors and control receptor segregation, endocytosis, and signaling, leading to modulation of immune cell activation, differentiation, and survival. Recently, we proposed that galectins may act as regulatory checkpoints that link tumor immunity and angiogenesis. So at the early 90s, we identified galectin-1 as a 14-kDa beta-galactoside binding protein that was upregulated in antigen-presenting cells and also in cancer cells, exactly as immune checkpoints. One interesting issue is that expression of galectin-1 in most human tumors correlate with the adverse prognosis and acquisition of metastatic phenotype. So, given these findings and the inhibition of immune responses induced by galectin-1, the anti-inflammatory effects and the immunosuppressive effects of galectin-1, we wonder whether galectin-1 may contribute to tumor evasion of immune responses, to tumor immune escape. And in fact, in this paper we published in 2004, we demonstrated that melanoma cells secrete high amounts of galectin-1 in its dimeric form in high concentrations that binds to CD45 on activated T cells and is able to kill activated T cells. So galectin-1 contributed to the immunosuppressive activity of melanoma cells. Blockade of galectin-1 in the tumor microenvironment resulted in tumor rejection that was particularly mediated by CD4 positive and CD8 positive T cell mediated responses that were mediated by interferon gamma, so particularly Th1 responses. So when we blocked galectin-1 expression, we found a reduction of tumor growth, and it was prevented when we eliminated either CD4 or CD8 positive T cells. We confirmed these findings in Hodgkin lymphoma. In this particular tumor type, we found that Steinberg cells in Hodgkin lymphoma express very high amounts of galectin-1 that contributes to this non-productive inflammatory Th2 type infiltrate. So we found here that expression of galectin-1 by Hodgkin lymphoma cells was regulated by an AP1 dependent enhancer that was regulated by Epstein-Barr virus, that secretion of galectin-1 creates immunosuppressive microenvironments in Hodgkin lymphoma, and that circulating galectin-1 levels are very good biomarkers of prognosis of Hodgkin lymphoma. So from the time we started and we demonstrated these effects in melanoma, many different labs and also our labs demonstrated that these effects that galectin-1 can activate immune evasive programs in several tumor types can be extrapolated to different tumors, and we were very happy that this effect was demonstrated in different tumor types. But how galectin-1 can suppress the immune response? So let's go back to glycobiology. And basically, galectin-1, as you know, interacts with LAC-NAC residues with galactose, beta-1,4, and acetylglucosamine, both in complex, branched, and glycans, but also in core 2O glycans. So, as you know, there are two glycosyl transferases that are critical for galectin-1 binding. So one is the MGAT5, or that the GNT5 encoded by the MGAT5 gene that contributes to beta-1,6 and glycan branching, and another one is the C2GNT1 or CGCNT1 that contributes to core 2O glycans and elongation of LAC-NAC ligands. But there is one glycosyl transferase, ST6-GAL1, that incorporates alpha-2,6 sialic acid, and galectin-1 cannot bind to receptors that are rich in alpha-2,6 sialic acid. So Marta Toscano from my lab, one PhD student, she demonstrated that T cells, particularly Th1, CD8 positive T cells, and Th17 cells, are covered by high amounts of covered by all the repertoire of glycans that are important for galectin-1 binding. So core-2 glycans and complex and glycan branchings, and low levels of alpha-2,6 sialic acid. In contrast, Th2 and regulatory T cells are covered by alpha-2,6 sialic acid, so galectin-1 cannot bind and cannot kill these cells. So we found that because of this glycosylation signature, galectin-1 bound selectively to Th1, CD8 positive, and Th17 cells, inducing dysfunction of these cells. And this could explain why, when we block galectin-1 expression, there is an increased frequency of interferon gamma positive cells. So then we demonstrated, this is the work of Tomás Dalotto Moreno and Mariano Salatino, they demonstrated what's the role of galectin-1 on regulatory T cells. And what they found was that in a model of triple-negative breast cancer, galectin-1 contributed to promote expansion of CD4 positive, FOXP3 positive regulatory T cells in the tumor microenvironment that contributed to systemic immunosuppression and to lung metastasis of triple-negative breast cancer tumors. So the question was whether galectin-1 may also affect antigen-presenting cells, because it was highly expressed in tumors and dendritic cells, particularly antigen-presenting cells, but particularly dendritic cells, are very close to the tumor when they take up the tumor antigens. So we studied the role of galectin-1 glycan interactions in the case of dendritic cells, and what we found was that galectin-1, when it bound to dendritic cells, it converted dendritic cells into tolerogenic dendritic cells by binding to all glycans on CD43. So this induces phosphorylation of the STAT3 transcription factors, secretion of IL-27, and also secretion of IL-10. So Juan Martín Larregui, a PhD student also from our lab, he demonstrated that GAL1 promotes tolerogenic dendritic cells by IL-27 and IL-10. We published this in 2009, and most recently we found that this effect was regulated by SATB1 and Notch in the tumor microenvironment, particularly in dendritic cells. So one of the most striking findings is that when we block galectin-1 expression, we found an enhanced anti-tumor response, an enhanced inflammatory infiltrate, but also a decrease in blood vessels. And we asked whether galectin-1 may act by promoting not only immunosuppression, but also angiogenesis. And this becomes particularly interesting because there is a crosstalk between immunosuppressive and pro-angiogenic programs in the tumor microenvironment. So, for example, BGF can induce immunosuppression in addition to its pro-angiogenic effect. So we wonder whether galectin-1 may induce vascularization of tumors. So to make a long story short, what Diego Crossi, a PhD student also from my lab, who is now associate researcher, he demonstrated that galectin-1 is released from tumors in response to hypoxia, in response to low oxygen tension, and it's overexpressed by tumors, it's released by tumors through a mechanism that is completely independent of HIF-1 alpha, which is a master transcription factor of hypoxia and angiogenesis, but depends on NF-kappa B, on the expression of NF-kappa B. So then when galectin-1 is released, surprisingly, when we perform gene arrays, RTK signaling arrays, we found that galectin-1 interacts with BGF receptor 2, so interacts preferentially with the same receptor as BGF, as vascular endothelial growth factor. Particularly, it interacts with complex branch and glycans on BGF receptor 2 and mimics BGF-like signaling. So we wonder whether galectin-1 may compensate for the absence of vascular endothelial growth factor, for the absence of BGF in anti-BGF refractory tumors. So those tumors that do not respond to anti-BGF treatment, whether galectin-1 may mimic and may induce angiogenesis by interacting with BGF receptor 2. So as a summary of these findings that were published in these two papers, so what we found was that in those tumors that are sensitive to anti-VEGF treatment, endothelial cells that are surrounding the tumor are covered by alpha-2-6 linked sialic acid, so galectin-1 cannot interact and cannot promote angiogenesis. In contrast, in tumors that are resistant to anti-VEGF treatment, endothelial cells are covered by beta-1,6 and glycan branching, has low levels of alpha-2-6 sialic acid, so galectin-1 can interact with immunoglobulin domain 3, 4, and 7 of VEGF receptor 2, promoting VEGF receptor 2 phosphorylation, dimerization, and also revascularization and angiogenesis, even in the absence of VEGF. So the question was whether this mechanism may be a mechanism of resistance in anti-VEGF treated patients. And we recently found that this is also true in a cohort of patients that were treated with bevacizumab anti-VEGF or Avastin. So we wish to integrate these findings in a whole model, whether galectin-1 can induce immunosuppression, promote metastasis, and also angiogenesis in a single model. So we did these experiments with Pilar Navarro from Hospital del Mar, Barcelona, and we selected pancreatic adenocarcinoma. Why pancreatic adenocarcinoma? As you know, these type of cancers have the lowest survival rates, the poorest prognosis, and they are resistant, completely resistant to immunotherapy. So we performed two different models. First, we cross mice, a mouse model in which we cross mice that are KRAS and P53 mutant with galectin-1 deficient mice. These transgenic mice, KRAS P53, are sensitive, they develop pancreatic adenocarcinoma, so we cross these mice with our galectin-1 knockout mice to generate this transgenic model. And also we co-culture different human pancreatic stellate cells with tumor cells or with immune cells. In this particular model, galectin-1 is particularly overexpressed in these fibroblasts, in human pancreatic stellate cells. So we co-cultured human pancreatic stellate cells derived from tumors, either with tumors or with immune cells. And what we found, combining these mouse models and these human models, we found that inhibition of galectin-1 results in suppression of metastasis, suppression of angiogenesis, and an increased frequency of CD4 positive and CD8 positive T cells. Particularly, we found a significant reduction of myeloid-derived suppressor cells, which are an enigmatic type of cell in the tumor microenvironment, by a reduction of CD11b positive, GR1 positive cells. So because this striking effect of myeloid-derived suppressor cells are a particular type of cells that are highly expressed and highly present in the tumor microenvironment, and they have both immunosuppressive and pro-angiogenic effects. So we wondered whether galectin-1 can modulate the immunosuppressive and pro-angiogenic effect of this cell type, myeloid-derived suppressor cells. First, we dissected the glycosylation signature of myeloid-derived suppressor cells, both monocytic myeloid-derived suppressor cells and neutrophilic myeloid-derived suppressor cells. We found differences in the glycosylation signature, but the most striking difference was when we compared tumor-associated myeloid-derived suppressor cells with other type of myeloid-derived suppressor cells. But they all showed all the repertoire of glycans that are important for galectin-1 binding. Galectin-1, we found that galectin-1 bound to myeloid-derived suppressor cells and synchronized both the pro-angiogenic effects of myeloid-derived suppressor cells, secretion of VEGF and angiogenesis, and also the immunosuppressive effects of myeloid-derived suppressor cells. How galectin-1 can do that? It can bind to, we found by proteomic analysis, that it can bind to glycans on CD and a complex of CD18, CD11B, and CD177, triggering phosphorylation of tau3 and triggering gene expression that is mediated by immunosuppressive genes and also pro-angiogenic genes that contributed to tumor progression. We just submitted this paper for publication. But we wanted also to understand the role of galectin-1 in a tumor that could be partially resistant to immunotherapy, like colorectal cancer. And so we recently published a paper. This is a paper published by Alejandro Cagnoni. The previous study has been done mainly by Ada Blidner and also by Camilla Bach. And in this particular study in colorectal cancer, we found that colorectal cancer cells, independently of the mutations, they produce very high amounts of galectin-1. And in this particular tumor type, galectin-1 cannot induce apoptosis of T cells, does not induce tolerogenic dendritic cells, but we found an expansion of a particular type of CD8 positive regulatory T cells that express CD11, CD122, PD1, and IL-10. So these regulatory T cells promote immunosuppression of tumor progression. Here you can see the lower number of tumors generated in the AOM-DSS colitis-associated colorectal cancer. So the 1 million question that you may be asking now is, how can we use galectin-1 as a therapeutic target in cancer? How can we design inhibitory antibodies or inhibitory agents? So the advantage of blocking galectin-1 is that when we block galectin-1, we can unleash anti-tumor immune responses and also prevent tumor angiogenesis. So first we started with glycoamines that block not only galectin-1, but also other members of the galectin family. Also, we tested small molecule inhibitors. The same thing happened. It bound not only to galectin-1, but also to other members of the galectin family. So then we decided to generate anti-galectin-1 neutralizing monoclonal antibodies. We have now 2 different antibodies that recognize different epitopes, one human anti-galectin-1 antibody, completely human, and one single chain. We published one of them, the best antibody that we published this year in angiogenesis. And what was particularly interesting is that this antibody has high affinity, high specificity. It does not bind to other members of the galectin family, but also to galectin-1, and blocks galectin-1 effects, galectin-1 induced angiogenesis and galectin-1 induced immunosuppression. What was really interesting was when we used a model of Kaposi sarcoma, which is highly pro-angiogenic, this antibody could block tumor growth by reducing angiogenesis, but also it acted at the molecular level because it inhibited phosphorylation of VEGF receptor 2. It prevented resistance of lung tumor cells to anti-VEGF. So when we combined anti-VEGF plus anti-galectin-1, tumors that were resistant became sensitive to anti-VEGF treatment. And this effect was accompanied by an increased influx of CD8 positive T cells in the tumor microenvironment. So in summary, in the tumor microenvironment, galectin-1 can promote tumor immune escape in a wide range of tumors. When we explored the possible mechanisms, we found that it can induce selective death of Th1, Th17, and CD8 positive T cells by a glycosylation dependent mechanism. It can deliver tolerogenic signals to dendritic cells, but also to myeloid-derived suppressor cells. And in these particular cell types, it combines the pro-angiogenic, which induces both pro-angiogenic effect and also immunosuppressive effects. It can favor expansion of regulatory T cells, both CD4 positive regulatory T cells and CD8 positive regulatory T cells, promotes polarization of M2 macrophages. I didn't speak about this work. It can link tumor hypoxia to angiogenesis, and activate VEGF receptor 2 signaling and preserve angiogenesis in anti-VEGF resistant tumors, but also can link commensal microbiota to tumor promoting inflammation. I also didn't speak about this work, but in general, galectin-1 behaves as a villain that can suppress immune response, promote angiogenesis, promote metastasis, and increase tumor growth. So, as I mentioned in the first part of my talk, immune checkpoints are also characterized by their ability to promote resolution of autoimmune and chronic inflammation. So I will summarize this part of my talk. We found from our very early experiments that protein or genetic delivery of galectin-1 can suppress inflammation in different models of autoimmune disease. So the other side of the coin, the yin and yang. So it has a detrimental effect in the case of tumors, but a beneficial effect in the case of different models of autoimmune diseases. This is our first model that we studied, rheumatoid arthritis. Then we confirmed these findings in multiple sclerosis, in uveitis, in orchitis, in diabetes. And this is summarized in the review we recently wrote for Trends in Molecular Medicine. And so we wonder if galectin-1 is an immune checkpoint that suppresses autoimmune diseases, then we needed to demonstrate whether galectin-1 knockout mice can have a spontaneous autoimmune phenotype, and was recently demonstrated by Marta Toscano and Veronica Martinez-Aló from my lab. So they demonstrated that when mice get older, they developed a spontaneous age-related salivary gland autoimmunity through glycosylation-dependent galectin-1 immune inhibitory circuits. So particularly, they demonstrated a Sjögren-like disease and a lupus-like disease in these mice when they get older. But what's the role of galectin-1 in the central nervous system? So in a model of multiple sclerosis, we found that during the resolution of the disease and in response to TGF-beta and IL-4, astrocytes in the central nervous system are the cells that produce the highest amount of galectin-1. Galectin-1 is released from astrocytes in its dimeric form. It can bind to CD45 on microglia, retain microglia on the cell surface, and deactivate microglia. It shifts microglia from an M1 type microglia type to an M12, preventing inflammation-driven neurodegeneration. So galectin-1 is also a CNS glycocheckpoint that suppresses inflammatory response and switch off microglia in the tumor microenvironment during inflammation. More recently, we published a study. It was 2 months ago, in which we demonstrated that this circuit, this on and off circuit, operates during intestinal inflammation using different models of intestinal inflammation. Luciano Morosi and Karina Marino, they demonstrated in my lab that galectin-1 glycan interactions are also important to regulate inflammation in the gut. I would like only to highlight a particular experiment that I like very much. You know that colitis can be induced by adoptive transfer of sorted CD4 positive, CD45 RB high T cells in immunodeficient mice. In this case, we have a high intestinal inflammation. However, when we transfer cells from ST6-GAL1 knockout mice, there is no sialic acid. So galectin-1 can interact, can kill T cells, can promote T cell dysfunctions. These T cells are not pathogenic anymore, and they promote a mild disease. On the other hand, when we transfer C2GNT1 knockout mice, there are no ligands for galectin-1. Galectin-1 cannot suppress T cells, and these T cells are more pathogenic, so they can cause a severe disease. I think this was an important experiment that demonstrates that this on and off circuit of galectins and glycans can operate in inflammatory microenvironments. So again, what's the question now? Can we capitalize on galectin-1 glycan interactions for treatment of autoimmune diseases. But there was a problem. We needed high concentrations of galectin-1 to induce immunosuppression. Galectin-1 is very unstable at sites of inflammation. So we wonder whether we can engineer galectin-1 to generate a more stable protein. So we studied the structure-function relationship. So we identified residues that are very important for oxidation of the protein, but also for resistant to the acidic pH of the inflammatory microenvironment, and also involved in the dimerization of the protein. And we generated what we called 3 super galectin-1 proteins. We patented this, but when we used the super galectin-1 proteins, we needed to use much lower concentrations to induce suppression of autoimmune diseases, and we plan to use this in future translational applications. So the third hallmark that is important as an immune checkpoint is that they contribute to immune tolerance during pregnancy. I will only like to show this slide, that we found that in pregnancy, during the first term pregnancy, galectin-1 is secreted from the trophoblast and promotes immunosuppression and triggers these immunosuppressive circuits. When we analyze pregnant galectin-1 knockout pregnant mice, we found an increased fetal loss in these mice that was rescued when we injected recombinant galectin-1, and also in women with recurrent spontaneous abortions, there is a high frequency of anti-galectin-1 neutralizing antibodies. This was demonstrated in these 2 papers. And the last hallmark that characterizes an immune checkpoint is that it can impair resolution of microbial infections and also limit allergic reactions. So to make a long story short, what we found is that different pathogens, different bacteria and parasites can use galectin-1 to subvert immune responses as a mechanism of immune evasion. This is the case of the parasite Trypanosoma cruzi, which is endemic in Latin America. Also the bacteria Yersinia enterocolitica, and also Chlamydia trachomatis. And in this particular type, in this particular infection, galectin-1 could bind glycans exposed by Chlamydia trachomatis with glycans exposed by epithelial cells and contribute to infection and inflammation. Finally, one of the major questions is how galectin-1 is released, how galectin-1 is secreted, because it has no signal peptide, it has all the characteristics of an intracellular protein. So we took advantage of a sepsis model and demonstrated that gram-negative bacteria can, particularly during the ongoing of sepsis, can be recognized. LPS from gram-negative bacteria can be recognized and activate the non-canonical inflammasome, particularly activate caspase 5, 4, and 11, either in human or mouse, promote cleavage of gasdermin release, and gasdermin promotes pores in the membrane that induce the release of galectin-1. Part of this galectin-1 can be in exosomes, and galectin-1 can act as a danger-associated molecular partner of an alarmin interacting with CD45. So galectin-1 is one of the alarmins that is released during sepsis in response to gram-negative bacteria. So in summary, we can think on galectin-1 as the sweet case of Dr. Jekyll and Mr. Hyde. It can be the bad guy or the good guy, but it always induces the same. It promotes tolerogenic dendritic cells, favors expansion and differentiation of regulatory T cells, dysfunction of Th1, Th17, and CD8-positive pathogenic T cells, and also link tumor hypoxia to angiogenesis. In the case of Mr. Hyde, it can thwart anti-tumor immunity, promote metastasis, and favor microbial infection. In the case of Dr. Jekyll, when it's the good guy, it can suppress autoimmunity, inhibits inflammation-induced neurodegeneration, and I didn't speak about this word, but also control allergic airways inflammation. So because it is a symposium on glycomedicine, I want to highlight that we also explored different the role of galectin-1 glycan interactions in different pathologies. And this is summarized in this slide: in oncology, in ophthalmology, in hematology, gastroenterology, reproduction, neurology, allergy, cardiology, dermatology, and rheumatology. So what I explained today on cancer, autoimmunity, and infections can be also extrapolated to inflammation driven by different diseases in different tissues. And the last slide, what are we doing to transfer these technology programs? We have two transfer programs, two main transfer programs. The first one is immuno-oncology and anti-angiogenesis with the anti-galectin-1 antibodies. And the second one is inflammation and autoimmunity with the super-galectins. We have another programs, but we are focusing on these two programs. First, we have the patents approved. We are doing preclinical studies of safety, toxicity, routes of administration, GM production. We are licensing these patents from our council research, from CONICET, and we just created what we call GalTech, Galectin Technology, a startup from our lab and from our institute. And the most important slide, I would like to thank now to all the members, the current members of my lab, researchers, PhD students, technicians that contributed to all this work, to the former members, to all the former members of my lab, and to all our great collaborators during the past years, and all the agencies, the national Argentinian agencies that supported our work, and the international agencies that helped to generate this work. So I thank you very much for your attention, and I'm looking forward to answer your questions.
Well, thank you, Gabriel. This was really a fantastic presentation. We really enjoyed. I think there are many questions. We don't have too much time. I have one very focused question. I mean, for example, in the case of colorectal cancer, how? I mean, we know that treatments are made according to the subtypes. I was wondering, and take it into consideration the heterogeneity of the tumors, that sometimes they express the terminal cellulation, the alpha-2-6 cellulation on N-glycans, and others do not, or in some cases even they have some heterogeneity. How do you see the next steps to use this antibody for treatment?
Well, thank you very much, Celso, for this question. Well, in fact, what we found when we analyzed different human tumors, but bioinformatics and also in tissue samples, is that galectin-1 is upregulated independently of the mutational status of the tumors. So this allows the possibility to treat these tumors independently of the mutations. You know that anti-PD-1, for example, works only in those tumors that are unstable, that are genetic instability. And in the case, we propose to use galectin-1 because the anti-galectin-1 antibody has both anti-angiogenic effect and also immunomodulatory effects. So we think that the effects that we have in tumor progression will be independent of the mutational status, because all colorectal tumors promote immunosuppression and all colorectal tumors promote immunogenicity, or most of them. So we plan, of course, to use as biomarkers of the response both galectin-1 expression and also the expression of glycans, so alpha-2-6 sialic acid, beta-1-6 and glycan branching, and core-2 glycans.
Thank you, Gabriel. There is a question here also that, do you know if core-4 glycans in the intestine bind galectin-1 as well?
Well, core-2 are the main receptors, so LACNAC ligands on core-2 are the main receptors, and LACNAC ligands on beta-1-6 and glycan branching. But there are reports that they can also bind to core-4. And also to core-1, for our surprise, because they have lower affinity, but they can also bind to core 1 and also to alpha to 3 sialic acid. So only alpha to 6 sialic acid can restrict galectin-1 binding.
Okay, 10-5 is also asking, well, now I lost the question, but she was asking if you can target specifically galectin-1 treatment for, I guess, for the proper site.
Yes, well, in fact, you know, when we started to work with different inhibitors, we first tested glycoamines, but the problem is that glycoamines, as I explained, bound not only galectin-1 but also other members, galectin-3, galectin-8, and we know now that they can, galectin-3, for example, can have pro-inflammatory effects, and also galectin-8. So the good thing about targeting galectin-1 is that galectin-1 is anti-inflammatory and contributes to tumor progression. So there is no way to target other galectins. So the best way is to use antibodies, blocking antibodies similar to PD-1 or similar to those used for CTLA-4, and we are now trying to put into nanoparticles. So we are now using nanoparticles to target anti-galectin-1 antibody, the whole antibody, and also single chain anti-galectin-1 antibodies for treatment of different tumors.
Very well. I don't know if there is any, but there's one more question here.
A quick question. Gabriel, it's very, very nice, of course, and I would like to ask you whether this strategy, anti-galectin-1, has been used in the case of cancers which are in pseudo-hypoxia, like when there is a mutation in the VHL, like in the renal cancer.
Well, this is thanks, Claudine is asking the question, no? Yes.
Yes.
Thanks, Claudine. Thanks so much. Yes, well, in fact, we have now a project that is being performed by different students in my lab, Camila Bach, Montana Mansell Coco, and also Marco Scheidegger, using renal cancer. So we are using renal cancer also because in renal cancer there is a good opportunity, because you know that we need a combination of anti-angiogenic and also immunostimulatory agents. So there is people are using a combination of anti-PDL1 plus anti-RTK inhibitors. So we think that the anti-galectin-1 antibody will have 2-in-1. So two possibilities also to block angiogenesis, pathologic angiogenesis caused by hypoxia, and also to immunomodulate. So we think that renal cancer is a great opportunity to start. In fact, we are discussing with clinical oncologists, with Galtech, which is the best tumor to start: colorectal cancer, renal cancer, glioblastoma. So because all the collection of different tumors in which we showed in preclinical models that the anti-galectin-1 antibody works.
Yes. Of course.
Has galectin-1 any chemoattractant effect?
Chemoattractant?
Yes.
Yes. This is a very good question. In fact, we didn't explore particularly, but we found in all our models that galectin-1 induces influx of different cell types, and also there is work performed by Victor Tixen from Amsterdam, and also from other scientists, demonstrating that galectins can also act as chemokines. In fact, if we have to define galectins now, that galectins can be cytokines with glycan binding activity, chemokines with glycan binding activity, or alarmins with glycan binding activity. So we think that we can extrapolate the concept of galectins that was previously published.
Thank you so much.
Okay, so we thank you very much, Gabriel, and I hope next time it's in presence. And thank you very much.
I miss all of you, so I want to travel to Europe. Celso, Pauline. So I feel like you are part of the family. So I hope to have the chance to travel to Europe or next year or in 2 years after the pandemic's allowed. We have a problem that we are vaccinated with a Sputnik vaccine, so we are not allowed to travel. We are a little bit here trapped.
Okay, so we thank you again, and we move to the very last session. I think I can say the few words from here. Maybe I can move to here, and I just wanted to, in this last very closing moment of this beautiful day, we have first. I would like to thank the scientific committee and especially Ghislaine for this fantastic day, this Jenner online or hybrid meeting, which was really fantastic, and the presentations were really amazing during the day. And yes, and then we have to move for the next slide, please. And I have to announce that, well, actually it was mentioned at the opening moment that the next Jenner Glycobiology and Medicine Symposium will be in Porto, in Portugal, in June 14, 16, in 2 years, in 23. And the next slide, I just wanted to show you that Porto is very easy to reach from Europe. Everywhere in Europe you can travel almost directly, and even sometimes you can go from the US or other of South America, for example. And Porto is very easy to get from the airport downtown, and very modern facilities. But the next slide, please, shows a little flavor from the city, from the old part and from the modern part, which is a very historic old city in the Atlantic facing country in the south of Europe. And so please book in your agenda for the audience.
But you have to remember to get off the train when you go to Porto, because it doesn't stop there.
Yeah, no, but this is the one that comes from, that's the one that comes from Lisbon, right? Yes. So don't continue.
It's a long journey back, especially when there's a cycle race.
Yes, but the metro station from the airport is very easy to go downtown. It's half an hour and it's almost direct. So, and yes, I think we can move to the next, just to remind you again to book in your agenda in 23 the 15th Jenner Glycobiology and Medicine Symposium. And again, thank you very much all here and all the attendance online. Thank you very much.
Good day's work. Overtime.
Good though, not too bad.
We've earned our glass of wine.